afternoon, everyone. On behalf, ladies and gentlemen, on behalf of the Executive Committee of Research, Executive Section of Research Committee of Indian Association of Pediatric Surgeons, I welcome you to the 6th Annual Conference. Now, I request the Chairman, Professor Mahajan, to say a few words as the Chairman of Research Section of IAPS.
He's my boss.
Thank you. It gives me a great pleasure to welcome all the distinguished faculty and delegates from India and around the world for this 6th annual conference of
There is another meeting.
Indian Association of Pediatric Surgeons. I especially welcome many senior members of IAPS who have joined us today. I thank you all for registering for the event. The EC members of research section have taken great pains to chalk out the scientific program, which will be introduced to you later by our secretary, Dr. Shilpa Sharma. I express my sincere gratitude to our national faculty, all of whom are leaders in their respective fields, for joining us. They will deliver lectures on many aspects of basic and clinical research, as well as innovations and artificial intelligence, both today and tomorrow. The Executive Committee of Research Section of IAPS extends a warm welcome to international faculty, Professor Agostino Piero, Professor Eric Skarsgård, Dr. Augusto Jani, Mr. Ashish Desai, Dr. Mahmood Al-Fiqi, Mr. Khizar Mansoor, and Dr. Ashish Bhardwaj, who have very kindly accepted our invitation despite being in different time zones. You will be listening to some of our international faculty today evening itself. We also welcome office bearers of Indian Association of Pediatric Surgeons who will join us during the deliberations. I am thankful to all of them for their support for this meeting. I hope you will enjoy scientific deliberations over next 2 days and get stimulated to engage in meaningful research. Thank you all. Now I request Dr. Shilpa Sharma to take over.
Thank you, sir. Good afternoon, everybody, ladies and gentlemen. On behalf of the EC of Research Section, I extend a very warm welcome to all of you to attend this virtual 6th annual conference of the Research Section of Indian Association of Pediatric Surgeons. We've had an overwhelming response this time with 274 registrations till 11:30 this morning, with 60 international delegates spanning over 90 countries, We welcome all delegates from India and abroad. This 2-day program covers sessions on advances in basic research, clinical research, innovations, and artificial intelligence. In the first session today, we have talks by Professor Rima Dada, clinical research from Professor Praveen Mathur, basic sciences on stem cells from Dr. Shio Mohan, and clinical research collaboration from Dr. Dhruv Ghosh. I now invite Professor P.K. Jana, our executive member representing the eastern part of India, to kindly coordinate the first session. IT, please unmute Professor Jana.
So I welcome everyone to this meeting. Good afternoon from India. I welcome everyone from India and abroad to this meeting. So without much wastage of time, let me introduce. There are 2 speakers. There are 2 chairpersons for this afternoon session who have been my teachers, Professor N.C. Bhattacharya and Professor Bireshwar Bhatnagar. Everyone knows that Dr. Bhattacharya is a retired professor and head Department of Pediatric Surgery at Guwahati Medical College, and he retired as principal of Tezpur Medical College. Presently, he is the medical director of KGMT Multi-Speciality Hospital in Guwahati. And Dr. Bireshwar Bhatnagar, he has been my direct teacher in AIMS when I was there, and he was Professor and Head Department of Pediatric Surgery at AIMS, New Delhi. At present, he is Dean, Research and Professor of Pediatric Surgery at Sharma University School of Medical Sciences and Research at Uttar Pradesh. I welcome both the chairpersons to start the proceedings. Thank you.
Dr.
IT, please unmute Bhattacharya sir. Bhatnagar sir is just joining.
I can see Dr. Bhattacharya has joined, but Dr. Bhatnagar, whether he has joined?
He will just join because he thought it's 3 p.m. sharp. He's here. Just 2 minutes more. But Bhatnagar sir will introduce Rima ma'am.
Okay.
Hello, Bhattacharya sir. In the meantime, you can say a few words on research section. Please unmute Bhattacharya sir. IT. Bhatnagar sir has joined. So please introduce Rima ma'am.
Good afternoon. It's my pleasure to introduce Dr. Rima Dada. I have known her for years and years. At the moment, she is working as a professor in the Department of Anatomy at the All India Institute of Medical Sciences in New Delhi. She has established a molecular reproduction and genetic facility in her department. And she is very actively engaged in both teaching, research, and genetic diagnostic services. She has a fellowship of a lot of bodies, reputed bodies like the National Academy of Medical Sciences, the International Medical Academy, Academy of Standards and Ratings, and the Indian Academy of Biomedical Sciences. She has received a number of awards. I won't go into the list of all the awards that she has received. She has published extensively, 170 odd original articles, 65 chapters, and she has a membership of many scientific bodies. So at the moment, Dr. Rima Dada is pursuing research on genetic aspects of infertility, reproductive failure, recurrent ART failure, and idiopathic recurrent spontaneous abortion and childhood cancers. In the last 8 years, she has been working on impact of yoga on the genome and the epigenome. And that is why I think she is just the perfect person to talk on the paternal factors and embryonic development and the role of yoga. Over to Dr. Rima Dada.
Thank you, sir. Can you see my slides?
Yes.
Good afternoon, everybody, and I will be talking to you today about paternal factors and embryonic development and the role of yoga. Just 1 minute, this computer. क्या प्रॉब्लम आ रही है?
You can try to reshare. They had come and then they went away again. So just try to reshare. Let's see.
Yeah. Now it is
moving forward. That's the problem.
Some problem was there, huh?
Yes, sir.
So shall we start now?
Yes, sir. Bhatnagar sir has introduced Reema ma'am.
Yeah, sure.
She's
Should I, instead of just 1 minute? ऐसे क्यों हो रहा है? I'm sorry, there's some problem here with the net and it's hanging again.
you can send me your slides. I can share from this end if it works.
Just 1 minute. Should I show it like this? I think it's better rather than putting it on the presentation mode. I think there's some problem. Is it okay?
That's fine. Yeah, it's fine. You can show on the,
Fine. Okay.
but we can't see your slides as of now.
You
Share
can't
it.
see this?
Yeah.
Just 1 minute. You share कैसे करेंगे? Just 1 minute. Can you see them?
Yes, we can.
So I'll just do it in this mode because otherwise it's not moving forward. अभी भी अटक रहा है.
Fine, no problem.
Okay, okay. Okay, so today I would be talking on our sperm year vectors determinants of embryonic viability and developmental competence. I would also highlight that sperm is a cell in crisis and why it is most vulnerable to environmental insult. And because of that, it has over a period of time accumulated a huge number of mitochondrial variations and nuclear DNA damage. And this is the reason for a number of genetic and epigenetic diseases which determine the health trajectory of the offspring, and how a holistic mind-body intervention like yoga can positively impact the genome and modulate the epigenome. So first, I would like to introduce a few concepts like genetic determinism. So we are brought up with this paradigm that the genes determine our destiny. And we inherit a set of genes from our father and our mother, and they determine our biology, our health and disease states. However, it is important to know that though we are built, we have this concept of genetics that any change in the nucleotide variation only can bring about a change in the expression of the genes. Now we know that over and above the genetic landscape, we have the genetics of free will, or the epigenome, or the epigenetics, which states that we can modulate the expression of our genes or the output of the genes by our thoughts, by our lifestyle, by our habits, by our diet, by our habits, and whatever we think. And that is really empowering because it actually shows us that we can modulate the output, the behavior, or the expression of our genes by our free will. And this is known as epigenetics. And today I would be focusing on how various factors can modulate the epigenome and also the genome and cause variations in the epigenetic landscape, and thus result in causing a number of diseases in the offspring. So it's also important to understand that genes are marked by our experiences, because whatever we think, our lifestyle, our social habits, our diets are going to determine the output and the are going to determine the output and the behavior of our genes. It's very, very important to understand that good habits switch on good gene programs, whereas unhealthy habits, for example, fast food, switch on gene programs which are detrimental to us. I would also be just touching upon that we already are brought up with this concept of developmental origin of health and disease, which states that what happens in pre-pregnancy, in utero development, and during the early years of life actually set the stage for various diseases and health states which the child experiences. It's also important to understand that now there is a great emphasis on studying the paternal factors which determine health and disease. Why it's important to study the paternal factors is because it's now believed that the sperm genome, unlike the oocyte genome, cannot correct the damage it accumulates. And this is very, very important because we see that the sperm is not equipped with the DNA repair machinery, and it's the cell which is most vulnerable to various environmental factors, pollutants, oxidative stress, and various environmental stressors. So there are a number of studies, and now it highlights that biological parenting begins much before conception. It begins when parents themselves are conceived. So our health and disease states are not only determined by the health and disease of our parents, but also our grandparents, in whom these germ cells are formed when our parents themselves are conceived. And so though it is important that we do hear that couples really improve their lifestyle, start having folic acid, shift to a vegetarian diet just prior to planning to have a child, but it's important to know that their germ cells accumulate all their marks of different experiences, what they eat, the experiences they have, the choices they make, all leave a mark on the germ cells. And these different time points are connected by various chemical modifications which occur in the DNA, like methylation marks, acetylation marks, which are then transmitted to the offspring, and they have transgenerational effects. So our diet, our lifestyle really impacts the output and the expression of our genes. So today I would be focusing on the paternal factors, and there are numerous studies now which highlight that the paternal factors should not be ignored. And the paternal factors play a major role, if not more than the maternal factors, in modulating determining the development of the embryo and also in the health and disease states. As I've been working on genetic aspects of infertility, recurrent pregnancy losses, both after spontaneous and assisted conceptions, and then idiopathic congenital malformations, we've been studying the paternal factors. And the first studies which actually highlighted that the paternal factors play a major role were these, that when male mice were made to smell cherries and they were given an electric shock, it was seen that several generations downstream, when their pups were made to smell cherries, they shivered without any electrical current or stimulation being given to them. So this showed that every experience that the father had is being transmitted to the offspring. And these are then transmitted by non-genomic pathways, which are by chemical marks. There are different studies which show that various environmental factors, diet, lifestyle, for example, when you smoke, you consume alcohol, or you have an unhealthy lifestyle, all these factors modulate the epigenome, and there are different time points when the germ cells are most vulnerable to these environmental factors. So when we know that when the germ cells are formed, there are waves of reprogramming of the epigenome, but then there are certain imprinted genes. Now, imprinted genes are those genes which are showing monoallelic expression depending upon the sex of inheritance. And what's important is that these imprinted genes, which are clustered on chromosomes 11 and 15 and certain other chromosomes, these are critical for early embryonic development. And these imprinted genes are very, very susceptible to environmental insults, and they maintain their epigenetic marks. So whereas other regions of the genome undergo extensive reprogramming, however, it is seen that the imprinted genes do not undergo reprogramming, and these retain their epigenetic marks, which have accumulated because of various environmental stressors. So there was a very big study which was conducted, which showed the influence of the environment on the gametic epigenetic reprogramming study, which showed that when you have a diet which is unhealthy, high in trans fats, salts, and sugars, it causes aberrant methylation of these imprinted genes, which can adversely affect the embryonic development. In addition to that, it's important to know that though we analyze the maternal genome in great detail, and we analyze various maternal factors when there are cases of infertility, recurrent pregnancy losses, abnormal embryonic development, which can result in early and late pregnancy losses. In addition to that, when you have congenital malformations, we have basically focused our studies on those cases with idiopathic congenital malformations or abnormal embryonic development. And we find that very few studies actually have done the work on the male gamete. And it's important to know that the sperm is just not a mere vector of paternal DNA, but at the time of fertilization, it transmits the oocyte activation factor, the centrioles. In addition to that, it transfers a huge number of transcripts, which are very vital for early embryonic development before the activation of the zygotic genome. And in addition, it transfers some of the microRNA, which are going to regulate the expression of various oocyte genes. In addition to that, it's also important to understand that the various transcripts which are transmitted by the sperm at the time of fertilization help in early embryonic development, which are known as the early paternal effects, which occur prior to the activation of the paternal genome. Then there are late paternal effects, which occur after there is activation of the paternal genome. And these are very vital. So the optimal dosage of the sperm transcripts, the microRNA, are critical for these early and late paternal effects. In addition to that, it's very important to understand the structure of the sperm, because this is the only cell which is not equipped to handle the various environmental insults, because during spermiogenesis, it sheds majority of its cytoplasmic antioxidants. And thereby, whenever there is oxidative damage, or oxidative stress, it cannot protect itself unlike other cells. In addition to that, we find that the sperm genome is highly inert. It has two parts. The outer part, which is bound by histones, which contain genes of promoters of genes of developmental importance. So it is actually coding for the transcripts. And then there's an inner portion, which is the one which is bound by protamines. Since 85% of the sperm DNA is bound by protamines, majority of its repair mechanism is transcriptionally and translationally silent. So unlike other cells, when the sperm experiences damage, it is not able to clear the damage. It has just certain genes by which it is able to detect the oxidative damage, and then it is able to remove that base, but it is not able to clear the damage completely. So it is actually dependent upon the oocyte to clear the complete damage that it accumulates. In addition to that, the peripheral portion of the sperm genome also bears the telomeres, which are very, very vulnerable to oxidative stress. So the most important thing to remember is that sperm is the only cell in the body which is not equipped to handle the oxidative insult. In addition to that, its repair machinery is largely silent and is dependent upon the oocyte to clear its damage. And both the oxidative stress damages both the mitochondrial as well as the nuclear DNA. So from our studies, we found that advanced paternal age, various lifestyle factors like smoking, alcohol consumption, environmental toxicants, psychological stress, anxiety, depression, excessive use of cell phones, air pollutants, all results in oxidative stress. This oxidative stress damages both the nuclear and the mitochondrial DNA, resulting in shorter telomeres, that is accelerated aging, the testicular aging. It results in accumulation of this base, which is 8-hydroxy-2-deoxyguanosine. I'll mention it again because this base is pro-mutagenic. It results in de novo germline mutations, which is responsible for cancers in the fathers, a higher incidence. And in addition to that, since the damage is very extensive and there is advanced maternal age also, there is an aberrant repair machinery in the oocyte, this base process post-fertilization, resulting in a higher rate of post-zygotic mutations. And in addition to that, the presence of this base actually also results in epimutations or dysregulation of the epigenome. And in addition to that, there is fragmentation of the DNA. So we see that when there is nuclear and mitochondrial damage, it is the cause for not only infertility, but when you bypass these by mechanisms like assisted conception, or sometimes there is spontaneous conception, it is actually the cause of congenital malformations, recurrent spontaneous abortions, neuropsychiatric disorders like autism, childhood cancers, and dominant genetic diseases. It's seen that 75% of de novo germline mutations occur in the sperm genome as compared to the oocyte genome. And there are various studies which have now shown that as we are delaying the age of marriage and having children later, there's an increased incidence of spontaneous abortions and various complex neuropsychiatric disorders which are correlated with the paternal age. And the oocyte is only able to repair 40% of the sperm damage. And therefore, this damage tends to persist. And there are various studies which have shown that accumulation of damage and the most important cause of damage, there are numerous factors, but the most important cause of damage in the sperm is oxidative in nature. And this is a landmark study by Dr. Aitken, who's considered the father of modern andrology, which showed that there are 9,000 loci in the sperm genome which are vulnerable to oxidative damage, which are consistently damaged when there is oxidative stress. And they found that this damage is proportional to the size of the chromosomes. But there is one chromosome which is most vulnerable to damage, that is chromosome 15, and that is the chromosome. And in that region, which is having the imprinted genes, there is maximum damage. And that bears the sites for infertility, some genes which are responsible for maintaining fertility. So that is getting oxidized. In addition to that, that has a cluster of imprinted genes and genes which are important for neurodevelopment. So these are sites which are consistently found to have oxidative lesions. In addition to that, it's been found that because of oxidative stress, there is aberrant methylation. And we find that there are a large number of methylation defects in couples who experience pregnancy losses, or there is abnormal embryonic development. And I would be just showing you that how yoga can positively impact these factors. In addition to that, when couples are infertile, more and more couples are opting for assisted conception. And it's very important to understand that when you are actually culturing these germ cells in media, they do experience oxidative stress. And since the environment is different from the actual environment, a higher incidence of genetic and epigenetic diseases are found in children who are conceived by assisted conception. For example, this article which appeared in the New England Journal of Medicine shows that infants who are conceived by intracytoplasmic sperm injection, these are the cases which have very poor quality sperm DNA. They have twice as high risk of major birth defect as compared to other infants who are naturally conceived. And in addition to that, there is a much higher incidence of methylation defects in children who are conceived through assisted conception. Then there are numerous studies which shows that fathers' habits, as I told you, that conception begins much before the child is actually conceived, and it actually depends upon the lifestyle of the parents. And you can see that when the fathers smoke, it has been seen that their children are found to be obese. In addition to that, alcohol consumption also results in differential aberrant methylation. In addition to that, it's been seen that smoking also causes different levels of microRNA, which are actually the regulatory RNA. In addition to that, it's been seen that both maternal and paternal stress and anxiety actually can impact the neurodevelopment. In addition to that, it's been seen that the microRNA profile, which is also, as I told you, it's regulatory in nature, can also result in obesity. And in addition to that, when parents lead a healthy lifestyle, especially the father, it can actually cause reversal of these marks and result in the birth of a healthy baby. So in our studies, we found that there are some cases in which the people are actually having normal sperm counts. There are cases with normospermia. So they are normal as per WHO guidelines, but yet they have high levels of oxidative stress and DNA damage. And these are basically due to a large number of environmental factors like water, which is leached with endocrine disrupting chemicals, smoking, alcohol consumption, excessive use of cell phone, a diet rich in trans fats, salts, and sugars, high consumption of non-veg food, which is having endocrine disrupting chemicals. And through our studies, we've actually quantified the free radical level, and we find in cases with congenital malformations, a large percentage of men, and even cases with recurrent spontaneous abortions, we find that a large number of men are having seminal oxidative stress. And since mitochondrial DNA are the source and target of free radicals, we find that in these cases, for example, we have chosen this infertile population because these are the ones who go in for assisted conception, and a higher incidence of genetic and epigenetic variations are found in these cases. So you can see that the number of variations found in these cases with unexplained infertility is much higher as compared to the men who are the fertile controls. They also have lower antioxidant capacity and much higher levels of DNA damage. So in these cases, you actually find that there is lower ATP levels, because of which there is abnormal sperm morphology and also abnormal motility because of abnormalities in the axonemal apparatus. But in addition to that, they have lower ATP levels and higher free radical levels. And higher free radical levels not only results in DNA oxidation, it results in A basic site, single double strand breaks, and DNA fragmentation. But one of the major impacts of oxidative stress and the huge number of factors like advanced paternal age, be it unhealthy social habits like smoking, alcohol, diet high in trans fats, salts, and sugars, stress and anxiety all culminate in oxidative stress, which also dysregulate the epigenome, resulting in global hypomethylation as well as locus-specific hypermethylation. And when there is global hypomethylation, it results in unmasking of repetitive elements, which then actually results in complex chromosomal translocations as well as genomic instability. So in our study, we also quantified the levels of damage in these people, and you find that by comet assay, that is single cell gel electrophoresis, you can see the people who are infertile, who go in for assisted conception and are encountering repeated IVF failures. You can see they have much higher levels of fragmented DNA. You can see these tails are much longer as compared to the controls which have maximum amount of DNA which is localized in the region of the head, showing you good quality DNA. We also quantify the DNA damage by flow cytometry by a technique known as sperm chromatin structure assay. And each dot here represents the sperm, and the number of sperm towards the x-axis are the ones with the damaged DNA. And you can see these people who are encountering repeated IVF failure, the infertile cases where there is a male factor involved, they have higher levels of DNA damage. We also study the telomere length because that's our biological clock. And this is very vital that the sperm have adequate telomere length because that determines the rate of cleavage. And indiscriminate use of antioxidants can result in too low free radical levels, and it is required that there are optimal free radical levels to maintain the telomere length. We also quantify the level of this base. As I told you, this is the base which is both pro-mutagenic and also induces epimutations. And this is the cause for de novo germline mutations, which correlates with the unhealthy lifestyle like smoking, alcohol consumption, and exposure to endocrine disrupting chemicals. So there are many causes of DNA damage. The chief among them are oxidative stress, abortive apoptosis, that the cell begins apoptosis, but it's not completed. So such cells persist with DNA damage, and this is because of dysfunctional mitochondria and testicular maturation defects. So we quantified the levels of DNA damage beyond which a couple cannot conceive spontaneously. We also quantified the levels of DNA damage when a couple conceives, but they have the congenital malformations or they experience recurrent spontaneous abortions. And as at the time of fertilization, they also transfer numerous transcripts of great developmental importance. And you can see that in these cases, as compared to the controls, you find that the sperm transcripts levels are highly dysregulated, and therefore this impairs the early embryonic development. So we published these papers where we showed that a large number of sperm transcripts which are critical for early embryonic events are dysregulated. And by practice of yoga and meditation, you can normalize the levels of these dysregulated transcripts, which I'll just show you. In addition to that, we also see, as I told you, that since the major damage is oxidative in nature, and this is the base which induces both mutations and epimutations, the accumulation of this base, we actually studied fathers of those children who had sporadic retinoblastoma, because there were studies which showed that fathers who smoked and cases who went in for assisted conception have a higher incidence of children with certain cancers, especially retinoblastoma. And we find that this is the base which is actually resulting in de novo germline mutations and epimutations. And we also found the persistence of this base in the children who had retinoblastoma versus the healthy children. I would just like to touch upon congenital malformations. And in our study, we again found that in these cases, these are idiopathic cases where there were 2 or more children with congenital malformations. Again, we found high levels of DNA damage correlating with the oxidative stress. This is the study we did on retinoblastoma. So we actually excluded those cases where there was a positive family history and only included those cases in which there was a negative family history. These cases were sporadic, and we actually found that the seminal oxidative stress and DNA damage is much higher than the control levels. And the other important thing that we found was that the children who had sporadic retinoblastoma, they were usually the last born child when the father had advanced paternal age. So that means that they had accumulated high levels of DNA damage. And there are studies which have shown clearly that the incidence of abortions and autism actually increases with the paternal age. And we've done this study along with Dr. Shilpa, where we studied this autism spectrum, this disorder, and we correlated with the DNA quality, and we found that a large number of genes which are regulating developmental processes, synapse formation, sensory motor conduction, and inflammatory pathways, the genes regulating these pathways were dysregulated in these men who had these children. Now I would just talk about the impact of yoga on the sperm genome and the epigenome. And it's important to know that post-practice of yoga, we found that there was a significant decline in the free radical levels. These were randomized studies, and you can see that post-practice of yoga, there was an increase in the levels, the total antioxidant capacity, because yoga actually resulted in upregulation and increased expression of genes which are coding for various antioxidants. Now, the WHO recommends that we should be having 5 servings of fruits and vegetables to combat oxidative stress. And the Physicians Committee for Responsible Medicine says that we should be having 10 servings of fruits and vegetables. But our studies have shown that if you practice yoga, you can show there's a significant increase in the total antioxidant capacity. We also did a study where we found that when you take antioxidants, a large number of antioxidants at therapeutic levels, though they bring about improvement in the sperm membrane integrity, so the motility improves and their fusibility and fertilization capacity improves, but it has little impact on the DNA integrity. It's because the DNA is highly compact and it's not able to penetrate at that level, though some antioxidants do. But when you use higher dosages of these antioxidants, it actually causes it actually causes damages and impairs various physiological processes like capacitation and acrosome reaction. So yoga causes an upregulation in the total antioxidant capacity. You can see here that the base which induces mutations and epimutations show a significant decline post-practice of yoga. The dysregulated transcripts, when the individuals start practicing yoga, they start normalizing over a period of time, and genes which are helping to maintain integrity, the genes for 1-carbon metabolism, which are required for synthesis of S-adenosine methionine, which is required for the replenishment of glutathione, as well as for DNTP synthesis, show a positive fold chain. In addition to that, as I told you, that oxidative stress causes a dysregulation of the epigenome, causing genome-wide hypomethylation and affects the imprints. We see that post-practice of yoga, there is a significant increase in the methyl cytosine levels. That means there is re-establishment of the epigenetic marks. And whereas the group that did not practice yoga did not show any change. Oxidative stress results in the formation of hydroxymethyl cytosine because of oxidation of the methyl cytosine bases. And we can see post-practice of yoga, there's a significant decline in the level of hydroxymethyl cytosine. In addition to that, this was the first study to show, so it was a pilot study, that yoga can actually impact the sperm epigenome, and there is an increase, it targets a large number of areas, and some genes, actually you cannot read it because I've not put it in the presentation mode, a large number of genes which are helping in play an important role in spermatogenesis, embryo implantation, which are helping to maintain the telomere length, helping in cytokinesis, are hypomethylated, showing that they are increased in expression. And this correlates with a decline in the DNA damage and a decline in the oxidative stress. In addition to that, we have done studies, though this was done in rheumatoid arthritis, but what I wanted to show you was that yoga is also impacting not only the nuclear genome, but it is also acting on the mitochondrial genome. And this was the first study again to show that yoga can improve the mitochondrial integrity. There's an upregulation in the expression of genes which are helping in mitochondrial biogenesis, which are helping to maintain the mitochondrial integrity. And also there's an increase in the mitochondrial copy number, and a key molecule which helps to maintain the nuclear DNA crosstalk also shows an increased expression post-practice of yoga. In addition to that, you can see that when you practice yoga, the number of these dots, which are the sperms, which are towards the x-axis, has shown a significant decline after 6 months. Though oxidative stress decreases in 10 days, but this DNA damage takes 2 spermatogenic cycles to show significant improvement. And you can see that there's a significant decline in the DNA damage post-practice of yoga. So I would just like to sum up by telling you that paternal factors are critical determinants of embryonic viability and developmental competence, and optimal free radical levels are required for maintenance of genomic integrity of the nuclear and the mitochondrial genome, helps to regulate the epigenome, which is critical for the birth of a healthy offspring. And yoga and meditation decreases the oxidative stress, helps to maintain optimal levels of free radicals, decreases the nuclear and mitochondrial DNA damage, helps to regularize the levels of various transcripts which are required for early embryonic development, and helps to modulate the epigenome. And thus yoga has tremendous transformative power and really can prevent the onset of diseases, and also can help to modulate the sperm genome and epigenome in such a way so that the load of genetic as well as the epigenetic diseases in the offspring is minimized. Thank you.
Thank you, Dr. Dada. I think it was a wonderful talk. And in my mind, it raises a lot of questions. So maybe I'm a little premature in asking these questions, but I think these are things which possibly need further studies. And because I think from whatever you've told us, it should be possible for us to identify people who are at risk of causing congenital malformations. And if so, then possibly we can extend it as a service. I mean, the couples who are desirous of knowing whether they are at risk or not, they could be tested, treated with yoga, retested, and then they should be allowed to try for a conception.
Absolutely, absolutely. Actually, these studies are going on for the last 7-8 years, and we are having regular patient inflow from the ART center, IVF and andrology clinics, and many of them conceive spontaneously because the DNA quality improves. So, but the thing is that men are not aware, and initially all of us, they waste so many years just because the women is being investigated, and it's the last resort they come to get investigated. And they are very resistant to even believing that the problem is in them. And then you have to motivate them to come for yoga. That's another problem. But we found that when they are doing yoga under our supervision, we have a very good yoga instructor. Their results are much better than these people doing yoga at home, because then they do it watching the television, then the effect is not that significant as compared to when they're doing yoga under our supervision.
Another thing which I wanted to ask you is that with all this information available to us, is it not possible to go in the reverse direction? I mean, look at people who've had congenital malformations in their children, look at the genomes and identify specific genomes for specific congenital malformations, and see if yoga or some similar things can actually modify the genome before a conception takes place.
Absolutely. The study that we have done in autism, we went like this. We actually studied the sperm DNA of fathers who had an autism child in the last 3-4 years. And then we told them to do yoga to quantify the DNA damage. So there was a significant decline in the DNA damage. So definitely there's an improvement, but we need to really increase the awareness so that people actually, realize that it's not just 3 months before conception that you start following a healthy lifestyle, and that every experience and choice you make is impacting your genome so profoundly. And I think the men need to be really aware, because though the oocyte is going to correct the damage, and oocyte is able to really protect itself from the oxidative damage, but the sperm does not correct the damage. So it's really that men should be aware and beware, regarding smoking, alcohol, a diet rich in trans fats, salts. It is so lethal for the sperm because it's not able to clear the damage.
Right. So there was a question for you in the chat box, but I think the latter part of your talk answered that query. That question was from Shilpa. So that you answered in the later part of your talk. So if there's any other query or any other interaction which people might want?
Yes, sir. I am Dr. Javle. My question to Rima Madam is, I have a therapy called Lifetron therapy, which is basically an antioxidant therapy. It pumps electrons in the human body and reduces oxidative stress. Now, I want somebody to collaborate with me for the objective evidence, like you have DNA damage and everything. So if you can do that, we will have a revolution. If you want, I'll send that published article to you, and the machine also. I can give the machine free of cost, and we can do a joint study.
So I'll discuss with you later, definitely.
Yes, ma'am.
So any more interactions? I think I'll let the moderators and the organizers decide how much time we have for discussion. And
another 2 minutes are there. So there is one question in the chat box regarding use of yoga when pregnancy has already started. Yoga and antioxidants when pregnancy is already started. During that time, is there any role of yoga?
Definitely, our yoga instructor does teach them some prenatal, some exercises to be done. And definitely, it has been seen that it enhances the blood flow in the uterus. And we've done numerous studies in other complex disorders also, like in glaucoma, depression. It shows that it increases blood flows to various centers in the brain, promotes neuroplasticity. So it is going to have an effect. And I also see a question in which people are saying that how do you know it's the effect of yoga and not diet and improvement in lifestyle in general? What I would like to say is that we have only selected those cases in which there has been no recent change in lifestyle. But it's important to know that yoga is just not on the mat, it's an entire lifestyle. Yoga encompasses the lifestyle. It's not just asanas, it's both regulated breathing practices as well as meditation. And it encompasses healthy eating, whole plant-based foods, and that they are all counseled about. So it's entire lifestyle, and you basically, just like you practice dental hygiene, we need to integrate yoga into our lifestyle.
But, carrying this question further, if fertilization has taken place with a defective sperm, how is yoga going to affect the abnormal fertilization? That has already happened.
Yeah, so that's the problem, because when you are taking antioxidants, you're actually improving the sperm membrane characteristics, and DNA does not impact the fertilization capability. But yes, what happens is yoga actually upregulates the DNA repair mechanism. So it is going to enhance, though definitely the damage once is there, is going to be found in every cell post-fertilization, predisposing the embryo to genetic and epigenetic diseases. But definitely it will upregulate. We have seen in our studies, it upregulates the repair mechanisms. So the damage is going to be less. We've quantified the level of expression of various genes which are helping to maintain the genomic integrity.
Yeah, okay.
The one more comment I wanted to add is, when you do yoga, your peripheral stem cells also get mobilized into the bloodstream. So we've done studies in which C34 count increases in your bloodstream when you do yoga. So that may also be an added factor, how it works.
Right, absolutely, absolutely.
Thank you. Thank you, ma'am. Thank you, ma'am, for that wonderful lecture. And on behalf of Organizing Committee, I want to express my gratitude, and Organizing Committee has prepared a little memento for you. That is online memento, basically. I hope it is visible.
Yes, sir.
Yes, sir.
Yeah. So thank you, ma'am. Grateful.
Thank you.
So maybe you can send it online and she can make a printout at home.
Yeah, sure, sir. Sure, we will do that.
Thank you so much.
So, Dr. Jena,
That was a wonderful session actually, and really eye-opening that yoga can affect so many things. That's why our forefathers were practicing yoga. So
can we have a few words from Bhattacharya sir, if we can be unmuted?
Acha, theek. Dr. Bhattacharya, can we like check her? Dr. Bhattacharya sir, unmute yourself.
Please unmute, sir.
So left lower bottom mein ek wahan, yes.
Yeah, no. No, it's a very wonderful talk given by Professor Rima Dada, and I think it's a very good area of research that yoga can change the situation so much. We didn't know it. It's a sort of new knowledge to us that yoga has so much far-reaching effects in the human development. And even it can reduce the incidence of congenital anomalies, not only anomalies, even childhood cancer. It's a very, very wonderful area of research. And I think we are very much benefited by this topic today delivered by Dr. Rima. Thank you very much.
Thank you, sir.
I think this was the perfect opening for a research meeting.
I think so.
Thank you.
Thank you.
So it was a wonderful session. I thank all the chairpersons and the speakers. Though the next section, the chairpersons are Professor Yogesh Yarin and Dr. Shilpa Sharma. Dr. Yogesh Yarin, everybody knows, he is the Director Professor of Department of Pediatric Surgery at Lady Hardinge Medical College, New Delhi. And at present, he is the President of the Indian Association of Pediatric Surgeons. And he has been the past Secretary and Treasurer of IAPS. The second chairperson is Dr. Shilpa Sharma. She is Additional Professor, Department of Pediatric Surgery at All India Institute of Medical Sciences, New Delhi. And she is the secretary and the force behind our research section of IAPS. So now I invite all the chairpersons to chair this session.
I think Dr. Yogesh Yarin will join. He is already joined. So unmute. Ask to unmute.
Yeah, hello. Dr. Mahajan, I'm there. Thank you.
Yes, sir. Yes, sir. Please.
Welcome, sir. Welcome.
Let me introduce the speaker. Just 1 second, please.
Professor Shiomohan.
Dr. Shilpa Singh is the Director, International Center for Stem Cells, Cancer and Biotechnology, New Delhi and Pune. He's PhD in Human Biology, Grossherden Hospital, LMU, Munich, Germany, and as well as PDF in Molecular Medicine, Mount Sinai Medical College, NYC, and UMass Medical School, Worcester, and Leeds Institute of Molecular Medicine. He's worked on hematopoietic stem cells, myeloid differentiation and myeloid leukemia, and embryonic stem cells and induced pluripotent stem cells. Stage is all yours, Dr. Singh.
Unmute.
Dr. Shio, your slides are visible. You can begin.
Dr. Shio, sir, you have to unmute yourself. सबसे ऊपर है. And Dr. Shio, sir, unmute yourself.
Dr. Shio, are you able to unmute yourself? If not, just write in the chat box, unable to unmute. IT, please send the request again to unmute. He will then click that box.
He is sending. He is now unmuting. So, sir,
Can you hear
video
me?
also.
Yes, he's done.
I'll start the video, but then it might stop because of the connection or the internet speed.
Okay.
So if you allow me, I'll just keep the audio on.
Okay. Okay, sir.
So I'd like to thank the organizers and Dr. Shilpa, Sharma, Dr. Yogesh, and everyone. Thank you for giving me the chance to speak. I'll put the slide on full screen mode. Let's see if it works. Seems like nothing is happening when I click on the slideshow.
Yeah, slideshow, yes. That will help, yes. Yes, yes. Now slideshow, sir. We can see.
All right, okay. So I'll talk about stem cells and how they can help in gametogenesis. So they are needed in assisted reproduction technologies and they can help in assistance in patients without functional gametes. So these days, because there are restrictions and lots of rules and regulations to do experiments on humans, so the stem cells can be used for developing model systems or understanding the basic mechanism, how gametogenesis happens and what goes wrong, what mutations happens and disorders. And then it can be used for treatment once proven. So as you know already probably that 10 to 15% of the reproductive population is infertile and 25% of married women suffer from infertility. So this can be for many different reasons, infertility, whether it is due to defective genetic development or genetic disorders, mutations, or by environmental factors like pesticides or other pollutants, or because of disease or disorders for which people are taking treatments like anti-cancer or others. So it can cause the damage, and in vitro, because of these reasons, then we need to develop the treatment. So whether it is stem cell treatment or any other possibilities to develop the functional sperm or functional sites. So what happens in vitro gametic formation, it can be done from gonadal stem cells or pluripotent stem cells like embryonic stem cells or induced pluripotent stem cells. So it's happening in male. It happens from the adult germline stem cell population called spermatogonial stem cells, and in female, oocyte development happens from egonial stem cells. So people have already done a lot of work on these. They've developed cultures and protocols and have understood the basic mechanism, the markers, transcription factors, etc., which are responsible for driving the differentiation process in normal and in under abnormal circumstances, or when mutation happens, then obviously something goes wrong and you can have defective sperms or oocytes. So in terms of stem cell technique or cultures, first mouse embryonic stem cells were cultured in late 70s. So they were quite early done, and the protocols were developed in 80s. And human embryonic stem cells, it took a longer time because it is more difficult to culture. So it was first published in 1998 by James Thompson Group, and that basically revolutionized the field from 99 2000 onwards for human embryonic stem cell cultures, and they were differentiated into almost all the different cell types of the body, including the sperm and oocytes, which is still going on development, because the problem is in making it completely functional without any DNA damage or mouse. So induced pluripotent stem cells, they were developed in 2006, and next year in 2007, human induced pluripotent stem cells were protocols were published. So from 2007 onwards, again the focus shifted from embryonic stem cells to induced pluripotent stem cells, because it also doesn't involve destroying an embryo. So lots of ethical and religious questions that is sidestepped if induced pluripotent stem cells are used, because in iPSCs you can basically take any cell type from the body and convert it into pluripotent stem cells. So there are different mechanisms by which we can make iPSCs, whether it is protein induced or whether it is other methods. So in terms of sperms development in culture, for male it was developed in late 90s and early 2000s, and in female mouse, OSCs were isolated and cultured in 2004 and 9, and human OSCs were cultured in 2012. So now how the recent development in different technologies has also helped in figuring out these mechanisms, how they differentiate and or identifying the mutations, etc. So 2-3 techniques that I have listed. One is FACS, fluorescence activated cell sorting. So it is now possible that you can isolate single cells. So you can isolate single cells in sterile condition and culture them for whether developing sperm or oocyte, or for any other purposes. Then in microscopy, again, we have jumped a quantum leap kind of with the development of cryo-electron microscopy in 2017. So you can actually look now at the molecule level, at protein level, single molecule level, or within the DNA. So I just hope that these machines or this technology comes to India also. For this Nobel Prize was given. In sequencing technologies, again, the things in the 80s or 90s, which used to take years and years of sequencing, it can be done now in days. And in humans case, or in smaller genomes, virus or bacteria, you can sequence the whole genome even within 2 hours. So that, and then cell culture techniques and protocols have also developed quite a lot that we can do these different cell types in vitro culture, develop differentiation protocols are there. So all this is contributing into this field also in terms of generating sperm or oocytes in vitro, and then it can potentially be used in actual treatment or application. So I'm just showing you an example where you can see that from embryonic stem cells you can generate sperms, and again there are markers for sperms or oocytes and different developmental stages which you can confirm, which you can see from these pictures. So they develop nicely into sperms, and there are protocols published for developing from different stages. You can see here of different stages of development and the markers. So right from the embryonic stem cells, the PGCs, chromocytes, chronia, and spermatocytes, oocytes, etc. Further maturation, and different markers at each of these stages have been identified. So with the help of these markers, we can isolate these cells at each different developmental stages, and then that can be used either in the research purpose for understanding the basic mechanism, or during the clinical applications in treatment also it can be used when the protocol is established. So these are markers for mature ones. Now, the model which is there proposed is that you can take the samples from ovary or from testes, and then you can isolate these OSCs using the techniques that I just described, and then culture them. And now again, techniques have been developed now, CRISPR or other methods are there for gene correction. So with the sequencing, you can identify the problems. If there are any DNA damage, as in the previous talk, Dr. was talking about DNA damage, and those things can be identified, corrected, then they can be developed through the mutagenesis and mature. So it can be done from embryonic stem cells, or from the live samples, or from induced pluripotent stem cells. So in induced pluripotent stem cells, what happens is, first, any of the cells, whether it is from skin, kidney, liver, hematopoietic system, from almost all cell types, now protocols have been developed through which you can convert them into induced pluripotent stem cells. And then these cells, using the differentiation protocol, can be differentiated into gametes. And then they can be selected using either FACS or DNA damage, etc. can be verified with fluorescence microscopy or PNC. And again, it can be used for the treatment or for the research purpose to understand the mechanism. So this is it. I'm just keeping it very short. I hope that in next 5 to 10 years, it can be actually used effectively. And in experimental therapy, obviously, it can be done in vitro animal models, but in humans, because of ethical considerations or regulations, government regulations, it cannot be tried until and unless it is proven. That is it. Thank you.
Thank you, Dr. Shio. You said you had some video. If you want, you can try if it works.
No, I'm using mobile and the network is quite
Okay.
weak. The moment I even put it on I tried to put it on full screen and it didn't work. So
No problem. Thank you for your excellent talk. Anything that's come to your mind for the future to go from here?
I think we need to have these the protocols that I'm talking about for iPSCs or from embryonic stem cells. There are being done mostly in European countries or US. I just hope that in India, people do this high quality work, this kind of machines that is either here but it's not being used, or maybe funding problem is there, research funding. So it's not developing as fast as it should in India.
Do you think induced pluripotent stem cells can go the clinical way soon? Because in India, ICMR has banned its use as of now. We can only use it for experimental work, not for human clinical use. Do you think in future we will be able to use it? Do you
Yes,
see
I
potential
think
because you've worked on them in Germany?
I see the potential, and there should be some kind of protocol for clinical trial, whether it is in the case as a case like individual cases, which we call experimental therapy, where if somebody wants to take the treatment with consent, with informed consent and everything after proper explanation, then it should be allowed. Because until and unless you do some kind of experimental therapy on humans, it will never get developed. I mean, if you keep it banned that you can't do any testing in humans, then obviously in animals it is verified, but then you can't go to the next step. So there has to be some kind of allowance given that it can be done as some kind of experimental therapy on humans. So how it is done, since I'm not the clinical person, so how the committee decides, or which committee decides, or how the protocol is decided for clinical application, that that you can see what you guys can convince the government to allow some in some small ways instead of complete ban for human trial or experimental trial.
Thank you. Anybody else wants to ask anything or share a comment?
May I, Shilpa?
Yes,
May
sure.
I? Professor Shio, thank you for your wonderful lecture. That was very thought stimulating. Recently, from pediatric surgeons' point of view, the articles have appeared in which stem cells were used for regenerating esophagus. So, but this work was done in animals only, and its human application, as you have also said, is still to be explored, and what is the exact outcome, only time will tell.
Right. So I think that in developing countries, this is one issue that I'm, as a research scientist, I keep seeing that in China also, in India also, whenever there is some big development or advancement like in embryonic development, etc., then suddenly a lot of ethical question comes into consideration from journals also like Nature or Science, as well as from the scientists from Western countries. I mean, I didn't want to say it, but then it is banned in India or China, but then within 5 years or few years, the guys from Europe or US, or they just come up with the same or similar things, and it gets accepted, and then we also allow it. So I'm saying that why not we should take the lead, as a country, or as to get the science to the next level. So if some ideas are there, and if some proof is there, proof of principle is there in animal models, then the next logical step is to take it to the humans.
But Naga sir.
See, Dr. Shiv Mohan Singh, you've made a reference to experimental medical modalities, treatment modalities.
Yes, sir.
The biggest problem is that once these things, once the checks and balances are removed, there is rank commercialization and unethical practices start, and then it becomes very difficult to control it.
Right. You are absolutely right, sir. Yeah. And to control that, that's what I meant is that the government or the government hospitals or the government institutions, then they should take the lead. So the difference between US or Germany versus India in science is that in US or Germany or Europe, more than 50% of the research funding and most of the cutting edge development happens from the private sector. And even so, you can look at the data and just say explaining that. While in India, we don't have any private funding or initiative for this kind of cutting edge. It's only government, and then government also keeps it banned or very low funding. So either the government takes the initiative, or the private sector takes the initiative, or they work together. This is the only way forward in terms of if we want to develop the Indian science, biomedical science, science-based research output, whether it can be converted into clinical applications. So funding, yes, it has to be controlled that it doesn't go into unethical practices where private people start using it for just making money or profit without consideration. There should be rules and regulations, but within some framework. So, for example, government can take the lead. Like in Japan, Shinya Yamanaka developed the IPSC, and the Japanese government is actually doing the clinical trials for IPSC. So then Indian government should take at the national level, there should be like a national mission for trials using IPSCs for different diseases and disorders, not only gametogenesis.
Yeah, see, we had a recent example. We have a recent example. It's still happening. The use of convalescent plasma for COVID. Now, despite the fact that the ICMR and the government have made advisories to say that it does not work, it is still being practiced, and patients, who are desperate, are paying through their noses just to get that little bit of convalescent plasma for no benefit. So that is how things get commercialized, and then it becomes very difficult to stop it.
Yes, sir. It has to be balanced in some ways.
Sareen, sir, do you want to say anything as chairing?
Professor Sareen, are you there?
It's okay. Thank you. Thank you so much.
Thank you. Just for the benefit of the audience, Dr. Shilpa Singh is a hardcore researcher. He had a well-established job in Germany. He came back to his country to train more people into research, and he trains many trainees to how to do research and who stay with him for 1 year or 2 years and go through the lab work, which is still lacking in the government setup because of shortage of funds. So India is really grateful to him to train so many and then who have gone ahead to pursue their PhDs abroad. Thank you.
Thank you.
So
yes, please. Yeah. On behalf of Organizing Committee, I wish to present our token of appreciation. Thank you, sir.
Thank you, sir.
Taking time and speaking to us.
Thanks, Eva. Thank you for giving me a chance.
So it was a wonderful session. So this session is over. Now the next person, I think, next people to take over. Thank you very much, all the speakers and the chairpersons.
So,
thank you, Professor Jaina. I now invite Dr. Kirti Rathod to take over the next session. Are you there, Kirti? I saw you.
Yes. Good afternoon.
Good
Am I
afternoon.
audible?
Yes, audible.
yes.
Good afternoon, everyone. I'm going to coordinate the next two lectures. And for the first lecture, I'm going to invite the chairpersons, Professor V.R. Ravi Kumar. Sir doesn't need any introduction from us. He's quite well known and he's teachers of my teachers and even more. So he's a former professor and head of Coimbatore Medical College. And he's going to chair this session with Professor Pradeep Kumar Jaina, who's professor and head at SCB Medical College, Cuttack, Odisha. So I'll request the chairperson to start this next lecture. Thank you.
Thank you very much. And now I request Dr. V.R. Ravi Kumar to speak.
Yeah. Thank you very much, Dr. Rathod and Dr. Jaina. I have a very great pleasure in inviting Dr. Praveen Mathur, whom I know very well for 2 or 3 decades. He is the senior most professor and head of the Department of Pediatric Surgery and additional principal of SMS Medical College, Jaipur. Now, Dr. Praveen Mathur is a pioneer in laparoscopic work in Rajasthan. He was trained initially in Great Ormond Street, London, and subsequently was trained by Kusheri, and then he was trained in Baltimore. And as early as 2004, he was awarded the fellowship in the Association of Minimal Invasive portion of the ASI. Now, he is, apart from the laparoscopic work, he is also very much interested in the research work, and he has published a number of papers, especially on pouch colon. And then his work on the pouch colon, on the genetics, after he studied twins and also in the family, the same pouch colon occurring, he studied the genetic aspects of pouch colon. Along with Amelio Sexana, he has also classified a new classification to the pouch colon he has given. From the classification of 4 types, he has made it into 5 types. So basically, he is quite interested in the pouch colon, and we'll be very eager to listen to him about the genetic aspects of pouch colon. Unfortunately, down south in Tamil Nadu or the neighboring areas, pouch colon is extremely uncommon. In the last 5 decades, I would have seen 1 or 2 cases where the distal most portion of the atretic rectum is slightly dilated. The classical pouch colon is uncommon. And then I would also like to know whether there is some genetic variation between down south and north regarding the anorectal anomalies. Over to Praveen Mathur.
Thank you very much, sir. Thank you for the nice introduction.
Screen share.
Can I share the screen? Good afternoon, everyone. It makes me feel special to be a part of this academic meet. I owe an enormous debt of gratitude to Professor Mahajan and Professor Shilpa Sharma for giving me this honor. My today's topic is congenital pouch colon, role of screen. Yeah. Role of genetics or environmental factors. This is an era of pouches. You name anything and it is available in pouches. Milk, tea, juices, and even wine is available in pouches. Well, it seems the God also believes in this concept. Today, I will be discussing about those unfortunate newborns who are born with this anomaly prevalent in Northwest India and popularly known as congenital pouch colon. This study is dedicated to children whose endurance of suffering, whose courage in the face of congenital malformation, and whose smiles over tears inspire all who work with them to overcome childhood maladies. My first part of the talk would be covering the brief clinical aspect of the CPC, and the next part will be on the plausible role of genetics and environment and its implication on the disease process. This is a clinical photograph taken by me some 25 years back, which aptly describes this condition. CPC is a rare form of high anorectal malformation in which part or the entire colon is replaced by a pouch with a fistula to the genitourinary tract. The background is the incidence of ARM is 2 to 6% per 10,000 worldwide. Incidence of CPC is less than 2.3 worldwide, but the incidence of CPC is said to be about 5 to 18% of the total ARM. There is no family incidence has been reported. Only sporadic cases has been present, but suspected etiology is environmental exposure to unknown drugs or toxins. The unusual feature of CPC is the prevalence of this malformation in the Indian subcontinent with the highest incidence in the west and northwest part of the India. With the growing awareness of this condition, the CPC was given due consideration and was included in the Krakenbeck classification as rare or the regional variant. In 2005, two important events took place. NASA has identified 10th planet Xena, and CPC was introduced in ARM classification. Based on the anatomical landmarks of the length of the normal colon, CPC has been divided into 5 types. This classification is more scientific. It includes rare forms of the double pouch, and it offers clear guidelines towards management. This is the sketch of the CPC classification. This was also endorsed by other authors in the form of the issue and published in Journal of Pediatric Surgery. These are the operative types of the pouch colon, clearly depicting type 1, 2, 3, 4, and 5. The 5 is we have reported for the first time, and now there are 8 cases of the CPC have been described till date. It has been said that anything that happens once may not happen again, but everything that happens twice is likely to happen for the 3rd time. CPC is diagnosed by invertogram or the plain abdominal X-rays. The conventional teaching is single large featureless gas shadow occupying more than 3/4 of the transverse diameter of the abdomen is suggestive of CPC. We have observed that plain abdominal radiograph has a predictive value in determining the type of the CPC. In CPC 1 and 2, the apex of the pouch is directed toward the left hypochondrium, whereas in CPC 3 and 4, the apex of the pouch is directed toward the right hypochondrium. These findings can best be explained on the basis of the fixation of the ball on the posterior abdominal wall, as the points of the attachment are fixed in CPC and the pouch can enlarge only in the opposite direction. This is the radiological feature of type 5 pouch. This is an overview of CPC management algorithm. Stage procedure is still offered the safest option when compared to one-stage procedure, which is associated with high incidence of morbidity and even mortality. Please note the different color configuration, which denotes different staging procedures. Gray color denotes stage 1, dark gray stage 2, and black stage 3. Such guidelines cannot substitute the judgment of an experienced surgeon and are designed to allow the flexibility in practice. Now, the etiology of CPC is quite elusive. We have worked on a hypothesis that environmental factors could lead to triggering of the events which influence the window time of the cloaca septation, number 1, and number 2 is genetic inheritance or de novo. So we have started with a plan. The beginning can be made by studying the differences in the structural pattern of the normal tissue and the pouch colon tissue. Therefore, histopathological and immunohistochemical studies, proteomics in form of 2D gel electrophoresis and mass spectroscopy, and finally the DNA pattern by way of microarray and whole exome sequencing. The objective is simply to understand the genetic basis of the congenital pouch colon anomaly and to identify the genomic regions and genes present in the development of the CPC. We have performed histopathology with 2 stains, hematoxylin and eosine, and another is Mahajan trichome. We have considered IHC markers, smooth actin, desmin, myosin, neuronal, particularly neuron-specific NLOs, S100, GFAP, and cathepsin D. The majority of the findings are acute and chronic inflammatory cells, and the consistent finding is disorganized muscle bundles. Ganglion cells were normal. This is the normal appearing colon tissue with mucosa, submucosa, muscularis propria, and in CPC there is disruption of the mucosa, ulcerations, muscle fibers were disorganized. And on IHC scanning, this is hypertrophic muscle fibers disorganized in the muscle propria. And this is the, on mesenteric staining, we could find the division of the various bands, fibrous band, etc. IHC and IHC for neuron, especially here in this particular slide, this is the normal colon with the distribution of the actin tissue in muscle propria. Actin activity is normal, whereas in spouse colon it is patchy. And the most important finding which we have come across in CPC 1 and 2 is an additional muscle coat here in muscle propria, just below the submucosa. This could be the reason for the poor post-operative results in type 1 and 2. Immunohistochemical for neuron-specific analysis showing diffuse cytoplasmic positivity in ganglion cells in CPC specimen. An additional muscle coat inside 1 and 2. This is the finding we have come across in 1 and 2 time. Mature ganglion cells present. This suggests that CPC has distinct defects in the neuromuscular and reduced contractile protein in pouch dysfunction. Now, with 2D gel electrophoretic studies, this is the proteomic, and in this the normal distribution of actin myosin, whereas in pouch colon, this arrows indicate differentially expressed proteins. So there is reduction of the contractile protein in form of keratin, actin, and zinc finger protein, which in turn reduced contractility of the pouch and pouch dysfunction. CPC is a sporadic disease. No familial incidence has been reported. This is we encountered a family incidence of CPC in a distant family members and 2 incidents of dizygotic twins in which one is affected and the CPC while the other is not affected with it. The probants, we described the one large family with pedigrees over 5th and 6th generation with 2 members affected with CPC. The probants in the 5th and 6th generation respectively have a common ancestor. In the present study, the pattern of the pedigree shows that CPC may be running in the family as an autosomal recessive trait, as it tends to skip the generations, and affected offspring are usually born to unaffected parents. Now, dizygotic twin more in the form of in favor of the genetic theory, and on microanalysis of both the siblings suggested that these CNV contained genes like colorectal cancer associated ML genes which regulate the function of the HOX gene expression. HOX gene is responsible for formation of the body structure, organ and gut development during early embryonic phase. We could reach concordance with our whole exome sequencing study, which I will discuss just now. Whether or not rare diseases are genetic remains incomprehensive till date. Such rare disease native to Northwest India is CPC. In order to unravel the mystery, we have planned whole exome sequencing. Quad analysis were planned, comprising of parents, patient, and unaffected sibling if present were taken. 64 samples were taken, blood samples were taken. Genetic variants associated to CPC were found in 18 cases. Whole exome sequences performed with Sanger validation, and sequences thus obtained were aligned to human genome references. We have observed a number of rare mutations like rare missense variants, frame shift mutation, stop gain variants, stop lost variants. Frame shift variants, when mapped to the gene ontology pathway, this is a bioinformatic tool, shows a wide distribution of various cellular and metabolic processes. 2 of the 63 frame shift variants affected with spliced region containing EB, B and CT1 genes. They are found to be associated with colon, neural, brain muscles, maladies. And there is a high chance that these genetic mutations might be associated with the malformation of the colon tissue. When these genes were subjected to Gene Mania, another bioinformatic tool, a large number of them were shown to be co-expressed, while a small set make up of genetic mutations. Majority of them are associated with lectin-mediated interactions. This was published in Nature Scientific Reports. In the simplest sense, expressing a gene means manufacturing its corresponding protein. And this is a multi-layered process which has got two major steps. One is transcription and another is translation. Transcription is information of DNA is transferred to the messenger RNA in the presence of an enzyme RNA polymerase. And now the messenger RNA, which is now a single-stranded copy of the gene, which must be translated to a protein molecule. In the whole exon sequences, we sequence only exons which have a coding potential. But in this study, we got some intronic part also sequenced. Intronic part contains non-coding RNA, which are also known as regulatory RNA. They do not code protein, but helps in the regulation. So a long non-coding RNA serendipitously found from whole exon sequencing. Further, interaction prediction between long non-coding RNA with 6 of the coding proteins. We establish an interaction of long non-coding RNA with KIF13A, which is a bona fide gene found to be causal gene for CPC. Long non-coding RNA potentially shown to be intensify with red edge. This red edge means it is closely associated with this gene. Now the function of KIF13 is transportation of monomase 6-phosphate receptors from lysosome to Golgi apparatus. Number one. Number two, the translation machinery resides within a specialized organelle called ribosome. Messenger RNA molecule must leave the nucleus and travel to the cytoplasm where ribosomes are located. In addition, we checked the gene ontology pathway. It was observed that KIF13 is also involved in manifestation of the colon-related disorders, particularly RNA. This will be more clear in a 1-minute animation film. Which is a quad analysis involving Father knew. This is the sequence. Transcription and translation is going on, whole exon sequencing. I try to make it more easy of this complex process by way of an animation film. This is the exon and intron. Normally we extract only exon, but simultaneously we have found some mutation in the intron also, which is lying just adjacent to exon. And we have found some of the variants in intron also. Later on in future, it could be the biomarker for CPC. These are the causal genes and have some interaction expressive of CPC. Whether this intronic part is also stable is not is again is ascertained by another bioinformatic tool, which is known as RNA folding receptors. There is a hard science behind it. Now, very important part, we plan to conduct an experimental study on 8 Swiss albino mice. Animals were divided into 2 groups, control and test, comprising of 3 females and 1 male. Test-specific food was given to the test group. They gave birth to the litters in both the groups. Control group's litter were absolutely normal and healthy. And after 3 days, they were dissected. And the large intestine were found to be normal. But in test group, where we have given test extract containing some dietary intake given to the already pregnant females, and on 11th day, 5 litters were born. Within 48 to 72 hours, all litters were died. The litters were dissected then, and you will come to know this surprise finding of that in dissected film in normal patient, the GI organs were absolutely normal, whereas in CPC, the test group, the gastrointestinal organs were partially developed, yellowish semi-solid content, and the pouches inside the large intestine were found. The histopathological done in both the cases and the expression of actin were at par with our observation, which I have described just now. And this is the actin which I have described earlier also. So this is inconsistent and at par with the observation. I mean, this is a wet study, and we have done dry study, dry lab study by bioinformatics, and wet study on mice. So the conclusion is, from the large cohort, we have found the 2 cases of the zygotic twins in which one was affected and another was unaffected. From our recent whole exome sequencing, we have found certain mutation harbored in the CPC cases. Whether or not it is genetic or there is an influence of epigenetic remains to be ascertained. Role of the non-coding RNA and their role in regulation of CPC has been established. Identify mutation in both coding and non-coding regions affecting such phenotypes are not only of valuable interest towards clinical applications, but also important for enormous prognostic value for therapy. This can be used as a biomarker in future. Thus, we pave way for our studies from clinic to genetics. In summary, what I am suggesting is environmental factors in terms of the food habits and later altered expression of genes followed by adverse protein expression that went unchecked by self-regulatory mechanism that is within our body might be responsible for this phenotype. These are some of the landmark articles published in various journals. Thank you very much for your patient hearing.
Stop screen sharing.
Thank you, Dr. Pravin Mathur for the excellent exposition of a very difficult topic, the genetics. Genetics by itself, we have to understand the basics first, and subsequently we have to work with the genetics to find out what exactly wrong, and then to understand the whole subject bias, what you have done. It's really tough for an individual like me. I mean, for many of us, I hope so. I'm sure you need to evolve a system to teach people how to integrate our studies with the genetic variations that we see in so many congenital anomalies. If there are any questions to him by the audience, you can ask them. Dr. Shilpa?
Yes, sir. So that was an excellent talk. Pravin Mathur sir has been my teacher, and that's why I'm a pediatric surgeon here today. Thank you so much, sir. And he had invited Ravi Kumar sir when we were in Udaipur. So we had learned from Ravi Kumar sir also.
Yeah.
So I never forget these two in my life. So I wanted to ask, do you think we can prevent CPC in future in India?
There are three things. I just summarized that there are three things. One is food habits. Another is altered gene expression. And thirdly is unchecked self-regulatory mechanism within the body. If we can check food habits, in particular, because it is a region specific. In North India, it is highly prevalent, but there are only few cases reported from South India. So there is it is a region specific disease. So one should think of these food habits also into consideration, and maternal factors like when pregnant women are exposed to infection, cytomegalovirus infection, which can lead to these anomalies. We know everything, many congenital anomalies are the result of CMV infection. So this remains to be seen that we can stop it altogether or not. But at least we can check, we can improve our, I have not disclosed the text food extract which we have given, because it is unpublished and it long way to go. So if we can pinpoint those things, we can stop it, and then we can improve the nourishment also of the female. Then of course we can improve the at least prognosis of it.
And why haven't we been able to diagnose CPC, at least the type 1 and 2, during the antenatal period till now? At least we have not seen any case.
No, I have no knowledge, but it's very difficult to diagnose antenatally.
When the colon is so distended, there should be a suspicion.
Yes, I have not thought of it, but it could be. Yeah.
Thank you, sir. Thank you.
This was a very wonderful talk. I work in Cuttack, Odisha, and for the last 8-9 years, we documented all the CPC in our state. It quite common, and around 8% we have documented. We have already published paper. This is around 8%. But with Dr. Pravin Mathur advising so many things, I think next phase will be in that line. Thank you very much, Dr. Mathur.
Thank you very much, sir. Thank you.
So this is the conclusion of this our part. So next,
Dr. Jena, Professor Sareen wants to ask a question.
Sir, welcome, sir, Dr. Sareen.
Unmute.
Hi, Praveen.
Yes, sir. Hello.
Hi, how are you? Actually, my question has been already asked by Shilpa. I think we think alike. I also wanted to know whether, with your vast experience CPC, you ever known about antenatal diagnosis of CPC, and if yes, at what gestation? But you already said that you probably, I think I'll Google it and get back to you.
Right.
Dr. Jena.
Sir, that was a very wonderful things. Actually, we can detect this one, antenatal period, but our look or our things are coming to other way. We are not concentrating on the CPC. We are concentrating on other. That's why probably we are losing it. So this was a very wonderful and thought-provoking discussion. So genetics in CPC is a very, very interesting thing. I read the article, Dr. Pravin Mathur, what he has said. and I think we'll work on that. So now I hand over to the next people who are on the Dr. Kirti Rathod to organize, coordinate.
So before we go there, Dr. Jena, I would like to thank Professor Parveen Mathur for his excellent talk, and as a token of appreciation from organizing committee. Sir, please accept it. And thank you very much for taking time and speaking to us.
Thank you very much. Thank you.
Thank you for the wonderful lecture, sir, and thank the chairperson to coordinate this session. For the next lecture, I am going to invite Professor Sushil Buddhiraja, who's the head of the Department of Pediatric Surgery at DMC Hospital, Ludhiana, and Dr. Alpana Prasad, who's Senior Consultant Pediatric Surgeon at Sri Gangaram Hospital, Delhi. Thanks.
Good afternoon, everyone. Is Dr. Buddhiraja here?
Yes, ma'am.
Okay.
Dr. Buddhiraja
is here.
Dr. Sushil, can you hear us?
Yes, I can hear you. Yeah.
Sure. Okay. So Dr. Buddhiraja can go ahead and introduce the speaker, Dr. Dhruv Ghosh.
Dr. Dhruv, you can start sharing your screen so that we can see your CV slide.
As we know, the clinical research in medicine has been a domain of the Western world, but in the last 2 decades, the borders have been breached, and now research has come to all the other countries. But what are the key strategies that is going to this globalization of clinical research more effective? And what are the other technical factors, legal, financial, and ethical issues that can creep up? I think Dr. Ghosh is going to enlighten us about this. Dr. Ghosh is Hub Director of Indian Subdivision of NIHR and Global Health Research Unit, University of Birmingham, UK. He is Professor of Pediatric Surgery at CMC Ludhiana and presently working as a pediatric surgeon and pediatric urologist at in a university hospital in Muscat. Over to Dr. Ghosh, please.
Thank you, Dr. Buddhiraja and Dr. Prasad. I hope you can hear me. Thank you, Professor Mahajan and Professor Sharma for inviting me. And it's lovely to see one of my teachers, Professor Sareen, as the president of the Indian Association. The topic that Dr. Shilpa told me was to talk about getting global with surgical research. I've just added a line to that, Shilpa. It's delivering an effective research collaboration. It's more of a general topic and obviously not so high tech as the previous speakers. So it might be a bit of a dampener, or more of a community health talk. And so you feel free to have a snooze. I have no disclosures. I'm sure you're familiar with the Lancet Commission, which came out in 2015 and then was repeated in 2017. Around the world, 5 billion people lack access to safe and affordable surgical care. My talk is not specifically with pediatric surgery, but pediatric surgery obviously forms a very important part of affordable and safe surgical care in many parts of the world. Before the COVID crisis started, about 143 million additional surgical procedures were needed each year to save lives and prevent disability. And I shudder to think what has happened in the last 2 years. 33 million people used to face catastrophic health expenditure due to payment for surgery each year. And again, before COVID-19 hit us. Last year, the ASOS-1 and the ASOS-2 study came out, the African Surgical Outcome Study. Which though a fairly negative study, especially ASOS-2, one of the major conclusions was that patients in Africa were twice more likely to die post-operatively, even though they had less risk factors compared to many high-income countries. So there is low research capacity in countries with the greatest need for research to improve surgical outcomes. This is a very interesting slide, and I did not know this till I started working with global surgery, that there are more post-operative deaths around the world than malaria, TB, and HIV combined. And a lot of these deaths happen in countries with low surgical capacity with higher populations, etc. So in terms of absolutes, post-operative deaths constitute the third highest mortality rates after ischemic heart disease and stroke. Statistics that many policymakers and planners around the world do not talk about. So we have, for want of a better word, they're very fashionable research in ischemic heart disease and even in stroke. But globally, the research in surgery has been minuscule. Surgery has been the neglected stepchild of global health. Our friends in anesthesia are the invisible friends of the neglected stepchild. So it's some of these quotes I really love. We as surgeons are presently the Cinderella of global health, of the global health agenda. A lot of people pay lip service to us, but when it comes, when the time comes to dance, the room is almost empty. What about pediatric surgery? Are we the unborn child of global health? In many countries around the world, yes. So the requirement, like I said, 5 billion people have inadequate surgical care. There has been a massive growth in surgical infrastructure in many countries, and many countries even in the global south. For example, in our country, there has been a massive growth in surgical infrastructure. And there has been a growth in surgical practitioners, maybe not so much in pediatric surgery, but I'm sure in other specialties of surgery. However, this has not kept pace with the increase in the need and want and maybe the population in many countries. What is the risk? The risk of an overstretching resource, lower standards of care, and increases patient harm. And finally, if we do research in global surgery, in surgical care, in surgical outcomes, what does it do to how does it benefit us? It increases quality assurance of surgical care. It engages the surgical community to improve patient outcomes by collaborative research rather than research being done in their own small areas. And it does create future surgical leaders across the world. So research in surgery is critical, according to what many of us feel. Then what is global surgery? So global surgery is basically a multidisciplinary field aiming to provide improved and equitable surgical care across international health systems. Around the world, there are different types of health systems. In our country, the health systems are very varied compared to, say, some place in Africa or in South America or in Europe or the United Kingdom. However, there are some pillars which are very common around the world, even in high-income countries. That is the need, the access to surgery, and the quality of surgery. And global surgery engages a range of individuals, which will include academic surgeons or academicians. It includes clinicians. It includes lawmakers. It will include economists. And there's something that is for all of us to think about, because if it is not economically feasible for our patient, that doesn't make sense. And it should include, finally, policymakers. Those are doors that we have to knock on very hard. Most importantly, and that's why painted in red there, are our patients. When we do research in global surgery, in improving outcomes, we need to talk to our patient before we start our research, because if it does not affect the patient, then is there a point in doing that research? So when we start with global surgery, I work with the NIHR unit of global surgery, which was initially based out of the University of Birmingham, but now has been sort of spread towards the global south. Our challenge was to conduct local research with a global objective. Very commonly, when research comes out of high-income countries, the questions are asked by high-income countries, which would be completely irrelevant to low-income countries. So the challenge is to ask the research question from the global south, from countries in Africa, in South America, in Asia, in South Asia, rather than from the UK or Europe. And also to take help of the research expertise of many high-income countries, and especially their facilities, to conduct the research which will affect and improve care in countries like ours. What does the Global South bring to the table? It brings a large data set, it brings a diversity of data set, and it brings a need for fast-paced improvement. I think in surgery, we need to go to a point across many countries where outcomes and surgical procedures and surgical care is not based on whims and fancies, but on facts. What should we strive for? In countries like ours, we should be striving for young surgical scientists and researchers. We should look at the diversity experience and grab it. And we should not forget the surgeons in remote areas. Even now, around the world, in many countries, a large section of our population actually lives in remote areas. And that is a data set we sometimes overlook because they are important. So let's not forget that data set. Let's not forget the surgeons who are working in these remote areas, sometimes single surgeons doing everyday calls. Not an easy thing. How can we help their experience to come out to the world and teach us? So this is how, this is sort of a start of why collaborative research is important. And that's what we did sometime in 2016-17 after a meeting in South Africa, when we decided about starting a research collaborative to improve care and started with low-cost interventions. When you start a collaborative, it's good to listen to everybody, listen carefully. We should plan carefully, and it's always good to listen to people who have done research. Now, you won't need to probably do it the same way, but it's good to have that knowledge so you don't make the same mistake. So stand on the shoulders of giants, what Google used to say some time back. Not everything colonial is bad, and I think we have learned a lot from many of our researchers in the global north, in high-income countries, and adapted that to the way we work in many of our hub and spoke countries. When you build a collaborative, you need to spread the word. Not everybody is going to be interested in a surgical research collaborative or to improve outcomes in the community vis-a-vis surgery. However, the more you spread the word, the more interested people you will get. This is when we started sometime in April 2017. And you can see in the table around there are a few people interested in listening carefully, but then there are also a few people who are tapping on their phones and might not be really interested. No harm done. The people who will be interested are the people you will take forward, but it is always good to spread the word. When you build a collaborative, you choose a team which will work for each other. We realize one thing, that everyone in a team is equal. So if it's a research associate or a research nurse or a research leader or a surgeon or anybody or a data manager or a hub manager, everybody is at the same footing. They have their own areas of work, and so everyone is equal. The colonial way of managing things, that you have somebody sitting on top giving orders, the others follow orders, really doesn't work in the long term. So everyone is important. Some people will not finally fit into your team. It happens. That's life. So you offload those people who are not fitting in, because the team is more important than individuals. Allow for independent decision making. It's extremely important at all levels to allow for independent decision making, because that will make people think. And always start small. And as you grow up, you will realize that if you have a good team, you grow very fast. When we built a collaborative, you have to do administrative planning, delegate work, chain of command, all those things there, but allow for mistakes. Mistakes will happen. That doesn't mean that you're doing something wrong. It's just a mistake. You learn from those mistakes and do not repeat them, so that you go forward in doing right things. Communication between team members, between teams, is extremely important. We meet regularly in our collaborative. COVID actually has made meeting easy over Zoom. And choose partners who are motivated, who are ethical, who will provide you with robust data, who will stay in touch, and you have to give ownership to your partners. This is us building a collaborative in tertiary hospitals, in small community hospitals, in rural areas. The top left picture is of a hospital in rural Maharashtra. At the bottom there is a picture of a hospital in Manali, and then we have some of our friends from Vellore. Your team has to be extremely motivated, because when we start working in global surgery, it's not that we are working in tier 1 and tier 2 cities. You have to go into the remote areas to capture the right picture, and your team will have to go through tough areas like this. Dr. Sareen will probably remember this area in Manali, going to Manali with a landslide, where our team was stuck for 3 days in a car before they could actually come down. So a lot of work, a lot of motivation is required in your team to work through these. When you build a collaborative, we can crib and complain about this, but you have to understand rules and follow them. National rules, data protection rules. Data is very important. It has to be protected because your patient's anonymity has to be protected. The patient has to be at the center of everything that you do. There has to be due diligence of all the teams that you work with, and your financial administration has to be completely above board. This is an Achilles heel, and if you mess up with your financial administration, everything will go south. And of course, follow your local rules and ethics committees and research committees. India has extremely robust rules when it protects how you do studies and trials, and I think this is something for many countries in the global south to learn from. Unfortunately, many of the other countries that we work with do not have such robust and good rules. And then you broaden your team. So when we started with surgery and pediatric surgery, but we started including all surgical specialties over the years. OB/GYN, extremely important. Cesarean section is the most commonly done surgery around the world now. Oncology, microbiology, and especially anesthesia. Never forget anesthesia. It is very important to have your anesthetist friends on board. Involve medical students, get them into this as soon as you can, because they come up with brilliant ideas, which many old people like me would never have thought of. There was a trick that we missed when we started, and we did not involve our nurses. And we realized that halfway down the time, sometime in 2019, that's a huge trick that we missed, and we had to really climb back to get the nursing teams on board. The nursing teams, involving the nursing teams doesn't mean involve them to collect data. It means make them research leaders, because at the end of the day, if surgical outcomes have to improve, nursing outcomes also have to improve. And they are a very, very, very important part of all that happens in surgery. Then you broaden the collaborative. You involve anybody who's interested in ethical, robust research. And I would not, I could not stress more on the word ethical. It is very important that research is done ethically. There should be no fudging of data. There should not be wrong data. The one thing that we want to, if we are recruiting in a trial, even if we recruit one patient, it's fine, as long as the data is absolutely clean. It is very important. Involve everybody who has a research or an ethics committee and can go through the processes. There should be nobody who should be left behind, because you have to have a full picture. When you build a collaborative, mistakes will happen, ideas and egos will clash, people will leave. It happens. The leader of the collaborative becomes very important here, because he is not the most important person. Again, it's the team. So accept, step back at times, recalibrate. Sometimes you have to move on. And that's how teamwork is. And I think a very important part of our teams are the research associates who basically make us surgeons do what we are supposed to do. And then you have the fruits of your thing. Falcon was one of the first surgical RCTs that we designed with our UK collaborators and the hubs in Africa and India. Our sample size was 5,400 odd patients, and we were able to randomize, because it was collaborative research, in just over a year. Falcon was a 2x2 trial, which was looking at surgical site infections and comparing medicated versus non-medicated sutures, as well as povidone iodine versus chlorhexidine paint. Pre-pandemic, we were recruiting more than 1,000 patients, and because we were in many of our countries and the kind of work that we do, we were able to continue recruitment through the pandemic, something that I'm sure no Western country would have been able to do. So 54 sites in 7 countries across 3 continents, and we closed recruitment 9 months ahead of schedule. And recently, our first article, our first results from this came out in the Lancet. We work on what we call collaborative, corporate authorship. So there is the initial authorship is of the entire collaborative, and everybody is listed as a PubMed citable author according to alphabetical order. And so it doesn't mean that somebody from the West takes precedence. It doesn't happen that way. And then we moved on to what we are doing right now. Is a different type of a trial, which is a cluster randomized trial where hospitals become clusters. Something that many of our senior surgeons would probably have been doing is changing gloves and instruments before closing fascia. Our sample size was 12,000 odd. We are projecting to end this trial sometime end of this year, beginning of next year. So your research question has to be based on felt needs. The question might be asked by HICs, but it's not important. We work with it and plan to flip it according to our needs. For example, to determine variation quality of cancer surgery worldwide. It might be very different for what happens in Europe. New questions come from the original questions. When you start working, we were working with colorectal cancer with adult surgeons, and then we realized what's the point of doing chemotherapy or doing genetics, because many of our patients were just going off the treatment pathway because of catastrophic expenditure. So we started looking at referral pathways and catastrophic expenditure. This is a paper which should come out very soon, done with our collaborators in Tata in Mumbai, as well as some of the other AIMS, AIMS Jodhpur and AIMS Rishikesh. Then you get sub-studies from the original question. When we were doing Falcon, there were a lot of people who did not come back for follow-up. So why weren't they able to come back for follow-up? Because they are in far and remote areas. Then we started looking at the feasibility of a telephone administered patient-reported wound health questionnaire to see whether that can be validated with what we see in person. The global question and the local question. So a lot of our high-income country collaborators would talk about nutrition in cancer surgery. We started looking at identifying low-cost sustainable nutritional intervention prior to cancer surgery. Then we started looking that these patients are actually not able to come to the hospital. So what's the problem in access to surgery? And so these questions, though global, are extremely important locally. And then we have our medical students, and this is something Dr. Sareen will probably remember. That's something he did in the 1990s in abdominal tuberculosis, and one of our medical students has recently sort of revisited this condition and looked at surgical outcomes. But more than that, she went on to look at what were the cost factors related to people coming for treatment, especially surgical treatment for abdominal tuberculosis. And Dr. Shilpa will remember this. This is something that she was intimately involved in, the COVID Surge Week study, which was a snapshot study done over a week in last year, which included 142,815 patients within a week, within a month in October. And one of the side effects was that we got a Guinness World Record out of this. This looked at how patients are doing after surgery and prior to surgery, whether they're comparing patients who had COVID or were asymptomatic, etc. And we came up with some very important conclusions. And now I've just got off the email yesterday that we are going to look at the Indian cohort of this. This is something that Shilpa and me would be very happy to work with to see how the Indian cohort behaved during this time. And we are taking it forward. And a lot of this, the COVID Surge Week study, was done by medical students, surgical registrars across the world. And if you look at the map, almost every country in the world was covered. This was an amazing study, and it was just because the team was so good. The most important question is you have to take your research questions from the community to give it back to the community. You have to talk to them. This picture, the permissions have been taken by for one of our patients, and you have to give back to the community. Before that, you have to listen to the community. What they want is very important. And so what we started doing with our research collaborative is what we call community engagement involvement. Before we start a trial, before we even think about a trial, we talk to them and see what are their needs, what are their questions, and then see whether we can develop a research question out of that. Like I said, the authorship is very corporate. Then we come from conclusions to guidelines. I talked about the Falcon trial. There's no evidence supporting 2% alcoholic chlorhexidine or coated sutures in improving surgical site infections. So whether we can move on to guidelines like we did for preventing surgical site infections. Every research collaborative globally has to have sustainability. When you start off, there is an enthusiasm. But after some time, it's the end of a good thing. We start off to a better one. When we started off, we were like, for want of a better word again, a startup. How do we sustain ourselves? We sustain ourselves by training the next generation of surgeon scientists and research managers, provide study, bespoke and tailored training for unit studies. And it could be a mix of in-person, open access, online training modules. So we are developing training modules in research and surgical research, which will be available free of cost to everybody around the world. It's not going to be one expensive course from say Harvard or something. It will include all these modules and more. We're trying to involve artificial intelligence and adaptive learning to see how we can improve post-operative care in the ward, in our surgical wards. So we're looking at emergency laparotomies, which are a high-risk group in the Global South and LMICs, and deliver an online training package which will improve care and catch deterioration much earlier. And then we have to do an economic evaluation alongside all our trials. In fact, we started doing it before we start a trial. It's a comparative analysis of alternative courses of action in terms of both costs and consequences. It quantifies the cost benefits of interventions and whether a surgical trial intervention may demonstrate clinical effectiveness, but if it does not affect cost effectiveness, then is it worth it, really? So, we looked at key resource use, and 70% of global population is at risk of catastrophic expenditure if surgery was needed. So we have to look at the cost of surgical complications to patients in terms of out of work, additional treatment costs, health systems, and particularly if no universal healthcare is available. And this data from LMICs is very, very little. So we look at health economics. Now, health economics does not stop at research. Our objective today is to bring health economics to the bedside. Me as a surgeon standing next to a patient have to be thinking in my mind, is this intervention, is this medicine going to get the entire family on the road and make them starve? Do I need something cheaper which will be as effective? And then we have developed a management training center and a data training center in Ludhiana, where we give training for research and researchers. And if you look at these various modules, it's in terms of not just research, but marketing training, how to engage stakeholders. That means policymakers, politicians, as well as our patients. And we support faculty for research and module development. We are hoping to start a fellowship in global surgery research in India very soon. We have already started a fellowship in global rural surgery research in India. And this is open to anybody. There are no costs involved. Like I talked about, and one of my other teachers, Philip, who works in Manali, rural surgery is a changeling. We don't talk about it too much in many of our conferences. It's a concept, but no definition. What are the objectives? How we can support people working in rural areas and doing surgery, because they are very important. And if we improve their capacity, it just helps the country. The enthusiasm for research is there. When we started, in 2017, there were 5 centers. There were 5 pins out there. And you can see with COVID Third Week and beyond, the number of centers. And this has grown. This is a bit of an old slide, and this has grown since I last updated it. Looking ahead, we now are thinking in terms of sub-hubs, where right now the hub is in Ludhiana, but give responsibility across the country, in the south, in the east, in the west, for people to manage on their own new research ideas, better coverage, and get data from everywhere. Like I talked about rural surgery, homegrown projects into health economics, training and education, machine learning. And then finally, being a pediatric surgeon, have been able to push through things, and we're starting with pediatric surgical research in our collaborative in pathways of care. If you look at the Kerala model for pathways in care in pediatric cardiac surgery, it's something to be learned from. Why shouldn't we do a surgical outcome studies in India or in South Asia, or a pediatric surgical outcome study? And this is something that we are developing right now, and would invite everyone to be part of this. Finally, this is what we want. And I hope at some point in our lives, we are able to get this across the world. Health is a right. It is not, it is something that we all should deserve. Thank you.
Thank you, Dr. Dhruv, for an excellent presentation. This concept of globalization actually has brought a big stimulus to the local people in the developing world, where there are people are collecting their own data and doing their own innovations. But when it comes to the research from industries, we are, as a globalization, if we participate in protocols and comparative studies, fine, that's good, we get the benefit out of it. But when it comes to new technology or new medicines, probably that same thing is not transferred to the people in the low income groups or middle income groups. that is a big challenge, and it's a big ethical issues when we come to the research. Actually, that has to be taken care. How we can overcome this thing. That's the main my main concern.
Yeah, I think Dr. Budhiraja, you're absolutely right. I think for many of us who have had a little experience working abroad, you sit and wonder. The need is more in countries elsewhere, but the availability of a lot of things is not there, which is sad. And hopefully, at some point of time, we will be able to take the initiative back to us and develop things that will improve care. I do understand it's a tough job. And it's not just one collaborative or one organization which can do it. All of us have to come together to improve care for our patients.
Thank you, Dr. Ghosh. Anybody else want to send comments or questions?
I think Dr. Sareen wants to come. Yeah. Dr. Sareen.
Kudos to Dhruv and Philip. I've taught them all, and kudos to CMC Ludhiana. I understand we've been doing thesis and all that kind of research locally for years, but for the first time, I tried doing collaborative research, having about 9 centers to do, nephron sparing surgery in unilateral Wilms tumor. And I realized when you do collaboration, how difficult it's going to be. And I urge Dhruv to actually hold a workshop online or otherwise to teach us all how to kind of go about it. And one more question to Dhruv is that I was trying to look into your slides, and I think you did mention somewhere, but I would like you to elaborate on how the data has to be robust, and there has to be independent monitoring boards at each hub. So as the data that filters and gets accumulated to be published eventually is very, very authentic. Thank you, Dhruv. Could you say a few words about that?
Yeah, thank you, Dr. Sareen. I thought this was going to be a very tough question from my teacher. And thank you for that. And I've just, when I was looking through the abdominal tuberculosis, and I was looking through many of your papers, and I do understand the kind of work that you tried to do even in the 90s. Maybe a lot of people missed a trick at that time, and if that had been done, we would have been a different stage, and I probably wouldn't be giving this lecture anyways. But yes, collaborative research can be very difficult. One thing I realized with collaborative research is that if I'm leading a collaborative, then I have to learn to listen to everybody rather than myself, and that is most important. If I'm leading the collaborative, I have to stand at the back of the line, not in the front of the line. It's my team which has to stand in the. Subconsciously, sometimes I have thought that I was right and everybody else was wrong, and to my detriment, I have found out no, that's not the case. I was completely wrong and everybody else was right. We all learn from our mistakes, and that's how collaborative research sort of works. It's just that we have to keep at it. This for us, it has been work for the last 5 years, and we were the kind of work that we did sincerely sort of put us into the NIHR's refunding scheme, and we were one of the 2 collaborators which were actually refunded last year. To your second question, we develop a team of research associates, research nurses, managers who go through the entire process of learning. Whatever the surgical researcher is learning, that's what the research associate and the research managers are also learning. So they know every bit of their of the trial. So when we involve teams, almost every month, we will go and check the data, check the protocols, if they are being followed in the right manner, if things are being done. And so those checks and balances is what we keep. We also do a 360-degree evaluation of not just the hub, of all our teams. So there is a 360-degree evaluation that has to be done by for me as well, whether I am up to the mark or not, because if I'm not up to the mark, then there's no point in me hanging around there. We do get the day-to-day data that is coming in from each site, and at the end of the day at 4 o'clock, the team sits together and goes through that data and sees if there are any red flags that are coming up, which shows that something is going wrong there. If it is, we sit that night and finish that off and see whether if there is a red flag, we stop, We go to the site, we check the site, see if there is a problem or not. It is a bit of hard work, but then at the end of the day, what we get is absolutely clean data. And the only thing that for me is that as a researcher, it's my reputation, and that should not go for a fix by somebody else falsifying data. So it is very important. So we are very hands-on on making sure that everything is correct.
Yeah, I think, it's not very easy for most of us to be doing research in the lab and do that whole body genome and all that. But there's a lot of clinical research that's possible, and there's so much of data, so much of patients that we have. I think we could be the world leaders if we learn how to collaborate and do more and more collaborations. I think whatever years of my work left now, 6 years or so before I turned 65 and thrown out of government job, this is something that I am now looking forward to. I mean, other than my teaching and whatnot, I think most important is that I encourage and involve more and more people to pool their data and do the things, the research, the way it is to be done, which is globally accepted, so that our credibility is increasing and we lead the world and tell them what to do exactly. Yeah, money and all other issues have to be learned and taught. And I think I would not mind, taking a small sabbatical and coming to CMC Ludhiana all over again to learn from you.
No, I think, thank you very much for that, Professor Sareen. I think it is very important. The first step would be the interest. So to be fair, and this might be controversial, but sometime back in one of the pediatric surgery groups, I did mention about global surgery, but I did not get any response from the pediatric. That's the reason probably a year ago, I spoke to you about this before you were going to take the interest is important. I do understand it's very important to do all the other research that has been done. Everybody has their areas, and one of the areas for you and me, and I think it comes from where we come from, is that to improve outcomes in a broader perspective rather than in a smaller perspective. And for that to happen, data is very important. I cannot go to a policymaker and say that, okay, you're going to do this. And we've been talking about pediatric surgery, insurance, etc., etc. What is the data that we give to our policymaker and our politicians? If we don't have that data, nobody's going to really take us seriously. It's everywhere in the world. That's how it is. So for us to do that, and it's very important for Shilpa, for you to be a part of this, because at the end of the day, I could do the research, I could collect the data, my team could do all that. I would not be able to change any policy because I do not live in a system or work in a system which is related to the government. You guys do, and that is why you guys are important. So for you guys are important in changing policy. So if I give you the data and if you change the policy, it works well for everybody. And that is the way I look at it. And that is the reason why I spoke to you and the last 1 year or so, is that this is something that we can do. And today I invite through this people to be part of the South Asian proposed South Asian pediatric surgical outcome study. We will be sending you the draft CRFs and the draft protocol. And if people can do that, then we have the data of what we are doing in our country and why we should improve it, if we should improve it.
Yeah, bravo. Thank you so much.
That's a wonderful lecture, Dr. Dhruv. You outlined all the difficulties in collaborative research, but collaborative research is probably the way forward. And I think to begin with, one can start at small level before we go on to the global collaboration or things like that. Absolutely. So I hope that from your lecture, people will think about this and maybe some positive outcome will come out in our country. Thank you very much.
Thank you so much, Professor Mahajan.
Thank you, Dr. Ghosh.
Thank you, Professor Buddhiraja.
Memento presentations.
Yeah. So, Dr. Dhruv, thank you very much for taking time and speaking to us. This is a small memento from Organizing Committee.
Thank you so much. And special thanks to Dr. Shilpa Sharma. She's been very, very supportive of our work. Thank you.
And thank you. And don't think your work is not reaching where it needs to. The paper was, I got a call from the DGHS and I made presentation for him and he presented it in front of the ministry. So that's how the COVID vaccination got a big boost.
Oh, you should send me a write-up of that, because I'm sure the rest of the world would love to hear that experience. Do send me a write-up about that, please.
Definitely.
Thank you so much. Thank you.
Thank you. Thank you.
So with this, we come to the end of first session. And I express my sincere thanks on behalf of Organizing Committee for all the chairpersons of the session one for conduct of the session. Thank you very much. So we will take a short break of maybe 10 minutes before we go on to second session. Dr. Shilpa?
Yes, sir. The program will be there online, but everybody can go out and come back, have a tea.
Yes, ma'am.
Professor Dubey, please take over.
Yeah. So welcome, Sangeeta, and please start your deliberations.
Thank you very much, sir. Is my slides moving, ma'am?
Yes, we can see your slides. You can read out your title as well, because we've not.
Yes, ma'am. Yes, ma'am. Sure. A very good evening, all. It gives me great pleasure to present a paper in this conference of research in IAPS, 6th conference. So the topic is systemic review and meta-analysis comparing the accuracy of urinary biomarkers in differentiation of uretropelvic junction obstruction from non-obstructive dilatation in children. With the advent of fetal ultrasound, the urinary tract dilatation is being diagnosed frequently, and this is one of the common cause of congenital anomalies of kidney and urinary tract in children. Non-obstructive dilatation due to transient hydronephrosis is the commonest cause, followed by uretropelvic junction obstruction. While differentiation of these two conditions is very crucial to prevent the loss of renal function, children are put through multiple invasive investigations like voiding cystourethrogram, diuretic renogram, etc. to arrive at a decision. Urinary biomarkers aid in a non-invasive assessment of renal damage, and the studies are promising in this regard. It is also useful in the prognosis of children following surgical correction. An ideal biomarker should be characterized by the optimal sensitivity, specificity, and area under the curve on ROC curves. So, in the past, there are studies focusing on various urinary biomarkers. The list goes on. But even with so many biomarkers, there is a lot of confusion over which one to choose. So, certain biomarkers are showing promising results, but few studies are limited due to bias and lack of consistency. Among these various biomarkers, we have selected certain biomarkers which were reported frequently in this decade. These are neutrophil gelatinase associated lipocalin, monocyte chemoattractant protein 1, carbohydrate antigen 19-9, kidney injury molecule, epidermal growth factor, and interferon gamma induced protein 10. NGAL is synthesized in the loop of Henle and collecting ducts. It is rapidly induced in response to any form of renal tubular injury. The level increases both in serum and urine following different abnormalities causing renal tubular injury. MCP is also increased in relation to tubular damage, and CA19-9 is getting increased in hydronephrosis and decreases after the relief of obstruction. Kidney injury molecule is persistently expressed in tubular cells until the damage is recovered, and prompt deduction in urine makes KIM1 an ideal marker of tubular injury. Epidermal growth factor is a polypeptide produced by the ascending portion of the Henle's loop and distal convoluted tubule. IP10 is a pro-inflammatory and angiogenetic protein, gets elevated in UPJO. So the objectives of this meta-analysis include to compare the commonly reported urinary biomarkers for the diagnosis of UPJO in children, to analyze the sensitivity, specificity, and accuracy of the common urinary biomarkers to identify the most useful one to diagnose UPJO in children. This meta-analysis was exempted from the IRB approval as it is a meta-analysis. We get the registration from Prospero. We used a Cochrane database, PubMed, Medline, and Embase electronic databases to collect the articles. The keywords used were uretropelvic junction obstruction, non-obstructive dilatation, urinary biomarkers, and children. The publication date ranged for studies from January 2010 to November 2021. Two researchers independently screened the articles for the review. We defined the study inclusion as population, intervention, comparator, outcome of the study design. The population included patients with hydronephrosis, either non-obstructive dilatation or UPJO. The interventions in the form of urinary biomarker assay with one of the agent of interest like NGAL, MCP1, KIM1, CA19-9, EGF or IP10. The comparator was between the outcomes of the biomarkers of interest. The outcome analyzed were the sensitivity, specificity, accuracy of the urinary biomarkers in the diagnosis of UPJO. Certain factors like caspases, prorenin, trefoil family factors containing papers were excluded as it was not commonly reported in this decade. So we followed this Prisma chart for selecting the articles. It was reviewed in 2 screening steps. We used these keywords to select the articles, and we excluded certain articles for adult outcomes, animal studies, review articles and guidelines, abstracts, conference proceedings, urinary biomarkers which are used in other causes like UTA, VUA, PUV. After this screening, we also excluded certain articles for incomplete data error, and finally we took 19 articles for this meta-analysis. The meta-analysis of sensitivity, specificity, accuracy was carried out using Meta Excel 5.3 software. The heterogeneity of various articles were reported using this I2 statistics with 0 to 25% as not important, 25 to 50% as moderate, 50 to 75% as substantial, and 75 to 100% as considerable heterogeneity. The difference was considered statistically significant if the p-value was less than 0.05. This shows the traffic light plot of risk of bias analysis using the Robust generic tool of all the 19 articles. We took certain factors like clarity on the study population, type of biomarker, biomarker methodology, whether it is clearly defined or not, cutoff value of the biomarker, sensitivity, specificity, data in each article, the diagnosis of UPJO, NOD clarity. All have been taken to make this risk of bias analysis. In this, 5 researchers have used the normal people as control. Remaining researchers have used the non-obstructive dilatation as control. 2 studies have given only the absolute urinary biomarker. Remaining studies have reported urinary biomarker creatinine ratio. And this is the forest plot showing the NGAL sensitivity and specificity. On the left side, you can see the I2 statistics to show the heterogeneity. On the right side, the 11 studies included for the NGAL sensitivity. It is showing the overall sensitivity of 0.78% and specificity of 0.76%. MCP, we included 7 articles for this to calculate the MCP sensitivity, which showed 80% with the confidence interval of 70 to 89%, specificity of 60%. KIM had both sensitivity and specificity as 75%. CA19-9 had 87% sensitivity and 74% specificity. EGF had 69% sensitivity and 74% specificity. IP10 had 74% sensitivity and 70% specificity. So we compared the specificity of the biomarker study, junction obstruction in children. The overall sensitivity was highest for CA19-9 and the specificity was highest for urinary NGAL. The researchers have also compared the combination of biomarkers and they have reported various biomarkers. Caracas supported IP10 and MC1. Oscuvancy supported KIM1 among IP10 NGAL MCP on CA19-9. UCell supported NGAL and CA19-9. He had studied children with a supranormal renal function and UPJO and reported reported that were better in predicting the requirement for surgical correction before deterioration of renal function. So in this meta-analysis, the overall accuracy was highest for CA19-9, followed by 77% for NGAL and 5% for KIM1. So we support a panel of biomarkers combining NGAL, KIM1 and CA19 for the best diagnostic accuracy. CA19-9 is taking the overall accuracy, followed by NGAL and KIM1. This meta-analysis has some limitations. Most of the studies included had only very small size and this may cause prone to error. Many studies had substantial heterogeneity. The cutoff value described within the groups for the several authors also reported the urinary biomarker levels from the renal pelvis of the obstructed kidney during the intraoperative period. Again, this may cause false elevation of the urinary biomarker because of the long-term obstruction at the pelviureteric junction. And we take this urinary biomarker because it is non-invasive. We can avoid the invasive investigations. When we take the urinary biomarker from the renal pelvis, that is not serving the purpose. So we should focus on the future research. The ratio of the urinary biomarker has a wide range, and this could cause confusion among the further researchers on what would be the ideal cutoff. So we have to standardize the kits and values to minimize the variability in the cutoff value. The value may vary between infants and older children, and further studies should focus on age and sex specific normal range in children. We should also focus on large sample size to study the urinary biomarkers. The focus on identifying the best panel of biomarker combination rather than checking the individual biomarker would be helpful to improve the accuracy of the result. So I conclude saying that the overall accuracy was highest for CA19-9 followed by NGAL and KIM1. Based on the current meta-analysis, we support a panel of biomarkers combining urinary NGAL, KIM1 and CA19-9 for the best diagnostic accuracy of UPJO in children. These are my references.
So,
thank you.
Thank you, Dr. Sangeeta. I'm having one question. May I, Shilpa? May I go ahead?
Yes, sir, definitely.
Hello.
Yes, sir, you can.
Can we rely upon these biomarkers because the sensitivity and specificity is just 70 to 80% and 78%? So can we label the any POJ obstruction as an obstructive uropathy or obstructive dilatation, and can we proceed for the surgery without any other investigations? Or do you suggest it for the sake of, say, in the follow-up, we can use such biomarkers as a non-invasive things, whether the further damage is going or not? Dr. Sangeeta?
Generally, for the screening test, if the sensitivity is more than 80%, we take that as a screening test, sir. Most of the studies have shown in the range of 70 to 80%. So probably for instead of going for the invasive investigations like renogram, EC scan or DTPA scan, this has been used, but we have to do further research on the larger sample size to come to the conclusion. This can be used to diagnose the uretropelvic junction obstruction. On follow-up, following the pyeloplasty surgery, to know the outcome also this has been studied. But still we need to do the study on the larger sample size.
No, but for diagnostic purpose, if you are going with this biomarker, and as you said, it is a sensitivity, it is just 70 to 80%, and specificity is 70 to 80%. Can we rely on that and go for surgery? Or
May I add a point, Dr. Dubey?
Yeah, please.
Can I add a point? See, there are
please, sir.
There are studies which have combined the biomarkers, not just one biomarker. They've used a panel of biomarkers. So when 3 biomarkers are used together, the sensitivity reaches 95%. So there are studies which support use of 3 ABC, like BCD. If all 3 are elevated or all 3 are down, then the sensitivity specificity is like 90 to 95%. So what we could do in this meta-analysis is because only one biomarker is considered. But when we have a panel and when we compare the panel, then probably we will reach a sensitivity of 90 and 95. Now, my contention is probably it will not replace a decision for surgery. But what it can replace is, like, for example, somebody with a moderate hydronephrosis, where we want to avoid a repeated DTPS scans, or we want to avoid quite a bit of invasive tests, like during a follow-up. So we know that the trend is correct. So in those cases, at least we can avoid multiple tests.
Yes, I accept that. That's what I was also saying, that it can be used for the subsequent follow-up cases where the equivocal results are there in the CT scan or even subsequently. One more question is there. All the biomarkers, as we know, that is result of damage to the renal cells. So such damage can occur with the even nephritis or even diabetes mellitus, or even in case of vesicouretic reflux. So how can we differentiate this biomarker? Because they may be present with such non-obstructive medical or surgical cases also.
Can I add a point there also? Sangeeta, 1 minute. I just want to add the point. See, you have an excellent point there. See, this biomarker panel is useful only for a single kidney problem, which an isolated hydronephrosis. Or if somebody has got a coexistent vesicouretic reflux or UTI, or even a bilateral PUJ obstruction, then it becomes totally useless. Because you are having multiple variables here, and then multiple factors can influence the biomarker, even the age and sex of the patient, and even the general condition. Like, for example, what you're told, like if somebody is unwell, the general inflammatory markers are increased, because that can be biased. So there are a lot of negatives here, a lot of biases are here. So it is probably useful only in a single kidney hydronephrosis who are absolutely having no UTI or any other problem. Dr. Sangeeta.
Yes, sir. As you said, we have certain limitations for using this urinary biomarker. For example, CA99 can get elevated in children who are having pancreatic problem. Urinary tract infection again can have increase in NGAL level. So the urine culture should be sterile before going for the urinary biomarker study. It is there.
Is there any questions from the
Dr. Sarin has got some valid points, May.
Yeah.
Dr. Sarin and then Dr. Ghosh.
My question is, when we say that we do not have globally accepted cutoff points, then it is very difficult which reference to take and then work on that. You know, if we had some consensus on the cutoff values of all these biomarkers, then it would have been fantastic. Number two is, I totally appreciate this work, because even now we realize over the years that, the renal dynamic scans do not work too well in many patients with us, especially for follow-ups or otherwise when they come pretty late. And we presented our paper, which was included in the UC Chakravarti Award in the conference in 2021. So I think now we need to start looking for things beyond the dynamic scans, which could help us to make a diagnosis and also to let us have follow-ups done later.
Dr. Dhruv, your hand is raised. Do you want to say something?
Yeah, I think that's a very interesting study, Dr. Ramesh. And just 3-4 small queries. Obviously, the meta-analysis has looked at various combination of biomarkers. Did you find in your analysis that there was a particular marker which sort of increased the sensitivity and specificity across the board? In terms of when that marker was included in your in the various combinations, was it more dominant in increasing the sensitivity or specificity, or nothing like that was there? My second question is, do you think using urinary biomarkers would be more useful in ruling out an obstruction in a specified subset rather than ruling in? My third question there is, do you think a prospective study using many of these, or rather more commonly used biomarkers, would help you do a logistic regression to make an equation which will then set cutoff points, taking from Dr. Sarin's point about wide cutoff values, whether we can have cutoff points? Are you thinking of proposing a study in your institute like that, where you would look at these prospectively rather than a review or a meta-analysis?
There were collaborative study could come in, no?
Yes.
We could collect some 1,000 patients, let's say.
Exactly. So your sample size would increase, and then you would be able to do. But the thing was, in the meta-analysis, was there a specific marker which was sort of dominant in increasing the specificity and sensitivity in your combinations?
CA99 had the maximum sensitivity, and NGAL had maximum specificity, sir. This is
so if you remove that, yeah, if you removed either one of them, did the specificity and sensitivity of the combinations go down?
Can I answer that?
Yeah.
Yeah. See, Dr. Yogesh, I'll first answer Dr. Yogesh's comment. It's a very valid point, because one of the striking things we found in this meta-analysis was the cutoff values proposed by the 11 articles for NGAL varies from 0.9 to 900. How can such a big variation can be there? Each study has got their own cutoff, and they are having a sensitivity for their own cutoffs. But that doesn't mean that there is a universal cutoff. The same is the case for MCP, same is the case for KIM, and probably CA99 is the one which has got the lowest variation in the cutoff level, and IGF and IP10 are the other two where again the range is variable. We really don't understand how a cutoff can be variable. I mean, this is very difficult actually, because if a creatinine is 1 is cut as a cutoff, we can accept. If a creatinine as 0.1 is cut off for someone and 100 as a cutoff for somebody else, that is totally unacceptable. Now, coming to the combination of biomarkers, this particular meta-analysis looked at only at the individual biomarker sensitivity reported from each paper, because the combinations have not happened exactly the way we wanted. Because some papers have combined MCP, KIM, and IP10. Some papers have combined IP10 with EGF and NGAL. So there are multiple combinations and permutations, and that depends on the various, the institutional protocol. So we really don't have a combination to compare. Then the third problem is there are variations based on age and sex and then underlying condition of the patient. So we still do not have a normal cutoff. So we have to look at like 1,000 or 2,000 normal children and establish normal cutoffs for all these patients. And until then, we cannot go further, because everybody keeps on studying biomarker without looking at normal cutoffs and variations with physiological and pathological variations in their body habitus. So without that, it is very difficult. But what we found is the individual sensitivity, specificity is highest for 3 biomarkers. See, if at all we are going to start a study, we are going to use the top 3 biomarkers and use them as a combination in our population next. That is our next project.
Yeah, I think that is actually the point I was trying to make, in the terms of the fact that there doesn't seem to be a uniformity in the various studies that were being done. And also probably you will have to also look at the subset of patients which you are going to study for this rather than everything else. So you just pick a subset of patients, like you said initially, what was unilateral obstruction without a reflux, without UTI. So those will be your exclusions. And if you have those inclusions, and then over a period we could run a meaning across the country and see whether we can get up with what the common biomarkers that you would identify. Thank you.
Thank you, sir.
Thank you. I welcome our chairperson, Professor Kaushik Saha. He's a professor from NRS Medical College, Kolkata. The next speaker will be Shashi Mewar. Sangeeta, could you please unshare your screen?
Yes, ma'am.
Shashi Mewar is from the College of Nursing at AIMS.
Yeah, hello, good evening.
Faculty. Good evening, sir.
Hello. Hello. Should I
Yes, sir.
start?
Yes, sir, you can start, sir.
Yeah, the first paper is Pediatric follow-up home-based care, a review by Dr. Shashi Mawar, who is Associate Professor, College of Nursing, All India Institute of Medical Sciences, New Delhi. Dr. Mawar, please.
Dr. Shashi, please unmute yourself. We can't hear you. Your slides are visible. IT, please allow Shashi iPad to unmute. Shashi, open your Zoom, unmute yourself, and then share your screen. Or if you can see the icon, just unmute.
Okay, done now. Am I audible now?
You're audible well, but we can't see your screen. So share your screen again.
Okay.
She's actually using a phone.
No, I'm using an iPad.
iPad use.
So like I've been using this since
Okay.
that podcast. Is it visible now?
Yes, it's opening.
Okay. Yeah. Done now?
Yeah, done. Please begin.
Okay. Okay. So first of all, I would like to thank the organizers for giving me an opportunity to speak at this forum. This is basically not a research article. This is a critical review of some articles which have been done in the past recent years for pediatric for surgery patients for follow-up in their home-based care. So basically, like the nursing care of children, it is quite challenging because of several peculiarities in social, in psychological, emotional, as well as physical reactions, and including their response to illness. Also, several times it has been noticed that the children, they are unable to provide necessary information related to their history as well as clue to assist in evaluating and management of their health problems. So therefore, like they are not as a single unit, they are as a two patients with us, that is the family or the mothers or the caregivers along with the child. So in recent years, there has been a considerable change in the concept of elective surgery with an increasing emphasis on minimal hospital stays. So because there are several kinds of complications associated with long-term hospital stays, so it is opted that the child should be discharged from the hospital as soon as possible, and a continuity of care should be maintained in their home settings. So the concept of enhanced and rapid recovery after surgery is constantly developing as well as improving based on the technological advancements. Therefore, there is a shift towards the community health services from the hospital-based care. There is a need for the delivery of surgical care that is more efficient and cost effective and has high degree of patient satisfaction with excellent outcomes, which is evolving in the current surgical practice model. So as a quality initiative, it may be examined whether an allied health professional telephone visit or app-based strategies or teleconsultations or video-based programs could safely substitute for an inpatient clinic visit. Post-operative follow-up is essential for monitoring successful outcome of surgical care by identifying complications in a timely manner and providing continued care wherever required. Because if a child is not monitored effectively post-operatively, then there are chances that there might be more of complications. So patients, they recover easily in the comfort and the familiar environment of their homes under the care of their family. It is not only their early recovery, but it also obviates the separation anxiety that comes from the hospital admissions as well as follow-up visits. So the methods which were adopted to do this critical review were that most of the research articles which were related to the strategies implemented to assist home recovery post-operatively, they were searched and reviewed critically. Several strategies were found in this home-based care. Few of them included planned discharge counseling, mobile app use, teleconsultations on home care, video-based teaching programs for several procedures which were to be performed by the caregivers in the home settings. All these strategies have been stated to have some advantages as disadvantages, and therefore they need to be tested empirically and implemented specifically for a specific person. All strategies, they aim to deliver services more efficiently, accommodate more patients on as the population increases, conserve resources, as well as deliver optimal care without compromising patient safety. So if we see the critical review of these articles, the results show that attending the follow-up clinics is the gold standard, as it is difficult for the surgeons to rely on the assessment of parents and caregivers of children's wounds. But a study done by Kasaman BP in 2012 really revealed a reliable parental assessment over a guided telephone interview. Post-operative follow-up by telecommunication is acceptable and can safely replace traditional clinic visit for low-risk procedures. And these are also two studies done in 2013 and 2016. Telephone follow-up frees up clinic space for patients who really need it. It is time-saving, cost-saving, as well as efficient for the patients and healthcare system. This has also been the results of two studies done in 2017 and 2013. These are some of the, this is a result of a qualitative study where the parents have given the reasons for their preference for telephone follow-up or clinic visit. So in the telephone follow-up, there's several reasons which have been stated by the parents. In 2021 itself, it is published. And they have said that there's a satisfactory patient recovery. It is time saving, as well as based on the previous experiences, they are able to take care of their child at home comfortably. Other than the telephone interviews, the other strategies which have been implemented in various articles, they are like planned discharge teaching programs. If they are deliberated effectively before their discharge to assess the understanding of the family and the acceptance of the instructions, as well as some time is given to clear their doubts, even that helps in providing good home-based care. So this was a study done by Vidya T. Raman in 2017, where the caregivers, they watched a standard video teaching available on YouTube. And this was developed in conjunction with the hospital educational and technical support staff. And the caregivers, they were highly satisfied with the post-operative care teaching. And the results also showed that there were no complications which were reported by any of the patients who had seen these videos. And another strategy is automated interactive voice response telephone calls. Following the discharge, they also address some patients' requirement for further support, specifically for few areas like problems related to surgery, worsening symptoms. That is what they should be performing if any of the symptoms they start worsening, or if they have any queries related to the medication issues. So if they are answered through the interactive voice response calls, they have also been proved to be effective. Then in a few studies related to the use of telemedicine for post-operative monitoring to provide safe healthcare from far off distances due to the recent advancements of mobile technology, it has been seen that there were no post-operative complications identified by the telemedicine evaluations, nor were any complications missed or misidentified. There were no readmissions, no additional clinic visits, as well as not even emergency department visits. Another study in 2017 also shows that telemedicine saved on an average of 119 miles of travel distance, as well as $357 per person. Novel applications, they are also used to improve the post-surgical care with new generation mobile phones. And mobile applications are also available, which are portable, convenient, popular, and they also provide precise information and are interactive. Therefore, they are being gradually implemented in the home-based follow-up care. Use of mobile health technologies for the post-operative care showed decreased complications in a study done in 2017. Patients were satisfied overall with the mHealth interventions and preferred over the traditional follow-up approaches in a recently published study in 2020 itself. So how we can implement these strategies? These strategies may be implemented through nursing personnel working in the hospital pediatric units itself, and they can do a follow-up through the telephones or through the a continuous monitoring can be done through the applications, and based on the assessment, interventions or specific treatments can be provided, or it can be implemented through the community health nurses who are working in different areas, as well as non-professionals also can take up these kinds of roles after some specific training or for the specific care of these children. Depending upon the level of care and close monitoring needed, the home-based follow-up care can be planned to maintain a continuity of care. So to conclude, a continuous care model from the hospital to home leads to decrease in complications for pediatric surgery units, and as it enables the satisfaction in care as well as confidence towards the health personnel. Thank you.
Thank you, madam, for the nice presentation. In this era of telecommunication, at least 50% of the patients can be followed up by telemedicine only.
And specifically in this COVID era, when patients, they were not able to visit the hospitals, if we had these applications in advance, or if we had some kind of structured teaching programs in advance, it would have been of help.
Plus, with the help of local GPs and local nursing staffs, we can very well use this program.
It can be implemented.
Thank you. Any question?
Yeah, can I ask? Dr. Shashi, your review or literature article is mainly from the foreign articles or European journals or American journals.
Yes, sir.
Do you suggest anything for Indian circumstances? Because mainly they are illiterate, or they may not have the access to the smartphone or something like that, as you might have seen in your own setup or in the AIMS setup. As we also face in this periphery and the Chhattisgarh area, most of them are not that much literate. So what do you think? What do you suggest for Indian circumstances? Because ultimately this is the final or future thing, because we are having a lack of resources, so we need to have a lot of beds for all the patients, so we need home care. So what do you suggest for them?
Sir, like nowadays, the use of mobile phones is increasing in India also, and not only in the urban areas, but even in rural areas. Presently, I'm already doing a study on one Utsarjan app on nephrotic syndrome children. So to the best, like what we have done is, most of the parents, they are using that app very frequently, and we are monitoring their condition on everyday basis for a follow-up of 2 months, and they are using it. But the only problem which we are facing is sometimes the data is not working effectively in our country. But the use of smartphones is there with most of the patients nowadays.
How you tackle their sympathetic level, because most of the time they are over concerned. Every other time they are just calling if you are allowing them, because this is what we face over here.
Yes, even I have faced that, and they have been just calling for their not only for that particular child is enrolled with us, even for their other health conditions. So I always tell them that, okay, this is the only time which will be allotted for talking to you, and only on this specific topic. So once or twice, once it is refused, they do not call us back.
Okay.
Because if we entertain, they will keep on going for it.
Yes, they will keep on going, and at odd times.
They will refer the relatives.
Yeah, yes. So is there any other questions from the?
I had a question. Two of the questions have been already asked by Dr. Dubey. My another question was, we deal with the a wide spectrum of the disease, starting from smaller, less severe disease to more severe disease. Will there be any legal consequences, legal issues if we miss out something, and it will be documented, it will be recorded? So is there any thing which you have thought about this particular aspect has been thought about, especially in our country?
For this, we can always inform that this is our limitation, and beyond this, you have please visit the hospital other than using that.
Is it legally permitted to be used?
We get an ethical permission, so it is a legal permission. Before doing anything, we take an ethical permission.
No, actually, may I answer this? Actually, he's not asking about the ethical part. He's asking. He's concerned about the medical legal part, because nowadays we are pulled up in a consumer court. So, but in this COVID situations, there is a lot of change, even on the government side also, and they have allowed even the telecommunication, especially during COVID time in last 2 years. So yes, we can say up to some extent, we can go ahead with the advices, but we should avoid to write down any specific prescription sort of thing or any specific advice, but it is a general assurance, general advice, general care in the post-operative phase or even in the follow-up phase. Am I right? Or do you want to add anything in this, Dr. Shashi?
You are right in this, and we can always take a consent that in advance that they are going to follow up to the limitations which are there available on through the telemedicine. Within those limitations, and if it is beyond it, they should be visiting. Otherwise, like some complications or something, if we are able to assess, we can always tell them to come to the emergency or to the for the follow-up clinics.
Yes.
Yeah, just for the minor queries and general queries, telemedicine is all right.
Yes.
So there is a Telemedicine Society of India. We recently had a big seminar, national one, with the NAMSCON, and there are many people working on this. They have established telemedicine centers in different parts of the country. They are evaluating all the cardiac patients and even diagnosing diabetes within families. So guidelines have to be in place. Once you have the guidelines and you have telepresenters for those who cannot understand what you're talking, and each meeting has to be recorded. So you have to tell them that this is being recorded. So whatever is being spoken about, and then the underlying lining that this is not for medical legal implications are not there.
Yeah.
Thank you. We can move on to the next speaker.
Yeah.
I'll be presenting the slides, and Vora sir will be speaking. Dr. Vora, are you there?
Yes, very much. Will I be also see the slides, and you, all the attendants, will also be able to see the slides?
Yes, everybody is able to see the slides.
Okay. May I start?
Yes, sir.
Please.
Good evening, everyone. See, this is a presentation on innovative food fortification with vitamins B12 and folic acid to eliminate their deficiency in women of reproductive age to prevent neural tube defects like spina bifida and hydrocephalus. We have been working on this after the International Conference of Spina Bifida Hydrocephalus in 2018, where we really discussed about the problem in India of fortification of food. And then we decided, why not try tea as a vehicle? So next, please. Next. Shilpa? Go to the next slide, please.
Yes, sir. The next slide is there.
Yeah. See, as we pediatric surgeons know, there is a tragic and silent epidemic of neural tube defects in India, with over 1 lakh babies being born with neural tube defects per year. Actually, they at present birth rate, it comes to 1,15,000 per year. That means nearly 300 babies born every day. And this has been going on for last 30 years. Even after the scientific community discovered that the cause for this major birth defect is folic acid deficiency before a woman becomes pregnant. That is in preconceptional period if the folic acid is deficient. But in India, in last 5 to 6 years, it has been found that B12 deficiency is also a factor as an etiological factor in NTDs. So if we have to correct this deficiency, we have to give these women of reproductive age vitamin B12 and folic acid. Next. And it's very cheap and simple supplement which we can add to the food, common food which is consumed all over India in all parts of states. See, there are, before going to tea, I'll tell you, there are some logistic deficiencies in Indian society. That means, say, suppose the Western countries have fortified their food with wheat flour with folic acid. Is it possible in India? In India, we have thought of fortifying wheat flour as well as rice flour and making rice again and giving it to it. But the constraints are, in India, we eat 6 types of grains. So which ones will you fortify? Secondly, there are different varieties of these grains. So each geoclimatic area has a different production of farm produce in that area. So people are used to eating that. So it's not possible. Secondly, if you do any fortification of a grain, it has to be bought from the market, which is not possible. What we do in India is we, according to our financial position, we buy either 1 week's supply of grains or a month or a few months and get it locally processed in local flour mill. So nobody goes to the market and buys wheat flour or rice flour or anything as they do in Western countries, because they all eat bread. So bread is bought from the outside, or even if they bake bread, they can buy wheat flour, not like us. So that is out of question. Third thing is, financially, for a nation to give fortified grain for 130 crore people, to supply that only for the women of reproductive age, which forms only a percentage of that population, it will be too expensive. So considering all these things, and the last thing is why it is not being implemented is also, in addition to these constraints, it is the complacency on part of the health providers and healthcare systems in India. This includes us also. We cannot just blame the government or FSSAI, but we are also included in that. We have not done for last 30 years. So we thought of using tea as a vehicle because it's ideal, implementable, cheap, and available all over the country. And it has to be bought from the market. Nobody prepares tea powder at home. And it is produced only in 4 states of India, where it is easy to fortify it with vitamins, whichever vitamins we want to fortify it with. So, and in the lab, we tested that tea, the vitamins are stable in boiled water it doesn't change the odor, doesn't change the color, doesn't change the smell, taste of the tea. So nobody knows that there is some vitamins added. And besides, even by FSSAI standards, vitamins are not drugs. They are food supplements. So instead of asking women of reproductive age to take tablets, which nobody does. I mean, we have been trying this for last 15 years through our foundation. But we are not being successful because women will not take tablets. So tea fulfills all criteria, is available all over India, even in the villages, and has to be bought from the market and is consumed by majority of women, except for 4 states in the southern India, where in next phase of our research, we'll try to fortify coffee also. Generally, a cup of tea is made with 2 grams of tea, and it is easy to fortify on a large scale. And then 2 grams of tea, either in the tea bag or loose tea, will contain the vitamins which we have added. So B12 and folic acid consumed daily over 3 months will correct deficiency of these vitamins. Next. So to prove this, we did a preliminary study. We did preliminary study on a group of women. As it is customary, we divided them into experimental group and control group. So, and before that, I must mention that it is proven, it is simple, implementable, and cheap solution in India. Correction of preconceptional deficiency of vitamin B12 and folic acid, and target group will be women of reproductive age from puberty to family planning operation. So if we advise women to consume this tea, which they can buy from market, like tulsi tea, ginger tea, or whatever tea, there are so many on the market. So they will go and buy vitamin tea for them. Next. So in our preliminary study, which was registered with the Clinical Trials Registry of India, it was carried out on 2 groups of, nursing students and homeopathic college students, because we could give them tea under supervised conditions in the college canteen. And there we wanted to find out which was the correct dose of vitamins to be given in 2 grams. So we divided them to 2 groups. One group was given for the experimental part of that group was given with folic acid 1 milligram and B12 either 0.1 milligram or 0.5 milligram. That was the difference in doses of B12. But a dose of folic acid was kept same, that is 1 milligram. And as you know, what you need is requirement is only 0.4 milligram of folic acid daily. So this is given per cup of tea. And both experimental groups had folic acid 1 milligram, and control groups were given unfortified tea. And tea was served always in under supervision in college canteen. Next. So blood tests were done for before trial and after trial, and hemoglobin, peripheral smear, B12, folic acid levels were measured before the trial and after the trial. The statistical analysis proved the efficacy of fortified tea in correcting deficiency of B12 and folic acid in both the groups, in spite of the fact that B12 was given in different doses. The pre-trial study also showed that 70% of women of reproductive age in India had deficiency of both these vitamins. This study was published in June this year in BMJ. I have given the link for that. Next. See, having done this study and proven statistically, as you can say that it is P is more than 0.05. So it is good, and we have shown this on the graphs in that paper. Please refer to that paper and you will get more details about our tests. Next, what we want to do is to prove that, see, for this first trials, preliminary study, we used tea bags because it was easy to fortify tea bags with micropipettes and correct dose could be given in that. And we were sure that the per cup dose will be exact. And that lady would take the 1 cup of per day. But now in India, we don't consume tea bags. We always use loose tea for preparing tea. Everybody has a different taste. Some people take 1 teaspoonful, some people take 1.5 or 2 teaspoonfuls. So we have to prove that the loose tea, 2 grams of loose tea would also provide the same dose which a tea bag would do. So we have now planned, and they have been registered with the CTRI. This is being done in Sangli district. We will also be doing it in different 3 centers in India just to make it multicentric trial. So in each 2 subgroups, experimental and control, with same corrective dose of vitamins per cup of tea daily for 3 months. See, in our preliminary study, we gave it for 2 months, and even 2 months fortified tea was enough to correct their doses. But when we want to give it at home, we have to account for some deficiency in preparation of tea and other things. So we want to give them at least 3 months prior to conception. But our advice would be that ladies of reproductive age, as soon as they become marriageable, they should start taking this tea daily, so that by the time they reach the stage of getting pregnant, they will have normal levels of B12 and folic acid. Next, please. So now let us tell you about all with B12 of 1 milligram and folic acid 1 milligram per 2 grams of tea powder, either in tea bags or loose tea per cup. This we have decided, because we want to give extra B12 to compensate for loss of absorption from the GI tract, because we all know that B12 is not as efficiently absorbed from the intestine. So in first group, we'll use tea bags. Second group, we'll use loose tea, 2 grams per cup. And both these will be done under supervision on students of medical college and dental college, because there we can really supervise in their canteens. And this will be done for 3 months. Because we consider that this corrective dose should be given for 3 months. The third trial we will do again with loose tea, but we will choose slightly knowledgeable village women who will prepare tea at home, but they will not mix up with other teas made in their home, and they will follow all the details of making tea, not boiling it too much and this, that. So we'll prove that tea made with loose tea, even at home, with some knowledge of good preparation, also will serve the same purpose of correcting the deficiency of these 2 vitamins. And so having done this study with a corrective dose of this, this will be followed by RCTs with maintenance dose on group 1 and 2 for 8 months, because we need not give corrective dose in the tea for all women for all the time. So once they correct their vitamin levels, they can be given lower dose for maintenance. This we will prove by giving that dose for 8 months and doing again their estimation of folic acid, B12, hemoglobin. And this time we are also going to do estimation of homocysteine, because if homocysteine is raised, that will means there is definite folic acid deficiency. And having corrected that deficiency, if we see that homocysteine has gone down, that will prove that it is being utilized by the body. Next, please. So now, after this, what we will do is we'll do some multicentric trials, and then our next step will be to convince the Ministry of Health, then the scientists of FSSAI, that is Food Safety Security Association of India, which will direct then Tea Board of India to market vitamin fortified tea by 2003. We are hoping that we'll finish all this trial. Actually, we should have finished this year only, but because of COVID, we could not do the trial on students who had gone home. So we are hoping that we'll carry out these trials from January, and whole year 2022 will be required for all these studies. And along with that, we'll do survey in those villages comprising of 75,000 population, which will have at least 15,000 families, where we will see what is the incidence of neural tube defects in previous 1 year of study. This will give us an idea. When we introduce fortified tea, then we'll see that there is reduction in the incidence. I think I have given enough this information about our project and research, and it has been reviewed by BMJ NPH and been published as original research and given to nearly 5,000 outlets through their press, so that people in other countries and other states of India would also take up this research and do it. Pediatric surgeon from Dibrugarh, Dr. Hemant Datta, has also come forward, and he is going to carry out similar trials in Dibrugarh, which is surrounded by tea gardens. Okay, any questions? I am willing to answer to my capacity. Thank you, Shilpa, and thank you all of you.
Thank you, Doctor, for a very innovative idea. But I want to know when it will be available in the market. How you want to level it? Like tea for male, tea for women, or tea for female? I mean,
yeah. See, no, we don't need to make difference between the women and men consumers. Only thing we can do is we can give just a second. I'll get the picture right. Sorry, yeah. Oh,
it is now upside down, sir. Yeah,
it's
now
better.
corrected.
I can see my picture better. So we'll have two teas available on market. One will be for corrective dose, with corrective dose. Another will be little smaller concentration of these two vitamins. It will be called maintenance dose. So there are other advantages if people like me who are beyond 60, who take this, the incidence of thrombosis in our blood vessels of brain or heart, it will be less, because homocysteine will come back to normal, which is a factor for causing this clotting. Secondly, the fetus who gets these vitamins in the right doses, they will have, not only they will escape the NTD, but they will also have brain development proper. So woman will not give birth to a child who will be slightly mentally retarded. That is another advantage. Canadians have shown that the deficiency of folic acid also causes central line defects like cleft palate, heart septal, cardiac septal defects, and gastroschisis.
Yeah, doctor, what is
So,
that part is appreciated?
we have,
well known. Yeah.
Beg your pardon?
Yeah, that part is well known and well appreciated.
Okay.
What I want to
I'm
know,
having one.
how will you want to market it? In which way you want to market it?
No, we'll ask tea boards to come out with this sample with this concentration of vitamins. It is
So,
there. They will
so
fortify it on a larger scale.
It's like salt. You can like salt is fortified with this iodine. So vitamin. So in the same way, all the tea can be fortified.
No, not all tea. This will come out as a special tea. So those who want to use it, they can buy it from the market, and the whole family can take it. Suppose in my family, there is my daughter-in-law is thinking of getting conceived. I will ask her, or even my daughter who is marriageable, I will ask her to consume this fortified tea all the time, so that when she reaches her in-laws and wants to have a baby, she will have good levels of these two vitamins.
Sir, shall we call it a fortification or supplementation? Because fortification is usually universal and homogeneous to the country, like iodine in the salt. And here you are making of a different concentration or different strength. And you are leaving it up to the population to choose whether they want it or not. So why would they go for a tea instead of direct tablet if they are so much aware of?
You mean to say tablets?
No, I mean to say folic acid. And see, if everybody is aware, suppose somebody is going to pick up the tea for the sake of that she's going to conceive, she will better take the folic acid and vitamin B12 tablet instead of taking tea, because the requirement is 0.4 milligram. We are not aware of the pre-conceptional status of the folic acid, and then subsequently we are taking 1 or 2 milligram every cup of tea. I don't know how much cup of tea is everybody is taking. It depends upon that. So I mean to say, if fortification is not there, so fortification means universally it is fortified with the tea, and everybody is taking it.
No, it is up to the FSSI and Tea Board of India to suggest and come out with the whole tea fortified with these two vitamins. As we have salt fortified with iodine for whole population, you can also have that, but it will increase cost. Very little cost will increase, maybe 5% or 10%. That will not affect the population. And one more thing which you have suggested about the instead of fortified tea, why not use tablets? No, sir, we have failed in that. It is very difficult to make women aware all throughout India. Government has got schemes to give tablets to all girls in the students in the high school and colleges. But even there, they don't take it, because the iron is added to that, and then they have some tummy upset. They don't take it. We have done a lot of study on that, and that's why we have failed, and we still have the higher incidence of neural tube defects in India, in spite of this for supplement of tablets in schools, because it cannot be really taken to the its desired effect.
I do appreciate, sir. Let me put it in a different way. Now, instead of government promoting the tablet, taking the tablet, the various company will promote like this is the pregnancy tea. You take it right from 15 to 45 years, like that.
I think
and then there will be more awareness in the
I think
scheme.
Dr. Vodadas, I mean, it's going too long.
I just answered the first question. His idea of promoting tea, fortified tea, will be taken by the tea companies.
Doctors, yes, that's what I'm saying.
We don't have, they will advertise. They will advertise on the TV as well as in newspapers, in all media. Take this tea and your baby will be normal. See, our job will be less. So it cuts both ways. We are giving them tea and see, actually, Director of Tea Board of India,
there are so many other papers also.
Director of Tea Board of India has directly telephoned me when he read that article, and he asked me, when are you going to direct us? Because they are also eager to come out with this specialty. So our job will be easy.
Yes, sir.
Thank you very much, sir. There are a few questions in the chat box, so you can answer that regarding tannic acid and absorption. I think we can move to the next session as our speakers are here. We thank the chairpersons of this session. Thank you very much.
Thank you very much.
Thank you.
I hand over to Professor Mahajan to invite the chairpersons. Professor Bajpai is the head of department and professor at All India Institute of Medical Sciences. Welcome, sir. And we have Professor Rajendra Saudi. He's a professor at GMCH SSH Nagpur. Professor Bajpai will now introduce the speaker, Professor Agostino Piero.
Good evening, Shilpa. Thank you very much for putting up a wonderful program and inviting Dr. Piero and other guests. It is my privilege to welcome Dr. Agostino Piero. He doesn't require an introduction. It's a tall figure in the fraternity of pediatric surgery throughout the world. And he is a very specific breed of scientist surgeon, which is very rare breed, but it is a very important breed because for the kind of work that surgeons do, it is us only who has to come out with research ideas. Otherwise, nobody will do research for us. And Dr. Piero has done this and more. He's also a minimally invasive surgeon. And in his today's talk, he will be discussing necrotizing enterocolitis, which has a very high mortality in premature infants. Its pathophysiology, which is multifactorial, often controversial. It has geographical variations. We see more of that in the West, but little less here, but nevertheless, it is increasingly seen. So the kind of research which Dr. Piero is involved with is help us understand the epidemiology, risk factors, pathophysiology, treatment strategies, and future directions. Now, this is just a welcome note to him. He has got a huge biodata. I'll request my co-chairperson, Dr. Rajendra Saudi, to introduce him to the audience in greater detail, please.
Good evening, everyone. It's a great pleasure to introduce Professor Piero, who is Professor of Surgery at the University of Toronto, Pediatric and Neonatal Surgeon and Senior Assistant Scientist at Hospital for Sick Children, Toronto. In addition, he is professor and full member of Institute of Medical Science and Department of Physiology, University of Toronto. Previously, Dr. Piero was Nuffield Professor of Pediatric Surgery at Great Ormond Street Hospital and University College of London, United Kingdom. His clinical interests and expertise are neonatal, pancreatic, minimal invasive, and neuroblastoma surgery. Among many prestigious affiliations, he is the recipient of Royal College of Surgeons of England and Edinburgh Fellowships. He is also an editor-in-chief of Pediatric Surgery International from 2018. In 2016, in recognition of his clinical and research activities, he has been bestowed for his services to medicine and charity honorary officer of Most Excellent Order of British Empire by none other than Queen of United Kingdom. In September 2021, he has received a very prestigious Rehabin Medal from European Pediatric Surgical Association. And interestingly, he is the only Canadian recipient or the first recipient to receive this award. With over 450 publications, including peer-reviewed articles, books, and book chapters, Dr. Piero is an internationally recognized pediatric surgeon. He has always focused on research that maintains strong translational potential. His team has carried out research which has received very competitive peer review grants, which are 71 in number, and the value of 22.1 million Canadian dollars, for which he has received for those grants, which include also a very prestigious one, CIHR Foundation grant. His team discovered a very novel treatment for NEC, for which we are all very eager to listen to, where remote ischemic conditioning, and for which also he has received a grant, which will now see the clinical data. With these few lines of his biographical sketch, I have a great pleasure to invite Professor Piero to deliver his lecture. Professor Piero, please.
Well, thank you very much for your nice presentations, Professor Bajpai, Professor Sanji, and thank you a lot to Shilpa for the kind invitation. It is a great pleasure to talk to you in India. It is a shame that now, because of COVID, I cannot be there in person. I've been in the past and enjoy your country tremendously. So let me share the screen and see if you could see my slides.
Yes, your slides are visible.
No, just a sec. Let me, hold on a moment. Okay, share the screen, this, share, sorry, just a second. I think I have a problem with the sharing of my...
It's fine, we can see your slides now in slideshow, they're visible.
You don't see the second slide, right? You see only one.
Only one slide, yes.
Only one slide, okay, perfect. Thank you very much. So I'm going to talk to you in this lecture about something translational, which is very close to my heart, and I think we are at the brink of discovering something new for necrotizing enterocolitis. Do you see the second slide? Yes.
Yes.
So the cause of NEC is unknown. This is a disease which has high morbidity, adverse outcome, the highest mortality in neonatal surgery, and a recent paper in New England Journal of Medicine showed that the cost just in the United States is between 500 million and 1 billion per year. So it's huge. So in surgery, what we see isolated neck with a perforation, we see multifocal neck, or we see the devastating panintestinal disease. And the mortality, this is an old paper which I wrote, you could see that the mortality goes from 12% to 67%. And also we reviewed that more recently, some time ago, and again, the mortality was unchanged. We even highlighted the fact that in spite of the fact that the mortality of babies is reducing all the time, and this is a paper we wrote some time ago in England, the mortality from NEC was actually significantly increasing. In Canada, we have one advantage. We have the Canadian Neonatal Network, the CNN, which is not the television channel, where we actually can get data from all neonatal centers. And you could see here that over the years, the mortality from NEC has been unchanged, remains very high, both for surgical and medical NEC, and in black is the line of the mortality in the middle. Now, one of the other problems that can occur with NEC is that almost half of the population of the babies with NEC develop severe neurodevelopmental impairment. And this is, we did a systematic review and actually comparing no NEC with NEC in premature babies. If you have NEC, you're much worse neurodevelopmental outcome. And if you need surgery, even worse. So it is important, in my opinion, not to reach the stage to do surgery. And in surgery, what can we do? We are very primitive. We could create a stoma, we could resect and do an anastomosis, like in this case, which is my preference in babies with neck. And indeed, we did a trial that we just completed, and soon we will have the results of these, in which we compare the stoma versus direct anastomosis. Now, there have been some trials in the field of neck, and I was involved in one of these, and Larry Moss was involved in another one, and you could see that putting a drain, and our trial has been all over the world. And in spite of the fact that our trial was done in many countries, including Australia, including South Africa, many countries in Europe, the results of our trial compared to the one just done in the States was exactly the same, showing no great deal of difference between drain and laparotomy. Still, the disease is what kills these babies. But most importantly, both of these trials did not reach the power, did not reach the level that was required to demonstrate the significance. And so the big challenge that we have is still in front of us is this baby with an abdomen which is distended, discolored. We don't want to reach that stage. You can imagine how much nasty looking intestine is inside that abdomen. So we need a novel therapy. And this is what my lab has been doing for many years and trying to solve this issue. And we think we are at the brink of discovering something very important. So we want to avoid the progression of the disease from just inflammatory state to perforation, to multifocal disease, to panintestinal. And indeed, we demonstrated that 47% of these babies, in our experience, progress in the disease. Not everybody have like a very sudden onset of the disease. Some of them progress in the illness. And so we have been using in the lab, starting from epithelial cells to intestinal organoids, to experimentation in pap mice, to reach the point of clinical translation in humans. And we were fortunate recently to publish on Nature Communication our experimental new finding that remote ischemic conditioning can indeed counteract intestinal damage in babies with necrotizing enterocolitis. So I'm going to show you some data that came from that paper. This is from human samples, not from animals on this particular thing. And when we compare the most affected area with the area which is less affected in the middle, or with the area that is not affected significantly by neck, we found that there is mucosal hypoxia and microvascular impairment. So how can we actually have a better macrovascular assessment? And we thought about something which has been discovered for the heart, which is remote ischemic conditioning, where you inflate a cuff, produce an ischemia of the arm, and you reduce it, and that will reduce the ischemia in other organs, such as the heart. And we were the first to look at it in the infants with neck. And so what is remote ischemic conditioning? Is like protecting a distant organ by giving something which is very similar to take the blood pressure. So it can be applied in many countries, doesn't need to be done with very fancy equipment. So what is our hypothesis? Doing this remote conditioning may actually influence the gut and not let it progress to severe disease. So we did experimentation, which I'm going to show you some, in pap mice to see if a tourniquet, in this case we put it in the leg in one extremity, and protect the intestine itself. We give 4 cycles of 5 minutes each with a 5 minutes reperfusion time above the blood pressure. And we use a model in neck pap mice, which are the size of my finger. And we found this was like a magic bullet. All the effects of neck were actually ablated in the mucosal injury. Also, we look at inflammation of the intestine. The white bar is the one who received RIC, or remote ischemic conditioning, and you could see that the return to normal, which is the breastfed here, BF, compared to neck alone. The oxidative stress is also reduced significantly. And then we give this remote ischemic conditioning at 3 time points. At the beginning of the induction, during the early stages of neck, and then later when the effect on the intestine is already more severe. And we found that this magic bullet was in the first stages of the disease, not when you have already established a necrosis of the intestine, like stage 3. And if you look at the mortality and the Kaplan curve, you could see here that the blue and the green line are significantly better than the one with neck alone or neck with given too late. So when we look at later on what's happening to these pups, obviously none of them has been resuscitated. They are the size of my finger. They eventually all die, but you could see that by giving RIC, it does prolong their survival significantly, the blue and the green line. And so how does he do it? Because he gets better. We measure the perfusion of the intestine and gets better in the wall velocity in the intestine. And then we look at something more sophisticated, which is the 2-photon laser scanning microscope. And I'm going to show you some data on that. So I had a fellow from Japan who actually devised this technique, which was unknown at that time, in the mice during the disease. So they were alive. And you could see here that we scanned the villi from top to bottom. And you could see that the villi, we could see on the left panel, you could see the villi, and you could scan it down to the bases and see the flow in the arteries there. So then we look at what's happening in control with this technique, and you could see the flow is normal. We could see the little arteries, the little veins, and you could see what's happening in neck, rolling of platelets. The flow is very slow. And I show you what's happening during RIC, and you could immediately see the difference with the flow being fast and not much of rolling of these platelets and other particles in the blood. So RIC seems to be protecting the macrovascular environment of the bowel. And we found that we quantify that, and we quantify these effects, obviously, in the bowel itself. If you look at the red line, I think this is what's happening in neck. When you have it too late, when you are, in this case, was P9, the red line, you could see that the response to feeding, normally you should increase the blood supply to the gut. But when you are premature, which is the black line, that response to feeding, it does not happen. And therefore, if you put RIC, which is the blue one, you counteract this phenomenon and you get more blood coming to the intestine. And we quantify that by looking at the tip of the villi, where the yellow bit is the necrosis. And you could see that there is more yellow bit in the neck alone compared to the one with the RIC given. You could see the beauty of this technique. You could see the vessel, you could see the simple villi in them, and see that there is no necrosis if you give RIC. And then we quantify that, and we managed to demonstrate that this was a clear effect. And we also look at the distance between the vessels and the top of the villi. So basically, we could demonstrate that by giving this remote conditioning, we affect the healthy status of the villi in response to feeding. Now, hypoxia and necrosis at the tip of the villi doesn't happen, and this is what we have shown. How does it happen? The mechanism of action, I don't want to bother you with more experiments, is due to hydrogen sulfide and nitric oxide. But the beauty of this technique is that you don't need to give nitric oxide precursors. You don't need to give hydrogen sulfide. You just do this maneuver, and actually the body does it for you. In addition to that, we found also very interesting effects on the brain. So if we give RIC, not only we protect the intestine, but we could protect the brain. And remember what I told you at the beginning of my talk, that during NEC, if you actually survive, many babies have a neurodevelopmental outcome impairment. And in fact, we demonstrated that the neuroinflammation, the inflammation in the brain that happened during NEC is completely ablated by giving this remote ischemic conditioning. As you could see here, the green and the pink bar. And we look at the blood barrier integrity, and also this one is affected in the brain. And where in the brain? Most likely in the hippocampus, there is a significant effect, which is the area of the brain which is important for memory, or in the cortex, which is important for cognitive function. So we've done a lot of information, and we think what's happening in these babies is that the T cells migrate and create neuroinflammation because the tight junctions in the barrier are broken. But doing RIC, these tight junctions are maintained, and therefore there is no neuroinflammation in the brain. So we concluded this to show is a new paradigm. You give remote ischemic conditioning and you protect the intestine, most likely you protect also the brain. So improve survival, improve integrity of the intestine, improve inflammation, oxidative stress, and neuroinflammation. Is this a new treatment? Probably it is. So now to prove that it's efficacious, we have to move it into clinical. And this is where I'm really excited that for the first time in my life, actually, I came close to something that could go from basic science to actually to clinical evaluation. So what we want to do is to get a baby like this not progressing to a perforation and recovering. And so we've done the first in human safety study in babies. We did it in 3 steps. And we proved that each step we could give 4 minutes, and then we determine how many cycles of ischemia we could give with RIC, and then we did it in 2 consecutive days. And this is one of my fellow doing it. You could see the cuff in the arm. You could see the pulse oximeter in the end, because obviously we want to make sure that we don't damage the extremity. And these are the babies that we studied in this initial safety study. They were down to 800 grams with all stage of Bell. And we look at oxygen saturation, capillary refill time, pain score, urine output in the arm. And we found that there was no significant effect. So this was safe. In the first stage, in the second stage where we gave 4 minutes or 4 cycles, and then in the third stage, which we give it in 2 consecutive days, on day 1 and day 2, there was no significant difference in pain, oxygen saturation, capillary refill time, or urine output. So we concluded the return of oxygen saturation in the arm is all present within 4 seconds. There were no new skin lesions. The children could do still the grasping reflex, so they didn't lose that. There was no mortality due to RIC, and the pain score did not have any effect. So this maneuver, remote ischemic conditioning, can be safely done in babies. However, we have not proven yet if it's efficacious or not. And so if you look at the trial that we did some time ago on drain or laparotomy, still we didn't reach the power. So why we didn't reach the power? Because we need an intermediate step where you do the safety first, which is the phase 1 trial. Then we need to do another feasibility test, which is a phase 2 trial, and then eventually randomize, apologies for my voice, many children in the phase 3 trial. So now we created a new consortium, I'm very proud of that, of 12 centers in 6 countries and 2 continents. We obtained funding, in this case, I obtained it from the United States. We devised a protocol, we are publishing the protocol, and then getting the ethical approval. The countries which are involved in this feasibility study are from Canada, from the United States, and from Europe at the moment. And so these are the centers. And believe me, this is only a feasibility trial, but it took quite some time for me to create this consortium and get the funding to do it. So what we are going to do, we are going to give 4 cycles, 4 minutes of ischemia, 4 minutes of reperfusion in the arm, and then we measure the oxygen saturation in the ends, as you could see here in the baby. This will be done by a research fellow or a nurse, which is not the one who takes care of the baby normally. So during these 45 minutes, that person will look after the baby. And then after that, so the people looking at the baby for clinical matters are not aware if they receive or not receive RIC. So it will be a blind mask trial. And we want to aim for feasibility of recruiting or randomizing and masking the intervention. The inclusion criteria, and believe it or not, it is still debatable how to diagnose medical neck, but we make it with the agreement between neonatology and surgeons, and we randomize within 24 hours because we want to be early in the disease. Remember, I show you that if we do RIC too late, it may not be efficacious because there is already necrosis going on in the bowel. The primary outcome will be feasibility of including these patients in the study, randomizing and making a mask intervention. And this is in preparation of the bigger trial, which may actually involve countries like you, if you be interested, where we will look at survival of these babies at 1 month and 3 months without any other complication of neck, such as perforation, necrosis, or strictures. So finally, from this brief talk, and I hope that it didn't go beyond the time allocated to me, the mortality for neck remains very high and is the highest in our specialty. This is one of the reasons why I concentrated so much on this disease. Remote ischemic conditioning offers some hope, and it's probably a bridge between basic science and clinical, and may actually represent a future change in the treatment of neck. Finally, I would like to thank my lab, the people in my lab there, which have been very instrumental in doing all this experimentation and moving to this stage. And also I would like to thank all the other centers, 12 centers in 6 countries, which now they are keen to get started this feasibility multi-center trial. And finally, the people who gave me the money to do this, the CHR, and recently the Treasure Fund from the United States for the trial itself. I'm really, really excited. I think this may prove that we could, after many years, move into something that could have an effect on babies with neck. I hope that I've been clear in my presentation, and I finish my talk. I'm very happy to answer any question. Hello?
Hello.
Yeah.
Chairperson, Professor Bajpai.
So you need to unshare me, right? Yeah, thank you very much. I hope that my talk came okay, that you heard it.
Yes, yes, very much. We enjoyed it very much.
Yeah, indeed. It was an exciting talk and throwing new lights on preventive and management strategy. I just would like to ask you a question. Most of the studies, if you review or you see systematic reviews or original articles, they say all the next studies are underpowered. And mostly the reason, yeah. So what do you see about this study vis-a-vis other studies and where it is likely to stand?
Yeah, that's a very, very important question. And this is why we did a safety trial first to show that this is a new maneuver, is safe in a little baby, which could be, let's say, 700 grams or 800 grams. But instead of going is safe now. We know that. But instead of moving into an efficacy trial, which may fail because you are not ready, you have to do this intermediate step, which is what I'm planning to do now, which is a phase 2 feasibility trial to show that these centers will be able to randomize, will be able to collect patients. We will be able to mask the intervention. We could see what are the barriers to not making them randomizing. And once we've done all that, I know it's diluting and prolonging the thing for another couple of years, but then we will be ready to do a bigger trial. So that's why most of the randomized control trials in other specialties go from phase 1 to phase 2 to phase 3. In pediatric surgery, did never exist. So they jump from phase 1 to phase 3. And that's the reason for failing some of these trials.
Right.
I've been doing trials before. And indeed, one of the trials which was published on NEC is the trial which I coordinated. And I made the same mistake, as well as Dr. Larry Moss, which was doing that in, for drain versus no drain. Now I don't want to make that mistake. It takes a little bit longer, but we could achieve our goal.
Yeah. Thank you. Very interesting. The topic is open for questions. Coordinators, if there are any questions to Professor Piero, please.
Can I ask one while we look at the chat box? Have you seen patients with neck having a thrombosis? When I was at Gosh, I discussed with Edward Kiley. He had given a patient pepaverine because the thrombus to dislocate the thrombus and dilate the vessels. Do you think RIC will help in those patients as well? Because the thrombus leads to an end stage necrosis.
Yeah, that's a very good question, Shilpa. I think babies with neck no doubt have thrombosis. And this could be due to the disease itself. Now, I'm hoping that we'll catch a population before the form a thrombosis. Once you have a thrombosis, say, for instance, of the SMA, then you lose the vast majority of the gut, if not all of it. So we want to be a little bit before, and RIC could influence that because he influences the blood supply get into the intestine. But once you get the thrombosis, I don't think RIC is able to do that. Remember that remote ischemic conditioning was described first for issue related to myocardial infarction, where you have a blockage, and it seems to be protective for that. So the same mechanism would be for the intestine.
So would you advocate to start it right away, like the moment you make a diagnosis and the baby is not improving, but not having perforation?
No, I wouldn't recommend at all to do remote ischemic conditioning until we clarify that it's feasible to do this on a larger set of babies, and until we clarify that it's actually efficacious.
No, we will wait for your trial, definitely. But
yeah,
what is your
I wouldn't do it. I wouldn't do it.
What
Probably
is your
what we could do is after we've done the feasibility trial, then I could get back to you, Shilpa, and ask you, do you want to collaborate in our multi-center trial?
No, we will wait for your trial. What I mean to say, what would you include as your inclusion criteria for your trial?
Or the inclusion criteria is medical neck, which we have struggled in these 12 centers to diagnose. So we need to have 2 clinical parameters like baby's abdomen, distended, blood in the stools, characteristics of neck, plus at least intramural layer or portal vein gas. So we want to be definitely certain that baby did not have sepsis or other things. And so medical neck, similar, let's say, to stage 2 neck, but stage 2 neck, like the Bell classification, is not very, very clear.
So we'll take up some questions from the chat box.
Sure.
What do you think would be? Yeah,
Yes.
Professor.
May I?
Yeah, sure.
Thank you very much for your lecture. We enjoyed it very much. What is the timeline like? 24 hours cutoff? Isn't it too early? Like, is it possible to pick up patients within 24 hours for inclusion into your study?
Yeah, I think it is, but this is what we are trying to demonstrate. I think it is because once you make the diagnosis, the first time we have to do the RIC maneuver will be within 24 hours. So you have 24 hours from when you and the neonatologist you as a pediatric surgeon and the neonatologist establish, okay, this baby has got neck, but doesn't need an operation at the moment, basically. So I start giving the remote ischemic conditioning, and you have 24 hours to do that, which should be adequate. As a matter of fact, I was thinking to be even more restricted and do it within 12 hours, but that's a little bit impractical because you have to imagine the baby being transferred from another center or whatever. So 24 hours should be achievable.
Okay.
If you wait longer, you may wait and the baby may have progressed in the disease and you may not have a significant effect from RIC.
So it may turn into surgical neck rather than medical.
Exactly.
Yeah.
Exactly.
So there are some questions from chat box. Dr. Shilpa, you want to read this out?
So there's one question. How did you decide the time limit of 4 minutes?
Okay. The previous experimentation and the previous data in humans, in adults, has been done with 5 minutes occlusion, of the arm with a thing, and 5 minutes reperfusion. We went for 4 minutes because we wanted to be not too damaging the extremity. Remember that we are dealing also with babies. We went down to 800 grams. Our population should be from 700 grams onwards. So that's why we choose 4 minutes. All our experimental data is based on a similar ischemia time, which is 5 minutes and 5 minutes reperfusion. We do it over 2 days. But of course, you could have a longer period of time on ischemia, but then you don't know if you damage the artery in the arm or the leg.
True. Another question, what do you think would be the influence of confounding factors like birth weight, gestational age?
It's a very, very good point. There could be some confounding factor, no doubt. But we will stratify for that, but we'll stratify for that in the efficacy trial. Now we want to just to show that it's feasible to do it in that population. Our trial will aim just to see if it's feasible to randomize, feasible to mask RIC in a multi-center setting. When we do the efficacy trial, stratification will be very important.
Another question, how many days do you contemplate to give the treatment for?
2 consecutive days. So we do it on 1 day and then we do it 24 hours after.
Was there any mortality at all unrelated to the RIC in phase 1?
No, there was no mortality related to RIC. There were 2 babies who died much later for cardiac problems. But that's because there was a population at risk with stage 3 neck. But none of them died in the acute setting or died of RIC, as far as we could tell.
Dr. Sanjay Rai wants to know whether you would like to use this prophylactically to improve the perfusion to the brain and the gut.
Yeah, this is a very, very interesting question. Yes, we are planning to look into that. At the moment in my lab, we are looking at the value of giving prophylactically to see if such a thing could be done. And then we open a can of worms, because then if that is feasible, if that is possible, that has a prophylactic effect, then you may think, okay, I'm going to give it to babies premature at risk of developing neck. And then will be a different population of what I want to study, at least at the moment, but they may be the target one for this maneuver. I don't have the answer to that, but we are looking into that.
Thank you so much. There are lots of appreciations. Thank you for a wonderful talk.
Thank you.
Our chairman, Professor Mahajan, would like to present a memento to you online.
Thank you.
So Professor Piero, thank you very much for taking time and speaking to us. I know it is very early morning in Toronto.
No problem.
Yeah, so thank you very much. We appreciate very much. And this is a small memento on behalf of research section of Indian Association of Pediatric Surgeons. Hope we are able to see you in person in future. Thank you very much.
It's been my pleasure, my distinct pleasure. I would really love if you could send me a PDF or something of this
Definitely.
over the internet,
It's
and I will
yours.
print it at this end and put it in my office.
Definitely.
Sure.
India is close to my heart. I've been there a few times, as you know, and enjoy tremendously your country. So
Thank
I'm
you
really
so much.
hoping that we will come out of this terrible pandemic and one day we could meet each other again. But thank you
Definitely.
much for inviting me, for the opportunity to talk to you. Thanks.
Thank you very much.
Thank you. Thank you.
Bye-bye.
Bye. So, Ansh, we go on to next session now. I invite chairpersons for next session, Professor Parthopratim Gupta. He is consultant pediatric surgeon, Professor, Department of Pediatric Surgery, Institute of Child Health, Kolkata. He is past president of Indian Association of Pediatric Surgeons, specialty board members in pediatric surgery, NBE, and project director, Smile Train. Then our other chairperson is Dr. Juju Jacob Kurian. He is associate professor, Department of Pediatric Surgery, Christian Medical College, Vellore. So please start the session, and we welcome Dr. Augusto Jani. The chairperson will introduce Dr. Augusto.
Good evening. Thank you, Dr. Mahajan. I have the pleasure to introduce Dr. Augusto Jani, who is a neonatal and pediatric surgeon at the Hospital for Sick Children in Toronto, associate professor at the Department of Surgery, University of Toronto, and scientist in the Development and Stem Cell Biology program at the Peter Gilgun Center for Research and Learning, Sick Kids, Toronto, Canada. He received his MD and completed his pediatric surgical training at Sapienza University of Rome, Italy. He earned his PhD from University College of London. The list goes on, but he went to Canada after having his trainings in Great Ormond Street and King's College, London. And with a fellowship in neonatal surgery in 2016, he joined the Faculty of Division of General and Thoracic Surgery at Sick Kids, and Dr. Zani's clinical interests are congenital anomalies and minimally invasive surgery. It's such a long list that I will request my co-chairperson, Dr. Jacob Kurian, to continue his introduction. Thank you so much.
Thank you, sir, for that. So, Dr. Zani's research focuses are basically on two major neonatal conditions which has the highest mortality. That is congenital diaphragmatic hernia and necrotizing enterocolitis. Dr. Zani has founded the Respire program, and his lab investigates strategies to regenerate fetal lungs using a stem cell-based therapy for pulmonary hyperplasia secondary to CDH. Dr. Zani is also investigating causes of brain damage and neurodevelopmental delay that occurs in NEC survivors and potential strategies that could be associated or employed to prevent NEC-associated morbidity. Dr. Zani's research is supported by international funding, including the Canadian Institute of Health Researches. He is a fellow of the American College of Surgeons as a member of various August bodies. He is also serving as the executive board of IAPS since 2016 and presently as the honorary secretary. He has authored more than 150 peer-reviewed articles and 15 book chapters. He has co-edited the book Pearls and Tricks in Pediatric Surgery, and he is also on the board of various pediatric surgery and pediatric journals, including Journal of Pediatric Surgery, Pediatric Surgery International, the European Journal of Pediatric Surgery, the European Journal of Pediatric Surgery Case Reports, and Frontiers in Pediatrics. It is our pleasure, Dr. Zani, to invite you to this August meeting, and we look forward to hearing from you.
Thank you very much for the kind invitation, the kind introduction. And thank you, Professor Mahajan and Professor Sharma for inviting me to this session. I have to say I can only echo what Dr. Piero just mentioned. I've been to India before. It's an amazing country. My wife and I love coming to India and love Indian food a lot. So we eat at least once a week Indian food. So it's, but it's never as good as it is in India, we figured out. I will start sharing my presentation, and I hope you can see it. Okay, can you see my slides?
Yes, we can.
Good. Okay, so we change gear and we talk now about congenital diaphragmatic hernia, which, as we just mentioned, is one of the pediatric surgical conditions with the highest mortality. About 60 to 80% survival rate is the reported rate globally for babies with congenital diaphragmatic hernia, but possibly this is even higher due to hidden mortality. And frustratingly, these figures have not changed over the last 2-3 decades. There's also a report this year from a large collaborative group on The Lancet looking at low and middle income countries where actually the survival can be even as low as less than 1%. Costs for the families and the society are tremendous. And in fact, the estimated cost for congenital diaphragmatic hernia annually in the United States is considered to be the highest. And you can see the figure there is $390 million. So it's quite significant. 60% of the survivors still suffer from long-term morbidity. And again, what is frustrating is that there is still low awareness, as for many other congenital anomalies, and therefore also low fundings for research in congenital diaphragmatic hernia. If you look at the graph, these are Canadian stats. Everyone in the street would know, heard about cystic fibrosis and spina bifida. Yet new cases per year almost double in congenital diaphragmatic hernia. But unfortunately, as I mentioned, there is low awareness. Now, we're all pediatric surgeons, so I don't need to convince you that the real problem for these babies isn't the lung, is the pulmonary hypoplasia, the degree of lung underdevelopment that they have, and not really the diaphragm. We can repair the diaphragm with very refined techniques, minimally invasive thoracoscopy, but these babies still have small lungs that are the major contributor to their morbidity and mortality. I show you this, we just have these graphs that have not been published, they're under review in one of the Nature journals, and is a scheme where you can see how, in fact, there's not just underdeveloped lungs, so pulmonary hypoplasia, but also ventricular hypoplasia. The heart is also a lot of times compromised, and there's of course increased pulmonary vascular resistance that obviously results from vascular remodeling. So it's a complex disease where one single treatment is not going to fix everything. And this is, as I just mentioned, the pulmonary parenchyma is affected, as well as the pulmonary vasculature, as well as the heart. But when we look at pulmonary hypoplasia, we need to look at it not just as the reduced number of bronchial divisions and defective alveolarization. The lung, in fact, is immature with undifferentiated epithelium and mesenchyme. And as I mentioned, also the vascular development is abnormal, with vascular remodeling, fewer vessels that have a thickened wall, and contribute to the pulmonary hypertension that these babies develop after birth. But there's something that we are all in agreement regarding the management of congenital diaphragmatic hernia, although there's so many differences in style and approach to this condition. The condition can be diagnosed as early as the anatomy scan, and so the ideal timing for intervening in these babies' lungs could be antenatal. And so we heard incredible research coming from Dr. Harrison in the 80s and the 90s, and then followed up by Professor Jan de Prest and colleagues in Europe in the last couple of decades. But unfortunately, none of these fetal treatments that have been tested so far are successful in fully rescuing normal lung development. And I can tell you that the first person that says this is Jan de Prest, that is one of my collaborators. We have been working for quite some years now, over 15 years, with amniotic fluid. And we have some very exciting and promising data that I'm going to share with you today, as we think that the solution for these babies' lungs could be found in the amniotic fluid. About 15 years ago, the group of Anthony Atala and very young Paolo De Coppia at that time described this population of stem cells that reside in the amniotic fluid that have potential for therapy. It was in fact exactly, I think, January 2007, and so almost 15 years ago. This population of cells is broadly multipotent, so meaning that it's able to differentiate into all three germ layers. It's easy to collect, has low ethical impact, and do not form tumors. And so over the last 15 years, there's been a wealth of literature and research confirming that amniotic fluid stem cells hold the regenerative potential towards different organs and systems. And specifically also in the lung, we know that the cells can integrate and differentiate into epithelial lung lineages. They can inhibit the progression of bleomycin-induced pulmonary fibrosis. They can repair damaged alveolar epithelial cells, but also they can actually make these hypoplastic lungs in a model of CDH grow bigger, as reported by Professor Tovar in collaboration with Dr. Pierre and Dr. De Coppia. However, it's our experience of many researchers working with stem cells of different nature, not just the amniotic ones, that whenever you administer the cells, you see a beneficial effect, but there's very low degree of engraftment in the tissue. And so for many years, we've been wondering about the paracrine effect, about this paracrine mechanism that is exerted by the cells, but with very low engraftment. And we've been trying to understand what paracrine factors could be responsible for that effect. And this is where the story of my lab starts about 6 years ago, where we started working with extracellular vesicles. Now, I don't know how many of you are familiar with extracellular vesicles, or exosomes, they're also called. It's something that is pretty interesting, and I will give you a little bit of a snapshot, of a summary of what we're talking about. Now, extracellular vesicles are nanoparticles secreted by any type of cell throughout the body, in animals and in plants. They are considered as an intercellular form of communication, whereby there's a sending cell that releases a message that targets a cell that can be even far. They're called with in different ways. As I said, you can hear oncosomes in the field of oncology. It seems to be very much responsible also of metastasis, exosomes, microvesicles, but the International Society for Extracellular Vesicles really settled on this position paper, where really they endorse the use of extracellular vesicles, and so this is how we're going to talk about them. We know that when they are small, just for easier reference, they are called exosomes. Where they have an intermediate size, they can be called microvesicles, and when they're bigger, they can be called apoptotic bodies. They are as small as viruses, especially the smaller ones, the exosomes. And if you see the red dot that is difficult to see in that big black circle, you can see that basically 1 million of extracellular vesicles would happily sit on the tip of a pencil. So in this video, you can see what a cell usually does. They package messages every second of our life, and they package them into these extracellular vesicles that are lipid membrane bound. Then the messages are released, and with a not very well understood mechanism of homing, they are taken up by a recipient cell. And here you can see there's a parent cell, and there's extracellular vesicles that are released, and then they can be taken up as with different modalities: fusion, endocytosis, ligand receptor interaction. But what's the role of the EVs in the cell in general? Well, but for many, many years, people thought that this was a way for the cell to dispose of their garbage. And in fact, if you go back, you can see that the first report was almost 55 years ago. But for many years, the people really didn't give attention to these extracellular vesicles. In the 80s, they observed that some could be very, very small, and they called them exosomes. But it was only about 15 years ago that EVs were considered in a different way, because in fact, this group from Sweden recognized that they can transfer functional nucleic acids between cells. And this is where really the world of extracellular vesicles started, and research in this field. So I'd like you to imagine EVs as cargoes or bags can carry cargoes and messages, not as garbage bags. And in the field of regenerative medicine, it's very well established now that they are considered the key mediators of stem cell paracrine signaling. Their effects are similar, sometimes greater, to those of parent cells, and their cargo has more regenerative potential than single molecule therapy. So we started looking into extracellular vesicles derived from amniotic fluid stem cells with a back of more than 10 years of research using those cells, those stem cells. And we reported this a few years ago in scientific reports. And Lina Antunians is the first author and lab manager in my lab. And we compared the different isolation techniques. These are our extracellular vesicles. As you can see, they're round and spherical. We use mainly ultracentrifugation as a technique to isolate the vesicles. And then according to the guidelines from the International Society for Extracellular Vesicles, we characterize them by size using nanoparticle tracking analysis, shape, we're using transmission electron microscopy, and expression of canonical EV markers using Western blot. And this year, in the spring, we were fortunate to publish our story, almost 5 years of research in my lab with many collaborators, as you can see in these slides, on Science Translational Medicine, whereby we showed that pulmonary hypoplasia can be rescued by the administration of amniotic fluid stem cell-derived extracellular vesicles in rodent, and you will see also in some human platforms. Vincenzo Catania was a research fellow in my lab and is now a pediatric surgeon in Bologna, Italy. He started established the famous model of CDH, which is by administration of nitrophen to dams at embryonic day 9.5. This is a model that's been around for more than 40 years. It results in pulmonary hypoplasia in 100% of the litter. Only 50% of the pups would develop CDH. And nitrophen is a herbicide that blocks the retinoic acid signaling pathway, which seems to be, in fact, also abnormal in human lungs of babies born with CDH. And so we had the control pups that whose mothers received only olive oil, and nitrophen pups whose mothers received, in fact, nitrophen. And then some others that were exposed to nitrophen but received the amniotic fluid stem cell conditioned medium, or the extracellular vesicles, or the EV depleted condition medium, or received another source of extracellular vesicles, which is from mesenchymal stem cells or mesenchymal stromal cells. And it was only when we gave the amniotic fluid stem cell extracellular vesicles that we could reproduce the same degree of branching morphogenesis that was shown in normal PAPs. As you can see here, it was measured with terminal bud count and lung surface area. We looked also into maturation of the lungs, looking at different key markers, such as surfactant, which is known to be decreased in nitrogen exposed lungs, and it was actually rescued. The expression was rescued in nitrogen exposed lungs that received extracellular vesicles. We also established for the first time, to the best of our knowledge, fetal lung organoids, where we looked at those derived from nitrogen exposed lungs that had decreased proliferation and decreased expression of, again, differentiation markers. And proliferation and differentiation markers were significantly increased and in some instances rescued by the administration of the amniotic fluid stem cell extracellular vesicles. We then decided to study alveolization. And with our collaborators in Brazil, in Sao Paulo, with Professors Braga and Dr. Figueira, who's now actually a postdoctoral fellow in my lab, we used their model of fetal rabbit congenital diaphragmatic hernia. And we had control pups that did not have a hernia, CDH pups in which hernia was created surgically in the fetuses, CDH plus tracheal occlusion that would mimic fetal, and CDH plus tracheal occlusion plus administration of extracellular vesicles. And as you can see here, very skillfully administered by Professor Braga in the trachea of the pups, and then a very rudimentary form of fetal, no balloon occlusion, but tracheal occlusion that was then performed. And interestingly, only when we gave the extracellular vesicles, we could rescue the maturation that we had, the maturation levels that we had found in control lungs. We knew that tracheal occlusion increased the size of the lung. And so you will see that the tracheal count here was gone back to normal levels with tracheal occlusion alone. And this is what we know from babies now that have undergone fetal in with the trials published by Jan de Prest and collaborators in New England Journal of Medicine in May of this year. And we in Toronto are also collaborators on these trials. But it was only when we added the extracellular vesicles that we actually could rescue, for instance, the expression of lipofibroblasts that are important for surfactant production. We also decided to do some experiments towards clinical translation and administer EVs to models of in vitro models of human lung injury using both A549 cells that are cells from adult human carcinoma, and so it's a very well established cell line, but also using human fetal pulmonary alveolar epithelial cells. And when we gave the extracellular vesicles, we proved, as you can see at the bottom, that they were entering the cells, but also they were rescuing homeostasis levels, proliferation, and viability. So then we wondered, what's in the cargo that is so magical that can make all this happen in these models? Well, we know that extracellular vesicles in general are bound by a double lipid membrane. They contain lipids like sphingomyelin and cholesterol. They contain proteins, but also they contain genetic material. And this is what makes them quite interesting. So we first did the proteomics analysis, looking into the proteins that were present in the EV cargo. It didn't find really no proteins that could be responsible of these effects on promoting lung development. So then we turned to small RNA sequencing. And we sequenced the cargo of the amniotic fluid stem cell extracellular vesicles, as well as of mesenchymal stem cell extracellular vesicles, as these would be a great comparator because we hadn't found the same beneficial effects as when we were giving the FSCEVs. And interestingly, we found a number of small RNA species, but specifically microRNAs were the ones that were mostly differentially expressed by the FSCEVs. And we noticed that there were a number of EVs, such as the MIR1792 cluster, that are crucial for normal fetal lung development. And to tell you just a very brief, very little about microRNAs. MicroRNAs are non-coding RNAs that bind to mRNAs and silence the translation to proteins. So they downregulate some biological pathways. And so we consider that in the EVs there are a lot of microRNAs, and in the lung epithelial cells that have been targeted by our EVs, some mRNAs were downregulated. And these are mRNAs that in fact had to be downregulated to promote normal lung development. And so with some, collaborators who are experts in bioinformatics, we came up with this interaction network between the FSCEV miRNAs, microRNAs that are contained in the FSCEV cargo, and the downregulated genes that were found in nitrogen-injured lung epithelial cells treated with the FSCEVs. And again, you can see here in the middle that some of these miRNAs are from the 1792 family. Why are microRNAs important? Well, not just for rats. You can see that there's been quite important papers in the last very few years, in the last 4 or 5 years, showing that babies with congenital diaphragmatic hernia, for instance, undergoing fetal, have in their tracheal fluid a microRNA signature that seems to be different from that controls. So they can also be considered potentially as biomarkers of the severity of this condition. And this has been shown in human babies as well as in the rodent models and in even avian models of CDH. So our new paradigm of therapy would be by giving the extracellular vesicles that are derived from amniotic fluid stem cells. The vesicles would target lung cells of different nature, bind to the mRNAs to then eventually promote fetal lung growth and maturation. Now, we did all these experiments at the pseudoglandular stage of lung development, because previous research groups, as I said, the one from Madrid, Professor Tovar, as well as one from Ann Arbor, Michigan, had been doing these experiments at around E14.5. But we thought, you know what, we should really look at something that is more translationally relevant. And that's why we then did experiments also in the canalicular and saccular stages to try and confirm whether EVs could be also helpful here. And we are just waiting to hear from Stem Cell Translational Medicine. The paper is currently under review. So Kaskalaju's postdoc in my lab looked at several pathways that are known to be dysregulated in the nitrogen model of congenital diaphragmatic hernia. I have to say, who has been a giant in this field of research is Professor Prempuri, who I consider one of my mentors. I worked with him many, many years ago. And his group has published for over many years beautiful research looking and pinpointing at the different pathways that are dysregulated in the nitrogen model. And we could see that some of these pathways, if not most of them, especially at the saccular stages of lung development, can be in fact rescued by the administration of extracellular vesicles. And when we looked at the cells specifically, well, we could see that, for instance, alveolar type 1 and 2 cells that are crucial for lung gas exchanges have decreased expression with nitrofen, and the expression is rescued by the administration of EVs, as measured here at gene and protein level. The same we found at the saccular stage. And it was not just with 81 and 82 cells, but also with the pulmonary neuroendocrine cells that are known to be upregulated, have upregulated expression in the nitrofen model, as reported by several groups around the globe, as well as club cells and ciliated cells. Their expression levels were rescued by the administration of EVs back to control levels. And again, this is all been looked at and measured, not just with immunofluorescence, but also with gene and protein levels. And then we looked also at the mesenchyme. Now, the fibroblasts in the lung are a very interesting population. They do a lot of things, but still we don't know too much. And especially in congenital diaphragmatic hernia, we know very little. But what we know is that lipofibroblast expression is usually decreased, and lipofibroblasts, as I mentioned before, are crucial for surfactant production. Well, I can tell you that the levels were decreased in nitrogen-exposed lungs, and they were rescued by the administration of EVs at both canalicular and saccular stages. And so this is a graphical abstract where you can see that in the nitrofen model, both proximal and distal airways, we have abnormal expression levels of various epithelial and mesenchymal cells that is then rescued by the administration of amniotic fluid stem cell extracellular vesicles. But what are the cellular processes that are influenced by FEV administration? Well, there's many processes, and one that is particularly interesting because it's very much involved with organogenesis, embryogenesis, and organogenesis is autophagy. And we turned our attention to this process because, in fact, one of our collaborators, Dr. Martin Post here in Toronto, just reported a couple of years ago that autophagy is crucial for lung branching morphogenesis in a mouse model. So again, my postdoc, Dr. Kalaj, looked into whether this process could be also dysregulated in fetal apoplastic lungs in the nitrofen model. And we confirmed the same at different time points. We also confirmed a decrease in the density of autophagosomes, that are the functional units for autophagy, but administration of the extracellular vesicles rescued the autophagy markers, Becklin 1, ATG5, sequestrosome 1, back to normal levels, as well as the density of the autophagosomes. We confirmed that the activation was localized in the fetal lung epithelium, where you see in nitrogen-exposed lungs, there is a decreased expression of Becklin 1, which is rescued by the EVs. And this is just the cascade of events where you see the red arrows shown in the CDH model, where the expression of the autophagy markers is dysregulated and is rescued by the administration of the EVs. But is this clinically relevant for babies with human CDH? Well, I can tell you that yes, it is. We are now able to collect lung specimens from babies, from healthy termination of human babies between week 15 and 20 of gestation. We grow them as explants, and then we block the branching morphogenesis and induce pulmonary hypoplasia by giving NSC 23766, which is a RAC1 inhibitor. RAC1 is an important molecule for branching morphogenesis, and indeed its expression is reduced in the lungs of fetuses, human fetuses with CDH. And as you can see here, the levels of the autophagy markers and the autophagosome density were lower in NSC-treated lungs and were rescued by the administration of EVs. as I told you at the very beginning of this talk, as we all know, pulmonary hyperplasia doesn't end with the epithelium and mesenchymal disarrangements. It's actually involving vasculature. And we know that vascular remodeling is unfortunately responsible of the pulmonary hypertension that these babies suffer from after birth. So we also looked at the effects of extracellular vesicles on the vascular remodeling. And for this, we injected the vesicles in an in vivo model of CDH. And we looked at, first of all, whether the EVs were reaching the lungs. We did it towards the end of gestation, because in fact, E18.5 is when these pups start having breathing movements and swallowing the amniotic fluid. We did some dose curve experiments and confirmed as you can see here, the presence of the EVs in the lungs. The EVs go everywhere, as you can see. This is not the way we envision in the future how to translate this modality, this promising therapy, but it's just very useful for our experiments. And so I'll give you just a brief overview of what we're seeing. Well, we know that in the nitrofen model, there's decreased number of pulmonary vessels and increased wall thickness. And you can see here, we rescue also vascular remodeling, or vascular development by administering amniotic fluid stem cell extracellular vesicles. So this is, a brief overview on the vasculature. There's more that is that we have in the lab, and some exciting experiments. And as I say at the bottom of this slide, stay tuned, because the effects on vascular remodeling are very promising. But overall, we are quite confident to say that extracellular vesicles derived from amniotic fluid stem cells are able to rescue branchial morphogenesis and promote lung epithelial and mesenchymal maturation in these experimental models of rodent pulmonary hyperplasia, as well as human lung injury. They do these at translational relevant time points, and seem to affect various biological processes, such as autophagy, which we know is a critical one for normal organogenesis. Their effects seems to be exerted primarily by the release of their RNA cargo. And we have identified some microRNAs that seem to be relevant to regulate lung maturation processes. So we believe that administration of amniotic fluid stem cell extracellular vesicles represents a promising cell-free therapy for babies with hypoplastic lungs secondary to CDH. So I want to thank all the people that have done the hard work in the lab for me today to be sitting in front of you and giving you this lecture. My collaborators that go well beyond the borders of Canada, and the generous fundings received at the national level by the American Thoracic Society and by other agencies, and you for your attention. Thank you.
Professor Partha, please unmute.
Thank you, Dr. Augusto Zanni, for real insight, and that too very exciting, in preventing the pulmonary hypoplasia and CDH, which all of us pediatric surgeons all over the world dread to see. I hope that you will reach your goal and formulate the standard practice so that worldwide lots of sick newborns with CDH can be saved. I would request my co-chairperson, Dr. Korean, to put his comments.
Dr. Zanni, excellent presentation on a topic that is actually not very familiar with the surgeons per se. My question to you is, I have one small doubt about this thing. How do you actually, this administration is actually into the amniotic fluid, isn't it? how do you actually make sure that the vasculature is actually getting it actually reaches the vasculature properly? Is there any method to actually, direct it towards the vasculature properly?
This is a fantastic question and absolutely right on target. Yes, well, I can tell you that we've done now single cell RNA sequencing. We sequenced the lungs of these these pups that have received the intraamniotic injection, and we see also differences in the cell populations, endothelial cells, pericytes that are important for the support of normal vascular development. But again, I not saying that this is the way we should go ahead for babies with CDH. I think that babies with CDH would benefit not from an intraamniotic injection. One of the questions that I often receive is how to translate this clinically. And I can tell you that I think the easiest way would be to combine this therapy at this stage in 2021, almost 2022, with fetal. And so that's why I mentioned Professor Jan de Prest. We're collaborating on this. And the idea would be to use large animals. This is what we're planning to do next year, to look into the safety and feasibility before going into a trial for babies. But if you really ask me what I envisage for the future, my dream would be to have vesicles that can target the fetal lungs. We know that the vesicles cross the placenta barrier because they're very, very tiny. So I would love to have vesicles that can be given as a drug to mothers that are pregnant and know that they have babies, they're carrying babies with congenital diaphragmatic hernia, and they can receive just an intravenous administration of the vesicles, and the vesicles would travel and target the fetal lung. This is very futuristic, and there's a few years down the line, obviously, before this can happen. But I think this is how it should happen in the future.
Very interesting concept. The topic is open for others.
Can I ask a question?
Yeah, sure, please.
Please.
So I've been reading about exosomes since last 4-5 years. We're doing one project in liver regeneration as well. So on Monday, I have a protocol to present for a clinical case of kidney failure. What I feel, and I want to discuss with you, I think exosomes alone are not enough. You need a carrier as well, because exosomes are like cytokines, growth hormones. They'll go and target a cell, but if the cell is not there, then the battle is lost. So if we substitute, I've added mesenchymal stem cells as well. So some cells to substitute the regeneration. Like in kidney, we know there are kidney stem cells, but in lungs, I'm not sure if there are any lung stem cells that the exosomes act on.
That's fantastic. First of all, congratulations, because to be already translating this into clinical, this is awesome. I agree with you. I think it depends very much on the condition. I think what we are, so for instance, if we were talking about congenital diaphragmatic hernia, we know that all the cells are there. They're just stunned. They're just stopped in development. The development is arrested. And so we need something that gives them a kick, I would say, and the kick starts the normal processes. And that's why I don't think we need to give anything on top of the exosomes for what we've seen so far. But in other conditions, absolutely, I totally agree with you. It should be a combination of potentially stem cells, extracellular vesicles, and whatever else is needed. So it's congratulations on your research.
Thank you.
So I don't see any questions excepting in the chat box, which I think, no, there isn't any other question for Dr. Jani. And it's over to you, Dr. Mahajan and Shilpa.
Thank you, Dr. Augusto Jani, for your wonderful lecture. That is really high-end research. And we hope that someday it is translated into clinical application for our small babies. So I congratulate you on your excellent work, and thank you very much for joining us and enlightening us on this relatively new topic. So on behalf of research section of Indian Association of Pediatric Surgery, a small token of appreciation. You've been very kind, and as I said earlier, it's very early in Toronto, and thank you very much for joining us.
Thank you very much for the opportunity to share my research, and thanks for your kind words. And as Dr. Piero said, I love to have that certificate sent over, and hopefully see you in person at conferences, and why not in India. Thank you.
Can I ask you one informal question?
Absolutely.
Do you have any confusion in the department when it's Augusto or Augustino with your name?
All the time. All the time. All the time. I have to say, sometimes it's now it's getting better, but it was we had a patient that they kept paging the wrong surgeon, and the patient was ready on the table. These kind of things that sometimes happen. In Italy, there wouldn't be a difference because they're both very uncommon names.
Okay.
But it happens often. Yes.
What a coincidence. I realized because you started calling him Dr. Piero instead of Agostino.
Agostino, yeah. Well,
Thank you.
Okay.
Thank you very much.
Bye. Thank you.
Bye. So with that, we come to the last lecture of the day, and it's my pleasure to see Professor Eric Skarsgård from Vancouver, and it's very early in Vancouver. So chairperson for the session are Professor Sandesh Parelkar, Head Department of Pediatric Surgery, State GS Medical College and KEM Hospital, Mumbai. And other chairperson is Dr. Sanjay Rao, Consultant Pediatric Surgeon, Narayana Health, Bangalore. And he is our Secretary of Indian Association of Pediatric Surgery as well. So I will request both chairpersons to formally introduce Professor Eric Skarsgård. And since it was very early, we have his recorded lecture. So we will play a recorded lecture, although Professor Eric is with us in person also. So chairpersons, please.
Greetings and good evening and good morning to the visitors from Canada. Welcome to this conference. It's a pleasure to be here, and it's been an exceedingly interesting evening session over the last few hours. I will start with the honors and then hand over to Dr. Sandesh. Dr. Eric Skarsgård is the Surgeon-in-Chief at the British Columbia Children's Hospital and Professor of Surgery at the University of British Columbia, Vancouver, Canada. Dr. Sandesh, can I ask you to please go ahead and complete the introduction, and then you can start the presentation. Is Sandesh online?
Unmute. Yes, Dr. Sandesh.
Am I audible?
Yes,
yes. Yeah, Sandesh, you are. Yeah.
Yeah. So good morning, Professor Skarsgård. As Sanjay said, he is the Surgeon-in-Chief at British Columbia Children's Hospital and Professor of Surgery at the University of British Columbia, Vancouver, Canada. He is the past chair of the Advisory Board for the Institute of Human Development, Child and Youth Health at Canadian Institutes of Health Research, past president of the Canadian Association of Pediatric Surgeons, and former governor of the American Pediatric Surgical Association. He is the founding principal investigator of the CIHR-funded Canadian Pediatric Surgery Network, which targets best practices for gastroschisis and CDH. I'm looking forward to your lecture on the clinical registry after a session of 2 lectures on the most recent research on neck and CDH. Over to you, Professor Eric.
So we will play a recorded lecture of Professor Eric.
Yes, sure.
So is the screen visible?
Yes, it is visible.
Yes, it is. It is.
Yeah. Okay, start playing.
Dear pediatric surgical friends of India, my name is Eric Skarsgård, and I'm a pediatric surgeon in Vancouver, British Columbia, Canada. It is a great honor for me to join you virtually for this meeting of the research section of the Indian Association of Pediatric Surgeons, and I would like to especially thank Professor Mahajan for the privilege of this invitation. Today, I'm going to speak to you about the utility of observational clinical data organized into patient registries and how systematic analysis of this data in the era of evidence-based medicine can create an evidence platform for driving and sustaining care improvements in pediatric surgery. I have no disclosures. Foundational to an understanding of how observational data from a group of patients organized into a registry can support improvements in care and outcomes is the concept of the virtuous cycle of knowledge or learning, shown here as a continuous circular process surrounding and supporting, in our case, the care of children undergoing surgical treatment. In this talk, I'm going to focus on 3 specific registry functions that support this virtuous cycle. The first is knowledge creation, or the identification of best practice. The second is the mobilization of this knowledge, leading to practice standardization, as in the creation of practice guidelines. And the third is a surveillance function, also known as benchmarking, which provides key performance indicators to individual hospitals within the registry network. Providing individual hospitals with benchmarked data reveals where improvement opportunities lie and forms the basis for quality improvement. In discussing evidence, I'm also going to discuss pyramids, which can classify evidence in terms of its strength and validity, but also allow the organization of clinical measurements into actionable units. Most of us are familiar with this evidence pyramid, which ranks evidence according to quality with randomized controlled trials and meta-analyses of randomized controlled trials at the top. As we move down the pyramid, we get to the levels where the majority of evidence in pediatric surgery exists, specifically cohort studies, case-controlled studies, and case series. This is not to say that pediatric surgeons haven't been at the forefront of evidence creation through randomized controlled trials, and I am not for a moment suggesting that we should stop doing randomized controlled trials, but I am suggesting that we need alternatives for a number of the complex conditions that we treat, where isolating and evaluating a single intervention at a time using randomized controlled trial methods is difficult, if not impossible. It is worth acknowledging some of the other specific barriers to RCTs in pediatric surgery. With few exceptions, our clinical departments have not been designed with RCTs in mind. However, that is changing, and increasingly pediatric surgeons with dedicated research personnel are facilitating RCTs at the point of care. Still, there are barriers that are unique to surgery. Sham or placebo surgery is unethical. If one of 2 different surgical interventions is proposed, how do we guarantee standardization and equivalency and equipoise amongst surgeons who are performing these operations? Clearly, there are also patient factors where patients may be unwilling to participate due to loss of autonomy, particularly when the risk profiles of the 2 interventions may be quite different. Increasingly, the concern of lack of generalizability of findings from RCTs beyond the inflexible trial conditions is of major concern. Given the time, effort, and expense associated with completing RCTs, a strong case can be made in favor of a more pragmatic approach with fewer patient exclusions and greater flexibility in how patients are treated, so that the results are more likely to have real-world application. This cartoon captures some of the challenges associated with RCTs as the sole source of high-quality evidence to inform best practice. This New England Journal of Medicine editorial appeared recently, and it suggests that we need to look beyond randomized controlled trials for evidence, particularly for the uncommon conditions that pediatric surgeons treat. The editors specifically comment that rare disease registries are especially useful for the collection of standardized clinical information on individual treatment and clinical status, which can enhance our understanding of best practice and improve the health of affected patients. This brings us to another pyramid, which is known simply as the knowledge pyramid. The concept here is that information is defined in terms of data. knowledge in terms of information, and wisdom in terms of knowledge. This also implies that there is a hierarchical structure that translates clinical measurements made during patient care into wisdom as to how best to treat patients. This is the foundational concept behind patient registries as knowledge sources capable of improving patient care. So, what are patient registries? Patient registries are defined as a collection of standardized information about a group of patients to be used for a predetermined scientific, clinical, or policy purpose. There is an expectation of maximizing data through multi-institutional data sharing, with an option to link biospecimens as a means of supporting basic science research. Increasingly and appropriately, there is a demand and expectation for patient engagement, giving patients a voice in what data should be collected and for what purposes it should be used. Patient registry data has essential elements. These include a data dictionary, which provides explicit definitions which ensure comparability of variables between sites. It's important that data variables, and particularly outcomes, be developed by the clinical end user, and it is the reason that administrative databases cannot substitute for registries in answering relevant clinical questions. Data collection techniques vary between registries, ranging from active to passive data abstraction, voluntary submission of templated data sheets, and questionnaires sent to physicians or patients or both at various stages of treatment and follow-up. Patient privacy must be protected. This is especially true for rare disease registries, where the risks of patient identity disclosure are higher, but are likely offset by the potential for benefit to the entire cohort through maximized participation and data use. Finally, registry data must be used for its predetermined purpose of creating and disseminating knowledge and wisdom. Patient registries are frequently part of another structure called the Clinical Research Network. While the registry's data is a central component of the network, registry stakeholders set priorities for how the data is used. These include clinicians, researchers, hospital administrators, and health service policymakers. Patient and families must also be engaged stakeholders. Clinical research networks have an essential infrastructure which supports functions of the network, including data management, project management, and access to the necessary expertise to conduct methodologically rigorous research projects and then disseminate the results. Here we see the countries where established disease-specific registries relevant to pediatric surgery exist. As can be seen, the conditions studied tend to be rare birth defects or acquired neonatal conditions like necrotizing enterocolitis. Here is an incomplete list of registries by their host country. Congenital diaphragmatic hernia and biliary atresia are the focus of several registries. Most of the national registries, including the Japanese and Canadian registries, are population-based, meaning they seek to capture every patient born with that condition. This is important, as these registries can be data sources for incidence and epidemiology studies. Sweden, which has a universal access healthcare system, is unique in its registry capability. Virtually every Swedish citizen is part of an integrated national health database that collects standardized data on demographics, provider characteristics, detailed summaries of treatment, including drug prescriptions, and long-term outcomes, both clinical and patient-reported. The Congenital Diaphragmatic Hernia Study Group Registry collects pre- and postnatal data on CDH patients from 79 centers in 15 countries. I've also noted the National Surgical Quality Improvement, or NSQIP, registry, which is hosted by the American College of Surgeons, which, as most know, is a surgical outcomes registry that samples cases and provides detailed 30-day postoperative outcomes that focus on complications as a target for performance improvement. The Japanese biliary atresia registry makes use of 3 questionnaires which are delivered to patients to be completed by their providers that focus on their initial presentation and treatment, short-term follow-up, and a specific questionnaire for those who undergo transplantation. Here we see some of the differences between the various registries according to study population, data collection technique, and aggregate numbers of patients in each registry. One of the real challenges of birth defect registries is maintaining follow-up beyond the birth hospitalization. Increasingly, follow-up for complex birth defects like CDH occurs in hospital-based multidisciplinary clinics, which makes ongoing data collection more feasible. I've described the success of countries like Sweden, where health service data integration allows detailed virtual follow-up, which is achieved by measuring and analyzing all the encounters that a patient has with the health system, including primary and specialty care from billings data, drug prescriptions from pharmacy databases, and even developmental outcomes from school performance databases. I'll now describe a little bit about our Canadian Pediatric Surgery Registry and Network, which we refer to as CAPSNet. CAPSNet was established in 2006 with national research funding, and our goal was and has continued to be the maintenance of a population-based observational clinical dataset for gastroschisis in congenital diaphragmatic hernia. We always thought we would expand CAPSNet to other birth defects, but primarily for reasons of funding, we've not managed to do that. The database includes a prenatal component, which includes maternal demographics and risk factors, fetal imaging, and other prenatal testing, which is linked to the postnatal dataset, which collects condition-specific risk, treatment, including details of surgery, and outcomes data up to the date of hospital discharge. Because the data we collect on individual patients is what is in the medical chart and is not used to influence treatment for that patient, we are not required to obtain consent for data collection. CAPSNet is accessible for research, and we have policies for data access and publication that are listed on our website. We produce an annual report which provides an aggregate summary of the database as well as site-specific report cards with disease-specific outcome measures for that site relative to the rest of the network. Here are the surgical data collection sheets that are collected for every case of gastroschisis and CDH in the CAPSNet registry. These represent a very small proportion of the aggregate data collected for each case, which numbers over 400 variables per case. These sheets are usually completed by surgeons, although they can also be completed by abstractors who are trained to interpret operative reports. You can see that we collect details on intraoperative findings, and wherever possible, we try to harmonize data definitions with those of other registries. For example, you'll note that we use the same anatomic classification of diaphragmatic defect size that the CDH study group uses. Now, I'd like to describe the types of knowledge or evidence that can be generated from registry data. Depending on the extent of data collected, which may include prenatal and specifically maternal risk factors, studies of disease epidemiology are possible. Detailed collection of neonatal risk factors allows the derivation and validation of risk of mortality predictors, which are essential for risk adjustment in outcome studies. Registry data linked to biospecimens can support translational research, including testing of various biomarkers as predictors of disease severity. Registry websites can be sources of information for patients and families seeking to learn about a new diagnosis and can provide links to advocacy groups, private foundations, and other valuable information sources for families. A few years ago, our national media picked up the story of 3 gastroschisis babies who were born within a few months of each other to mothers who lived on the same road in a small town in eastern Canada. I was called by reporters to see if we could address the concerning question of whether there might be an unknown environmental contaminant that was causing these birth defects to occur in such a small geographic area. As luck would have it, we had just completed a study looking at geographic variations in birth prevalence of gastroschisis, which clearly identified both large and small, geographically discrete areas where gastroschisis prevalence, which is adjusted for the local birth rate, is either higher or lower than the national average. Of particular interest are the pink areas, which are high incidence areas that are immediately surrounded by low incidence areas, suggesting the possibility of a local environmental contribution to causation. The other important observation was the higher general prevalence of gastroschisis in the northern regions of Canada, which is where our Indigenous populations live. And this fits with some of our other studies which have looked at maternal ethnicity and have suggested that rates of gastroschisis tend to be higher in Indigenous mothers. Another study used a case control methodology to compare the maternal exposures of documented gastroschisis cases to geographically matched non-gastroschisis controls. In addition to the usual associations of young maternal age, smoking, and exposure to illicit drugs, the predictive model suggested for the first time an association between gestational or preconceptual diabetes and fetal exposure to antidepressant medications with the occurrence of gastroschisis pregnancy. A study from the linked Swedish health registries has studied the epidemiology of Hirschsprung disease, specifically seeking maternal predictors. This study compared the maternal demographics of 600 cases of Hirschsprung disease with 3,000 controls and found that maternal BMI of 30 or greater was associated with a 70% increased adjusted odds of having a baby with Hirschsprung disease. The CDH study group has amassed over 10,000 CDH cases, which makes this registry incredibly powerful in its ability to generate knowledge related to treatment and outcomes of CDH. This important study documented for the first time the predictive relationship between defect size and mortality, a relationship that's been also shown to associate with length of stay, duration of ventilation, and severity of comorbidity at discharge. Another study done with CDH study group registry data showed that an illness severity score, which combined birth weight, 5-minute APGAR, the presence of associated chromosomal or major cardiac anomalies, and the severity of pulmonary hypertension on initial echocardiogram, accurately discriminated patients into low, intermediate, and high risk of mortality groups. The gastroschisis prognostic score was developed from CAPSNet registry data a number of years ago and uses the visual appearance of the bowel at initial surgical consultation to create a numeric score that stratifies cases into simple and complex gastroschisis based on the presence and severity of bowel matting, necrosis, atresia, and perforation. It has been shown to correlate with morbidity, duration of TPN, and length of stay, and was recently revalidated in CAPSNet using a brand new patient cohort. It has also been validated in countries outside of Canada, including low and middle income countries like Brazil. The Japanese biliary atresia registry collects data from questionnaires which are submitted by patients every 5 years to track long-term outcomes on the more than 3,000 patients surviving up to 30 years after their Kasai procedure. Here we see the number of registry patients surviving with their native liver in blue, those requiring transplant in green, and those who have died in red over a 25-year period. Having a clinical registry that captures disease-specific risk factors and outcomes over time that can be linked to biospecimens creates tremendous synergy between clinical care and basic research. Registry-linked biobanks allow a personalized medicine approach in that treatments can be selected based on the biological profile of a disease. It also enables regenerative medicine solutions whereby a new diaphragm to treat a CDH patch recurrence could be bioengineered from the patient's own stem cells. Having a patient registry for a rare disease like CDH also allows patients to self-identify as candidates for innovative therapies as they arrive. Since clinical databases are an essential infrastructure for the conduct of randomized controlled trials, is it possible that an RCT could be conducted completely within a registry? A population-based registry which captures every case of a rare disease like CDH or gastroschisis presents a perfect opportunity for point-of-care enrollment in one or a number of potential clinical studies. The registry trial has some inherent benefits over the classically designed RCT in that it is likely that there will be fewer patient exclusions, particularly if parental choice in assignment of treatment is allowed. This offers a much more pragmatic or real-world approach to the trial, which usually makes the results more generalizable. Another registry feature that is adaptable to trial design is an a priori decision by individual sites to offer treatment A, while other sites offer treatment B. This cluster RCT design can yield results in a far more timely and cost-effective manner. There are limitations, but it is likely that the benefits far outweigh the limitations. Here's an example of a registry RCT from Sweden. Virtually every patient requiring percutaneous intervention for ischemic heart disease in Sweden, Iceland, and Denmark is part of a registry called the Swedish Coronary Angiography and Angioplasty Registry. This trial compared angioplasty alone versus angioplasty plus coronary thrombus aspiration for patients presenting with an MI. The trial proved that there was no difference in 30-day mortality between treatments, but what is most remarkable is that 60% of all patients treated, which is the red line in this graph, a total of over 7,000 patients were part of this registry RCT, with an incremental cost per patient of only $50 US. Compare these costs with the usual costs of the classically designed and funded RCT, where the per patient costs are usually in the thousands of dollars, and one can begin to understand the value of registry RCTs, assuming that the results have validity. Before we leave the issue of sources of knowledge for improving care in pediatric surgery, I think it's important to have a sense of the relative contributions of publications coming from registries versus randomized controlled trials. A few years ago, I did my own literature search for studies published with registry data, which I defined as combined data from 3 or more institutions, versus RCTs or systematic reviews of RCTs for biliary atresia, CDH, esophageal atresia, gastroschisis, Hirschsprung disease, and neck. As you can see, registry studies account for the vast majority, approximately 84% of these studies. Assuming we have knowledge generated from a registry that is actionable, what can a registry do to support the mobilization of knowledge into practice, and in doing so, create new standards for how those patients should be treated? In other words, what is the process for creating wisdom as it relates to patient care? There are 2 points that need to be emphasized that are particularly relevant for the types of guidelines we might create for pediatric surgical conditions. One is that for the vast majority of conditions that we treat in pediatric surgery, there is not level 1 evidence, nor should there be attempts to create it. This article from the British Medical Journal made the point that no randomized trial proving the effectiveness of parachutes in preventing death has ever been done. This makes the point in the extreme, of course, but reminds us that not every treatment question should be subject to RCT methods. The other important consideration is the value of standardization as a means of reducing unwanted or unnecessary practice variation. We've shown that there's great variation in how babies with CDH and gastroschisis in Canada are treated and in the outcomes they experience. This is not to suggest that practice variation is bad. In fact, it is expected, and it reflects the fact that no 2 patients are exactly alike. However, Dr. Brent James, an American surgeon and quality improvement and patient safety expert, has famously said that it is more important that you do it the same way than what you think is the right way, which I think can be paraphrased to mean standardize as much as you can, but make the most of the evidence you have. A couple of years ago, we developed a clinical management guideline for congenital diaphragmatic hernia. For those of you who may be interested, the QR code on this slide will take you directly to the guidelines, which can be downloaded to both Apple and Android platforms. Since this talk is about the value of registries, a fair question might be, could these guidelines have been created without the existence of CAPSnet? The registry didn't do the work, but rather it was done by engaged stakeholders supported by registry infrastructure and united by a desire to improve care for CDH in Canada. And we can't really talk about value without discussing cost. I've made some rough calculations of the direct costs associated with creating national guidelines, including dedicated coordinator time, the expenses associated with a 2-day consensus meeting, with the costs of publication, brings the total cost to $47,000 Canadian, or approximately 2.8 million Indian rupees. What value these guidelines will bring to our customers, who are the clinical teams that use them to treat patients, will be measured by the outcomes of those patients. It is not enough to just create the guidelines. They need to be implemented into care at the bedside to benefit patients. It's important to obtain endorsement from our care partners, including obstetricians, neonatologists, and the specialty pediatricians with whom we share care of these patients. And for those of you who are tweeters, you will have seen the power of social media platforms in pushing knowledge out to a huge audience. Point-of-care decision support tools like the checklist shown here confirm that we are following the guidelines, but also provide team awareness of how the baby is doing based on a green, yellow, red visual assessment tool. This also allows documentation of compliance with the guideline, which is an important process measure to collect and report. And finally, an expectation of any clinical guideline is that it is viewed as a living, breathing document that must be refreshed periodically with new evidence, which may modify its recommendations. In fact, we are in the process now of doing that with the Canadian CDH guidelines. Recently, we've done something similar with gastroschisis, in which investigators from 2 children's hospitals in Montreal, Canada, used their combined site data to show the benefits of a standardized approach which emphasized early closure in the NICU using a sutureless technique, with significant improvements in a number of outcomes, including reduced ventilation, and without changes in morbidity. The final care improvement feature that a registry provides is a quality assurance function by giving feedback to individual sites on their performance on key clinical indicators relative to the rest of the network. By providing individual sites with their own data, which is referenced against other sites in the network, we are providing quality assurance data which supports hospitals in meeting their standards of accreditation. But more importantly, these data give hospitals perspective on where their improvement opportunities lie, which can serve as the basis for targeted quality improvement activity. Here are a couple of examples of the types of feedback provided to individual sites by CDH study group on the left for CDH, and for CDH and gastroschisis by CAPSNet on the right. Some final thoughts. The concept of value in healthcare is a measure of the clinical outcomes achieved divided by the costs necessary to achieve those outcomes. We can use this equation to create a value proposition for pediatric surgical registries. First, we need to define the registry consumer. I would suggest it is us, the providers, who are seeking better treatments for our patients. It is also our patients who seek and deserve better care, and it is the health systems in which we work which seek cost effectiveness and sustainability. I've listed the outputs of registries in the numerator. These include the knowledge they produce and mobilize, the engagement of our care communities and patients, and the ability to provide a platform for data-driven quality improvement. Although I gave you details on the cost associated with our guidelines, I haven't talked about the cost of maintaining registries, but I would suggest to you that compared to the costs of running multiple randomized control trials, registries are cheap. A final comment about sustainability. We need more registries, not fewer, and we need the resources to support them. If the sole purpose of registries is to create knowledge, they need funding through traditional competitive research agencies or foundations, which presents a real challenge to sustainability. However, if registries are seen primarily as providers of hospital quality assurance and improvement data, they become an accreditation requirement, which shifts the responsibility for funding to hospitals. I would like to close by sincerely thanking everyone for their attention and offer my special thanks to the Indian Association of Pediatric Surgeons for this unique privilege and opportunity. I hope that there will be a time in the future when I may meet some of you in person. Thank you.
Niravi. Thank you. Thank you, Professor Ari. Dr. Sanjay, unmute yourself.
Yeah, I am Professor Skarsgård. A very, very nice talk. Considering that where we are as a community of pediatric surgeons in our country, I think these forms of registries that stretch across the country probably are much more valuable to us, in terms of improving our outcomes. And because of the numbers, it is something that will add value. The challenge here, however, as you mentioned, is the costing of setting up these things, because the system is not unified, as in a centrally run health system such as Canada. So how have other countries managed where most of healthcare is out of pocket rather than from government-run health systems?
Well, thank you very much, Sanjay, for that question, and my sincere thanks to all the attendees for staying late. I know it's late there to join me, and it's a very good question for a country as large as India, and with a non-unified single-payer health system like we have in Canada, which very clearly is an advantage in setting these things up. I think you start with what's feasible. I think you start with a network of small hospitals that you maybe have some already share patients or share information systems perhaps, and you define what you want to look at in a very pragmatic way. The reality is that the data collection part of this should be relatively inexpensive. With increasing utilization of electronic records, it is possible to create a data excerpt from the data that's collected for clinical care to be the foundational data for registry. The one critical, I think, essential requirement for registries is as somebody in a coordinator position. And that's somebody who can engage with the different sites, provide timelines for data collection, help with IRB approvals, and be responsible for the compilation of the aggregate data. And that is, I think, an essential position and skill that a registry requires. But the rest of it, the actual data collection itself, which needs to be done by the hospitals, you can't sustain this if you need a research grant to pay for data collection. But if you assume that you can get data collected for free or for cheap, and you have a few hospitals that are willing to work together, I think that's the way to start.
Yeah, I think that's very, very reasonable. Thank you very much. I hope there are other questions on the chat.
Dr. Dhruv has raised his hand.
Yeah, Dr. Dhruv. Sorry, yes, Dr. Dhruv.
Thanks, Dr. Sanjay. Professor Skarsgård, thank you very much. That was a lovely talk, and something that many of us in many of our countries probably require. And like Professor Rao has just said, that the problem is a unified computer system which sort of shakes hands with each other, and the fact that we have various types of healthcare and various modalities of healthcare delivery. Children being born in hospitals which are not essentially children's hospitals. There might be small hospitals to capture that the entire data. It becomes difficult, because I think if we capture only a small minuscule part of the data, we don't get the entire picture, and that sort of defeats the purpose of a registry of outcome study. What do you think in terms of, and you talked about technology, but what do you think in terms of while collaborating with each other through word of mouth, emails, letters, etc., collecting data on paper case registries, which will obviously have to be much simpler than say the 40-point data collection that you had in Canada. It has to be much simpler because of a busier workload and at times single surgeon hospitals. But then putting it on a system like REDCap, for example, my only question there is in terms of data protection. Because a lot of these servers are outside the shores of a country. in Canada, if this happens, would policymakers have issues with this, with the data going outside your borders?
Yeah, very much so. And we are bound by federal and provincial privacy legislation. And for example, we have a lot of difficulty exporting data. Our children's hospital is part of the NISQIP registry, and it's a real challenge to send out data that that has been sufficiently de-identified so that it doesn't risk disclosure, and yet then loses the granularity that's required to be important. If you're thinking about reporting age of a patient and you are restricted to only being able to report over a birth date over a 6-month period, and you're talking about treatments of babies, that becomes very quickly quite irrelevant data. So I do believe that there is a requirement for data protection and management of data within firewalls. There is a requirement for data sharing agreements that address some of these things between hospitals. You mentioned REDCap, and I think that really is becoming the platform that a lot of people go to just because of its usability by clinicians. So, I don't, there are barriers, and I guess you can choose to look at this as something that is, not doable because of the barriers. But I would encourage people to start again with an idea that you can start small with 2 or 3 partner groups that are interested in doing this. Data from 3 hospitals is going to accumulate more quickly than data from 1. And, and that should be thought of as the value. I think there's 2 fundamental values. One is this amplification of data by registries and sites and site sharing. And the second, perhaps most important one, is the collaboration and partnership that's created by sites that are sharing data. And I think that should not be underestimated in terms of its value in amplifying the message that comes from what would otherwise just be a single site.
I agree with you. And just to mention softly, anesthetists, they seem to have done a very good job in Africa with the Africa Surgical Outcome Study, which was by word of mouth. And so yes, it is possible. I think the Lancet study that came out for outcomes in neonatal conditions was probably conducted in 2 or 3 hospitals in India, but you could amplify that data to sort of suggest things. But in terms of registry and final outcomes, I think we will have to find a way in India for everybody to sort of become slightly uniform and shake hands on what we're going to do.
Mm-hmm.
Thank you.
Thanks, Drew. Thank you very much. Any other questions from the audience, or?
Can I ask one question?
Please go ahead, Shilpa.
Actually, I tried to make a registry 5 years back. I hired a website, and for 3 years I collected data and I made it open to all. But it was very difficult to motivate others to put in their data. I also made it parent friendly. So that was also not so easy because they don't understand the language. Somebody has to bring them up and make them fill it. And finally, all the data goes to the website. So the website has to give it back to you. And it's not an ever ending thing. Like how long can you keep paying it? So how should the things be done? If you make it website friendly, others can enter data easily because it's open to everybody. But if you keep it hospital based, it's restricted to those 1 or 2 hospitals. And you don't get what you need. You don't get the raw data. So how should one go about it?
I guess I would start by who you think are the essential partners in data use. So your essential stakeholders. And I think, Shilpa, that you've highlighted a very important consideration for future design of registries, and that is how do patients fit into the registry? If you're looking for somebody who's incentivized to put their, to collect their data and to share it, particularly if it's going to personally, create opportunities for improved care outcomes, it's patients. And so I think, the word patient-powered or patient-driven registries is really apt. And, if you think about, the fact that we have to pay people to collect data, patients and parents will often do that for free, and they'll import the data. And then I think at that, if they are entering the data, you've already addressed some of the privacy concerns, because obviously there would be consents required that they would sign to be participants and partners. So I think that, wherever possible, we should be looking to patient engagement in the design of these registries, because that's really, I think, is going to be the future. And, with the increased attention to patient-reported outcomes, because one of the early things that the group has to do is decide what data they're going to collect, what outcomes they're going to look at. And I think to include patients in those decisions is critical. So thank you for that comment.
Any other questions? Any other comments? Yeah, I think it also boils down to an extent to funding, as I think this has been repeatedly coming up. And I think this is something that, as you say, is a challenge, and that's something that we need to address from our side, starting small amongst like-minded institutions and then trying to expand it based on what preliminary information we gather. And I think that's a very valuable lesson that you have given us, and it's something that we will follow through. So Professor Skarsgård, thank you very much for your presence here and for sharing your expertise. And I think it's been a 1, 2, 3 Canada for the last 1.5 hours, and it's been extremely thrilling, from laboratory to vesicles to more things that we can relate to in the form of clinical registry. So thank you very much. And I'll hand over to Dr. Mahajan. Thank you. So
Professor Eric, thank you so much for your, I can't see. So thank you very much for your wonderful, wonderful lecture. As everybody pointed out, data collection has been the bane of Indian medical system. We have a lot of data, a lot of good work is also being done, but there's no data collection. So with your lecture, everybody will take inspiration, and maybe we are able to put a system in place for clinical registries. So as we discussed earlier also, this is the area we are actually lacking, and your lecture will definitely give insight to people to start working on this particular aspect. I can't thank you enough because it's very early in Vancouver. It's, I think, 7 o'clock, and you have taken all the pains to get up early and be with us. So thank you very much on behalf of Research Section of Indian Association of Pediatric Surgeons. This is small token of appreciation and our gratitude for your presentation.
Thank you very much. And my sincere thanks and appreciation to all of you. It's really been my pleasure to join you this morning in Vancouver. It's not that early. Thank you again.
Thank you very much.
Thank you. Thank you.
Bye, everybody.
Bye.